champions out there. I believe every one of you who is watching this video is doing absolutely good. So my dear children, for those of you who do not know me, my name is Jasmine Joy. I teach biology and I am here for a secret mission. Not so secret anymore. It's a combined mission as well. You are also, it's actually secret for you because you are also involved over here. So what's our mission? Our mission is to complete molecular basis of inheritance. My dear children, this has been a chapter that have been torturing kids since ages. Okay, when I was in 12th, it used to torture me also. I was like Mr. Bean like this. Okay, I cannot do it. I don't understand anything. Right, this is how I was. But I will make sure that you are not going to be like this. That you are going to grab all those 13 marks that the chapter gives according to the second PUC blueprint that was released yesterday. So second PUC blueprint says it gives 13 marks just like the previous year. You'll get one 1 marker, one 2 marker and 2 5 marker questions out there. So basically, if at all you're able to crack it, 13 marks pakka in your second PUC. And I promise you that you are going to get your bestest marks in your second PUC if at all you complete this lecture. I'm keep putting a thing. If at all you complete this lecture completely, okay? Next thing, all your concepts are going to be super clear for your KCET. So whether it gives you a 5 marker question, because it have been giving 5 marker questions, sorry, 5 questions throughout the years, but now recently for 3, that is a shift, that is what we are seeing in molecular basis of inheritance, but still is a very important chapter in terms of KCET. So I'll make sure that your concepts are super clear for KCET or any other competitive examination that you have related to biology. Cool. And at the end, there's one more promise as well, that you are going to be the happiest for a reason. Listen to me. When you invest time somewhere and when you feel that at the end of it, you're able to solve questions, you are able to understand the concept, you're able to teach it to others, don't you think you're going to be the happiest? Yes, and I promise you all of it. And my dear children, I do not want to waste even an inch of your time, so I am just going ahead with the content. And thank you for showing so much love, showing so much of love for the one shots. I know the views are not too much, but I'm seeing your comments, and I know the views are going to come when it's exam time, because that's when you actually study. But I really want you to know that once, if you feel that the chapter is not clear, where should I go? You should know Jasmine Ma'am is here with her one shots. Okay, she might have already done it. You can come and watch it. Okay, and if at all you love the one shot, please give me some motivation by putting it down in the comment section that, ma'am, yes, the explanation was really good, or ma'am, this you have to improve. Okay, so all of this I expect from you guys. I also need a little amount of motivation for talking for 3 hours straight, right? And I think probably you are that thing for me. Okay, you are, my dear children, you are someone who makes me go crazy around teaching, and I really want you guys to be the happiest. That's all. So let's go. Okay, so what we're going to do is we are going to have some questions and I am going to answer that question, and that is how we are going to go ahead with the concept. So the first question is, ma'am, I know it's out of context, but I heard viruses also have DNA. Is that true? So listen to me. Virus is also an organism. It also needs to function, which is why it needs a genetic material. Okay, it needs to make sure that everything is being coordinated really well, which is why it needs a genetic material which says that you have to do this, you have to make this, you have to do this. It's not the brain. Basically, genetic material is responsible for each of the proteins and everything that is going to be made. So yes, you need a genetic material. Okay, so what I have done over here is I have put questions and I am going to give you answers related to those questions, and that is how we are going to cover the concepts. So the first question is, ma'am, I know it's out of context, but I've heard that viruses also have DNA. Is that true? So listen to me very carefully. Virus is also an organism. Yeah, and it also needs someone to say that, hey, Macha, you have to form this protein, you have to form this protein, otherwise it will not work. So yes, it has genetic material. Now that genetic material can be DNA or it can be RNA. Ma'am, are you teaching molecular basis of inheritance? Because as of now, why are you talking about viruses? Because in your NCERT, the first conceptual part that has come is on the basis of the organisms and their genetic material, how much genetic material that they have. Ma'am, is that very important? Yes, because it has given a KCET question, so we need to know about it. So in those organisms that were there, I found certain viruses also, which is why I thought I should be starting with that question. So this thing, bacteriophage 5 X174 is what? Actually not a bacteria, but a virus that affects the bacteria. It's a virus, and you know what? It has the least amount of genomic size. less, very less genetic content out there. It's just 5386 nucleotides. Just understand it is going to be really, really less. Then we have bacteriophage lambda. 5 you call it. Okay, so not bacteriophage lambda is there, not 5, sorry. Bacteriophage lambda, it has 48,502 base pairs. This is the second position. This is the one that is bagging the second position. Then these were what? Viruses that were eating bacteria. Oh, sorry, that were affecting bacteria. Now we have the bacteria on the third position, which is known as, it's a famous bacteria, it's known as E. coli, or you also call this as Escherichia coli, which has a genomic content of 4.6 into 10 to the power 6 base pairs. Now I know this base pairs, nucleotides and everything might be very, very new to you, but that's not something that probably you have to be stuck with. Okay, we have to know that which one has the least and which one has the highest, and somewhere here we need to know the numbers also. Okay, so this is a 4-digit number that we have, the bacteriophage 5x174, which is there, just has 5386 nucleotides are there. That means it is going to be very less. Then we have bacteriophage lambda with 48,000. Okay, then we have Escherichia coli. So it was 1,000, it became 10,000. That is how you can remember if at all you want. Then we have Escherichia coli. It has 4.6 into 10 to the power 6 base pairs. Now, ma'am, you're talking about all of it. Okay, what about us human beings? We are not in the list, huh? We are also there in the list. We are the ones who has the highest amount of genomic size, largest genomic size. In our body, Baba, if at all we talk about a normal cell, for example, I take my cell over here and I do some experiments and I check it under the microscope. I will be having nucleus in my cell, yes. Inside the nucleus, I will be having my genome, that is the DNA, okay, genetic material which is there. I have 6.6 into 10 to the power 6 base pair DNA. It's very long. Just understand, it's going to be too much. It's going to be too much. So if at all I want to stretch it around, it is going to be 2.2 meters long. 2.2 meters long. Just imagine that much amount of genomic size I have within my nucleus. Okay, it is 6.6 into 10 to the power 6 base pairs. I was talking about a normal cell. What about, okay, in human beings, you need to understand there are two types of cells. Basically, one type of cells which are there, they have the normal number of chromosome. You also call this as diploid amount of chromosome. That is 2n. Double the number of genetic material out there. That is something that is found in each and every cell of our body, except a few. So we're going to talk about that also. That thing that I'm talking about, which is an exception, is going to be what? Is going to be the gamete. Is going to be the gametes, you know, sperm, ovum. They cannot be 2n. They cannot be diploid in nature. They need to have half the number of chromosome. Why? Because the sperm and the egg has to come together, n and n has to come together, and then it becomes 2n, and that is how all the total body is going to be formed. Okay, so we are going to do some questions, and that is how we are going to understand the concepts. Okay, so we are going, my God. Okay, so what I've done is I have put some questions over here. Just imagine that you are probably asking me those questions, and then I am going to give you answers for that. That is how we are going to cover each and every concept. So here I have a question. Okay, ma'am, I know it's out of context, but I heard viruses also have DNA. Is that true? So listen to me. Virus is also an organism. Okay, virus is also an organism. Definitely, this organism needs to make proteins to survive, because proteins are very important for making up the body of the organism and stuff like that, right? It's very much important, also for giving much more information. For making other things, they are going to be building blocks and things like that. So yes, you are definitely going to need some genetic material. It can be DNA, it can be RNA, but the question is that you are asking me, viruses, they have DNA, is that true? So some viruses are there who have, RNA, some are there which have DNA. But ma'am, the next question that I have over here is, why are you talking about viruses and DNA over here? Because that is the first concept given in your NCERT from which a KCET question has come. Okay, so what they have done is, they have given you a list of organisms. They have said that this organism has this much amount of genetic material, and what you have to do from your end is that you have to remember it. Got it. And as I said, a KCET question has come from here, which is why I started off with this. I know that does not feel like molecular base of inheritance starting, but yes. So listen to me very carefully. These are 1, 2, 3, 4, 5 organisms that are given over here. This is the one that has the least amount of genetic material. This is going to be the one that has the highest amount of genetic material. So let's see the first one over here. It's known as bacteriophage 5X174. Bacteriophage 5X174, very big name. But you know what? It has very less genetic material within it. It's just 5,386 nucleotides. Now you do not know nucleotides. You do not know what is base pairs. You do not know anything else out there. So it's okay. You just know the number. You just know the number. You know that it has the least. What is it? Bacteriophage 5X174. What is this? This is a virus, not a bacteria. It's a virus that affects the bacteria. That is why it is known as bacteriophage. It is basically affecting the bacteria out there. Now similarly, its cousin is also right up over here. You know, cousins are always mostly same. Bacteriophage lambda. Okay, that was 5 and this is going to be lambda. It says that I have, okay, Macha, you have to be really careful. I have more genetic material than you. Okay, I am obviously the boss. So 48,502 is the base pair. So you can just imagine this is in thousands. This is going to be in 10,000. I mean, that much difference is there. So 5386 is the number of nucleotides that you have over here. It becomes 48,502. That means if the number is increasing, I can just assume that over here the genetic material is more, right? Now next thing that comes in the list is Escherichia coli. Who is that? This is the bacteria that these guys were affecting, is it? So listen to me carefully. These were the viruses. Then there is a bacteria which says that even though you eat me, okay, even though you affect me, not eat me, affect me, I want to tell you, I am leading the list. Okay, I'm leading the list. It has almost 4.6, please remember, into 10 to the power 6 base pairs. Okay, everything is 666. 4.6 into 10 to the power 6 base pairs is there as a genomic size of Escherichia coli, which is a very famous bacteria. It's actually a VIP person. Okay, it's actually a VIP person. All the experiments are done on E. coli only. Now, everywhere human beings are there. We are parasites, right? Even in this list, competing with the virus and bacteria, we are over here saying that, Macha, you have this much. We have more than that. We have more than that. So in human beings, if at all you see, I have written human haploid genome, human diploid genome. What do you mean by that? So, Baba, listen to me very carefully. You and me, if at all I take a normal cell of our body, normal cell, what do you mean by that, ma'am? Abnormal cells are also there? No. There are two types of cells. One are known as gametes, okay, gametes, for example, your sperm, the egg and things like that. And the other cells which are there, I'm talking about them, they are the normal cells which are there. Those normal cells that we have in our body, they are going to be diploid in nature. That means they have 2 sets of chromosome. Okay, they have 2 sets of chromosome. Okay, and over here in the case of gametes, they are going to have half the number of chromosome, only 1 set of chromosome. Why like that? Why partiality like that, ma'am? Listen to me. The sperm, if at all it carries half the number of chromosome, and the egg, if at all it carries half the number of chromosome. Okay, if it carries half the number of chromosome, n plus n becomes 2n. It will be just like, okay, this zygote is going to be formed, and that zygote will be having a normal cell like all of us. That zygote is going to become a human being. I mean, zygote is going to develop into a whole human being like this, right? So it should be having normal cells all throughout the body. Now, whenever in that person's body gamete is going to be formed, again it will become something like this. That is a whole different thing, right? Now I am telling you something like this. If I say the half content is known as haploid content, and if I say the double content is known as diploid content, and I give the number over here, the haploid content is 3.3 into 10 to the power, 9 base pairs, into 10 to the power 9 base pairs, 3.3 into 10 to the power 9 base pairs. What is going to be the diploid content? What is going to be the diploid content? 3.3 into 10 to the power 9 base pairs from here, 3.3 into 10 to the power 9 base pairs from there. So it becomes what? I can just multiply into 2. It gives me 6.6 into 10 to the power 9 base pairs. You get it? So this is the diploid content that we have in a normal cell. This is the haploid content that we have in the case of gametes. This is the logic that we have. So we have the most content over here in diploid, then haploid, then we have Escherichia coli, that is a bacteria, and the people who eat the bacteria, that is bacteriophage out there. Okay, bacteriophage 5 is the first one. 5 first, okay, is the first one to have the least amount of genetic material out there, and with that I can probably try to solve this particular question. This came in KCET 2017. Let's try to solve it. Match the number of nucleotides of genome of column 1 with organisms of column 2 and choose the correct options given below. 5, 3, 8, 6. I don't know. Right now, though I know. Okay. It is going to be 5x174 bacteriophage. 5x174 bacteriophage. Cool, cool. Now, I have a suggestion to you. Whenever you match the following question, try to solve the A and the last one first. Okay, I'll tell you why. If at all I'm able to apply that, let's check that. 3.3 into 10 to the power 9 base pairs. This is what half the content that we have. Half the content that we have. Okay, and there is no 6.6 over here, and there is only one man over here. There is no haploid and diploid man, so D goes with Q. A goes with S, A goes with S. I have 3 options. I'll cancel this option, elimination method. Okay, and D goes with what? Q. D goes with Q. I just have one option and I've already found my answer. I didn't even have to match the other things. I've already found my answer. Now it's time to recheck. Rechecking you have to do, okay? Now, if you talk about 48,502, that is going to be there in what? Bacteriophage lambda. Bacteriophage lambda, 4.6 into 10 to the power 6 base pairs, it is going to be E. coli. Here everything is 6, please remember. 6, 6, 6, 6, 6. Okay, here is going to be 3.3 into 10 to the power 9 base pairs. Remember that in the case of human beings. That is how we are going to solve the question, and we have done it, and Baba, we have got 1 mark in KCET already. Done. Clear? Easy, right? Yes. Now we are going to go ahead with the next thing. Ma'am, how does our DNA look? Cute enough, huh? It's very cutesy, or not so cute. So listen to me very carefully. We are going to go ahead with the next concept. That is going to be what? The structure of DNA. Similarly, we have 13 concepts. You are on the second one. Okay, structure of DNA. Now over here I have written so many things, but I want to use this particular slide to explain you things, and you have to be very, very careful while I talk about it. Okay, so the first thing that a DNA would be having, so DNA is made up of 3 things. We are going to decode one by one. This thing that I have over here is a pentose sugar. What sugar? Pentose. What do you mean by pentose? Pentose means it has 5 carbons. It has 5 carbons. Okay, where is the first carbon, ma'am? I don't see anything. So here is going to be the first carbon, here at this position. This is second, this is the third one, this is the fourth one, and the fifth one says, Macha, it's too hot inside, I am going out. Okay, so this is going to be the 5th carbon. First, second, third, fourth, and 5th. Oh, pentose sugar, that's all, ma'am? Yeah, that's all. For now, that's all. Okay, the next thing that it has is going to be a nitrogenous base. Nitrogenous base. And where is the nitrogenous base attaching? On the first carbon. It says that nitrogen base, I want the first position. Okay, then you take it. You have the nitrogenous base right over here. Now, I want to stop you right over here, and I want to tell you something. When a pentose sugar goes and combines with nitrogenous base, you have a name for this structure. You call this as a nucleoside. What do you call it? A nucleoside. Side. Okay, nucleoside is there over here. Cool, cool. And this particular bond, obviously it's not in air, there is a bond over here. It also has a name. You call this as N-glycosidic linkage. Where is it? You call this as N-glycosidic bond. Okay, so what do you have? Pentose sugar is there, 5 carbon sugar, and nitrogenous base is there. Ma'am, this bond is known as N-glycosidic bond. It's on the first carbon and the nitrogenous base. And basically over here, this structure which is being formed when both of them combine together is known as a nucleoside. Okay, done. Now, one person is over here who's super jealous of everything happening like this. Okay, and do you know what is that person? Who is that person? It's going to be the phosphate group. It says that, okay, you did all of it without me, huh? Fine. But then the sugar says that, Macha, don't be sad. Don't be sad. I am going to be there with you. Okay, I am going to be there with you. Don't be sad at all. Let me make a bond over here. First position, it's okay, NB. 5th position. Okay, from here, if at all we start looking, it will be the first position. You can think like that. So this is going to be the phosphate bond that we have. Okay, sorry, not the phosphate bond. This is going to be the phosphate group that we have. So I have one more person over here. This is going to be phosphate group. So it says that until now you were known as nucleoside. Now I do not want that to happen. Since I am also there, I want to change the name. Now when this and this comes together, all 3, 1, 2 and 3, all of them, they come together, you will not call it as a nucleoside now. Rather, you will call this as a tide, nucleotide. Tides are bigger, no? Tide. Okay, it is going to be bigger. So nucleotide is being formed over here in the combination of all of 3. And now, ma'am, I have a question. You said that this is known as N-glycosidic bond. What do you call this particular thing as? What do you call this particular thing as? Baba, by the way, what is it attaching to 5? No, the 5 carbon over here. There's a carbon, okay? Here also there's carbon, carbon, carbon. This is the 5th carbon that we have. The 5th carbon is attaching to the phosphate group. And over here, there was an oxygen also in between. Okay, so you call this particular bond as, that is not visible over here. Let's just forget it. But anyway, you have to remember the name of this bond. It's known as phospho. Since phosphate group is there, ester bond. It is known as phosphoester bond. Phosphoester bond is what you call it. So we have N-glycosidic bond and we have phosphoester bond right over here. Then we have formed a structure that is known as nucleotide. Cool. Now, why do we need nucleotide over here, ma'am? And by the way, DNA is not at all cute. Okay, by the way, DNA is not at all cute. So before I go ahead and say that why do we need nucleotide and what is happening over here, I want to or probably I shall say it later. Okay, fine. Then let's go ahead and let's see why there is a use of nucleotide over here. So what happens is that when you want to form the DNA, what are we made up of? We are made up of small cells. Cells comes together to form tissue come together to form organs. That is how it goes, right? Similarly, nucleotides, they have to come together to form a strand of DNA. Okay, so if you have something like this over here, similarly, you are going to have another one right over here. Another one right over here, and I'm going to make that first carbon, second carbon, third carbon, fourth carbon, and the 5th carbon. The 5th carbon is doing what, ma'am? Obviously making bond with a phosphate. Obviously making bond with the phosphate. What about the first carbon? The first carbon is making a bond with the nitrogenous base. Okay, done, deal, that's not an issue. Now, it cannot be in air. If at all I say that these things are making up DNA, they obviously need to combine at some point. What is the point? Can they combine over here? No. Phosphate, which is said, is already disappointed that this guy came in the first position. So it wants to make sure that I do more such that the pentose sugar likes me more. So basically, it says that you cheated me by going up there and pairing with the nitrogenous base out there alone. I am also going to do something like this crazy thing. So it is going to make a bond over here with the pentose sugar of another nucleotide. But this time it's going to be the 3 carbon. This is the 3rd carbon which is going to make the bond, and let me tell you again, oxygen has been involved over here also. In this particular situation also oxygen has been involved. So again, what do you call this? Same bond that we have, right? I will call this as phosphoester bond. I am going to call this as phosphoester bond again. Now if at all you only think 2 phosphoester bonds are there, and if you have been asked what is that connecting linkage between 2 nucleotides, this is a nucleotide, this is another nucleotide, what is that bond that you have between 2 nucleotides? What can I say? This is phosphoester, this is phosphoester. Can I say it's a phosphodiester bond? Di means 2, right? Diester bond. Yes, Baba, you can say that. Phosphodiester bond is between the 3rd carbon of one pentose sugar and the 5th carbon of the next pentose sugar, and in between that you have the phosphate group. What do you call this as? I'll use a different color altogether, and I do not have too many colors, so I'll use red. Okay, phosphodiester bond. This is known as phosphodiester bond, and that is the one that links between 2 nucleotides. Ma'am, I have understood that. Okay, now same way, so many nucleotides are going to come together. Yes, so many nucleotides. Suppose just imagine over here you will be having so many nucleotides. Okay, that is how it goes. So there will be one line of DNA that is going to be formed. It's going to be one line of DNA that is going to be formed. Now listen to me very carefully, very carefully. Okay, if I say that this is going to be the one line of DNA that is going to be formed, here what do I have? At this particular end there is not going to be, there will be one end, no, at the end there will be one end. One has to stop somewhere. When it stops, this is something that you see from the top. What do you see? This is what you see from the top. So can I say that this end can be named as the 5' end? Why? Because at the top I see the 5th carbon with the phosphate group. Okay, so I'll call this end as the 5' end. Okay, what do you say? Now this end also, this end does not have 5' end? No, it does not have 5' end because 5' end is at the top. What does this have in the lower part? Any guesses? I'll tell you. So basically the 3rd carbon over here had an OH group. had an OH group. You usually do not make it. Okay, while you make diagrams like this, as I said, oxygen was there and the oxygen was there and the bond it went off. That is a whole different thing, but it was there, right? So here also you had it, but now you had made a bond, which is why this is not visible. So you have an OH group over here on the third position, and this is going to be there in the lower side. This is going to be there. If at all I think of it as a thread, at the top I have the 5' end with the phosphate group saying hi. At the lower end I have the 3-OH group, which is waiting for some other nucleotide to come, but there is no nucleotide, so it has made a strand. Made a strand. So this is going to be known as the 3' end. A DNA has a 5' end and a DNA has a 3' end. This is just a single strand, ma'am. What I have seen at the diagram of DNA, it looks like this. The diagram of DNA that I have seen, I have studied, it looks like this. So basically there are 2 strands. Yes, there are 2 strands. And for making those 2 strands, you need one more thing like this. You need one more thing like this. Okay, so just imagine over here you have one more strand like this, ma'am. Okay, I have just imagine that this something like this is repeated like this. So what is going to happen here? It is going to be what do you say? Yeah, there's a nitrogenous base, and basically here is the phosphate group. This is how it looks. There also it is going to be looking like this. There also it is going to be looking like this. This phosphate that we have over here is already engaged. Okay, this phosphate is already engaged because it is making the bond between the nucleotides. Okay, and if at all we want both the strands to be together, if at all we want both of these strands to be together, we have to make sure we do something. So listen to me, there is a bonding between both the strands, and that can only happen, okay, that can only happen if at all, Baba, instead of, suppose this was the nucleotide that we have over here, which is having nitrogenous base on this side and the phosphate group over here. This phosphate group is attached to another, what do you say, another thing like this, right? So phosphate is not free over here, and you have nitrogenous base on that side. The only thing that you can do is flip it up. Okay, the only thing that you can do is flip it up, but that does not work that way. That does not work that way. So what are we going to do is, if at all a strand was like this, okay, I'm going to make it like this. I'm going to make it ulta. I'm going to make it ulta. When I make it ulta, whatever was that side comes this side. Whatever was that side comes inside. So basically now what is going to be there is that your nitrogenous base, which is there in the next strand. In the next strand, suppose there is a nitrogenous base over here, it is going to come inside. It will be coming inside. But the thing is that it was like this 5' to 3' over here, but in this particular direction it is going to be what? It is going to be what? 5' here, and the 3' will be at the top. It's ulta. Why are we doing this? Such that the nitrogenous bases over here, they make a bond between each other. They make a bond between each other, and such that your DNA which is there, basically both of the strands, they stay together. That is why we did the whole drama. That is why we did the whole drama of doing things. So I have a term for it. You say that the DNA strand is double-stranded. The DNA which is there, it is going to be double-stranded, and not just that, they're going to be anti-parallel. They are parallel, but they are anti-parallel as in one is going to be there. Okay, one is going to be there, which is going to be in 5' to 3' direction. The other one which is there is going to be opposite. It's going to be 3' to 5'. Why? Because I want the nitrogenous bases to point inwards, such that I can make bond over here. What is this bond? This bond is known as hydrogen bond. You call this bond as what? Hydrogen bond. And depending on the types of nitrogenous bases you have different type of hydrogen bonds out there. So there can be 2 type of hydrogen. There can be 2 hydrogen bonds, or there can be 3 hydrogen bonds. Not types of bonds. There can be 2 or 3 hydrogen bonds out there. Got it? And that is how your DNA double strand remains intact. Otherwise, there is no chance for it to remain intact. Got it? So what have we studied until now? Not to confuse you, but to make sure that we have reviewed everything. Okay, let's start. The first thing that we studied, ma'am, is that DNA has a pentose sugar, 5-carbon sugar. Okay, and it pairs with a nitrogenous base over here. It makes a link over here. You call this as N-glycosidic bond. Okay, this bond is known as N-glycosidic bond. Together, pentose sugar nitrogenous base is known as nucleoside. Now one person becomes super jealous and says that I also want to make a bond. Okay, so phosphate group comes and makes a bond over here. Now this whole family is known as what? Nucleotide. It is known as nucleotide. Okay, okay. Now this bond that we have between a carbon and the phosphate group, the fifth carbon and the phosphate group, is known as phosphoester bond. What do you call it? Phosphoester bond, because over here phosphate and oxygen and everything is being involved. Phosphoester bond is what you call it. Okay, now you have a nucleotide. Similarly, you have one more nucleotide. And to make sure that both of them are attached together, you have only one thing protruding out, and that is what? Phosphate. That is phosphate. So this third carbon which is there, okay, it also comes and attaches with this phosphate. Again, the same type of bond. Over here also oxygen is being involved. Over here also oxygen is being involved. Actually over here you had OH group. OH group. Okay, C was bonded to an OH group, and that is somehow being dissolved out there. Okay, and that's how you form the phosphate. That again you call this as phosphoester bond. So if at all I have been asked, two nucleotides are combined together for what bond? Phosphodiester, because two bonds are being there. Okay, okay ma'am. Now you spoke about one more thing ma'am, that is DNA is not single stranded, it is double stranded. So for both of them to combine together, we have to make sure the nitrogenous basis of both the strands, they are pointing towards each other. Which is why if one strand is in 5' to 3' direction, 5' means this thing which is not bonded to anything. This is the 5' end. Okay, and over here in the lower portion you will be finding the third carbon not bonding with any phosphate, because there is nothing below. So the OH group remains intact. The carbon's OH group remains intact. So you say this is a 3' end out there, which has the OH group. Here you have the phosphate group, here you have the OH group at the end. Okay, okay. Now when you make a double-stranded DNA, what is going to happen? As I said, to make it point inwards, we're going to flip the strand. We are going to flip the strand such that it points inward. That is why we are going to have anti-parallel strands. Got it? Got it. Now listen to me carefully. This pentose sugar which is there over here, it has a name. It actually has a name. Okay, and what do you call it? You call this as, suppose it looks like this: first carbon, second carbon, third carbon, fourth carbon, and fifth carbon. We are not making what all it joins with or what all it has and all, but initially it used to look something like this. The second carbon had an OH group. This pentose sugar on the second carbon position, it had an OH group. In this case, I'll call this pentose sugar as ribose. I have a name for it. I will call this as ribose. Okay, ma'am, is this the sugar that we have in DNA? No. In DNA, we have something different. We are going to have something different, and I'm going to make that over here. In DNA, we have again a pentose sugar only. First carbon, second carbon, third carbon, fourth carbon, and fifth carbon. But over here, you need to understand, okay, instead of having OH, this O has been removed. This O has been removed. So what do you call this as? Deoxy. Deoxy means oxygen is gone. Deoxyribose sugar. The name of this particular sugar becomes what? Deoxyribose. So ma'am, this is a sugar that is there in the DNA. Yes, that is why you call this as, you call the DNA as deoxyribonucleic acid. You call this as deoxyribonucleic acid. That is a full form of DNA, because it is made up of deoxyribose out there. And this thing, what is the use of this thing, ma'am? It is used to form the RNA. RNA is what? Ribonucleic acid. Okay, ribonucleic acid. So in that, this particular sugar is being used. This particular pentose sugar is going to be used. Always remember, in DNA, deoxyribose. In the second position, you do not have the O. Ma'am, why is that so? Listen to me. Our ribose sugar, which is over here, which is having an O group, is someone who is very much, what do you say, you might have seen people who go and talk to everyone. Okay, they are very excited. They are happy-go people. So they go talk to everyone. But the problem is that, okay, they can be probably, when they talk to everyone, they know a lot of things. Now they're stressed because they know a lot of information out there. There could be a lot of things out there. In general, I want to say that the OH group over here makes the sugar, or makes the RNA molecule, okay, not the sugar, but the RNA in general. RNA is made up of this ribose, very, very reactive. It is going to go and react very much. It is going to go and react very much. Okay, whereas when you remove that O, it becomes more stable. And we want that. We do not want our genetic material to go react with everything, because that can bring changes in us when we fight. Okay, just think. Okay, ribose is the one that fights with everyone. Deoxyribose is the papa one. Does not fight too much. So when we fight, even if we win, we lose. Even if we win, we lose. Why? Because we have hurt someone, or there might be some sort of damage. We have used our anger. This gives a very negative emotion, right? So same way over here, what happens is when this guy is not involved in any fighting, any changing, any reaction out there, this is in peace. But this is not in peace. It can be changed. That is a big problem. That's a very big problem. We need a genetic material to be stable. I hope this is clear. So what type of pentose sugar do we have? Deoxyribose. What is not there in deoxyribose? Oxygen is not there in the second position, and that is second carbon position. Got it? Got it. Cool. That's all. That's all. Now we're going to go ahead and probably we are going to study more about it, but let's cover this thing that we have studied so far. Okay, a nucleotide has 3 components. Okay, nucleotide which is there has 3 components. One is going to be the nitrogenous base. I'm not reading anything right now. Okay, then is the pentose sugar, deoxyribose in DNA and ribose in RNA, and there is a phosphate group. Joins to the 5th carbon, makes the molecule acidic. This particular phosphate group is the one that makes the molecule DNA acidic. Always remember. Okay, DNA is known as acidic thing. Okay, because it is negatively charged. This negative charge comes from the phosphate group itself. Okay. Okay, now listen to me carefully. We were speaking about this particular thing, this whole thing, and I told you nitrogenous bases which are there, they have to come together, they have to form the bond. That means that nitrogenous bases over here are super important for us. Okay, nitrogenous bases over here are super important for us. And always understand, nitrogenous bases which are there, they are of 2 types. They are of 2 types. One is basically, I'll give you the trick to remember this, okay? One is basically the purines, and the other ones which we have is going to be pyrimidines. Pyrimidines. Purines and pyrimidines. In purines comes A and G. A, G, O, G. It's actually adenine and guanine. Adenine and guanine. A and G. Okay, in pyrimidines comes thymine, you call it as T, here A, here G, and apart from that you have uracil, and the last one that we have is going to be cytosine. Okay, so guanine, adenine, guanine, thymine, uracil and cytosine. Over here, why are we talking about this? Okay, listen to me. Adenine says that I am going to base pair. I am going to make a base. I'm going to make a pair, a hydrogen bond that is there, only if there is thymine on the other side. Otherwise, I'll not make the bond. Otherwise, I'll not make the bond. So, adenine and thymine are couples. Okay, and they have what? How many hydrogen bonds? They have 2 hydrogen bonds between them. 2 hydrogen bonds. How do you remember? T for thymine, T for 2. Okay, okay. What about guanine and cytosine? Guanine and cytosine, G and C almost look some similar, no? So they are very similar, which is why they have 3 bonds. Hydrogen bonds only, but they have 3 hydrogen bonds. Now you have to imagine one thing, okay? Suppose this is a DNA strand with A, G, C, T, and it goes on. And I want to make a new DNA strand. I had a single strand DNA and I want to make a new strand of DNA. What is going to happen? The nucleotide containing thymine as a nitrogenous base, the nucleotide, pentose sugar, containing thymine as a nitrogenous base is going to come and make a pair over here. Okay? And make a pair over here. So we had made, suppose, why is not working? Okay. We had made a DNA which is this much, which is going to be this much. Then another nucleotide comes which is having what? Cytosine. G base pairs with C. Then another nucleotide comes which will be having guanine because C base pairs with G. Then another nucleotide comes which base pairs with what? A. So that is how things are going to go. That is how things are going to go, and that is how basically your double strand is going to be made, if at all we have to make it. So we as of now know one thing, that nitrogenous bases, ma'am, are of 2 different types, purines and pyrimidines. Purines are A and G and pyrimidines are T and C. And there is uracil, ma'am. You didn't make uracil over here because, listen to me carefully, DNA which is there has thymine, does not have uracil. Uracil which is there is basically something that you find only in the case of RNA. In the RNA, you do not have thymine, rather you have uracil as a nitrogenous base. In RNA, they do not know what is thymine, they just have uracil. Okay, cool. And I'll tell you what's the difference between a uracil and a thymine, and who came first and things like that also. Just give me a second. I had a picture, where is it? Just give me a second. Over, come on, yeah. Here it goes. If you see this particular picture, okay, this is thymine and this is uracil. This is nitrogenous base. This attaches to the pentose sugar, if you remember, right, via the glycosidic bond. So this is thymine and this is going to be uracil. If at all you look at them carefully, what is the difference that you can find? OO, NH, NH, OO, N and C1 sugar, that is all there. One thing that is different is, over here you have a methyl group. You have what? A methyl group. CH3 is known as methyl group. This is something that is different in thymine, DNA. Okay, so ma'am, what are you trying to say? I am trying to say that this guy was the first guy to be there. Uracil was there already in the gang of pyrimidines. They were there. Uracil and cytosine, both of them were there in the gang of pyrimidines. That was when you had RNA. Okay, that was when basically we're talking about RNA. Now we know RNA had a lot of issues there. It has one more O group, which makes it highly unstable. Do you think I can let go of that particular fact and make it the genetic material? No, that is very difficult. I cannot make RNA the genetic material because it had one more problem. The problem was that uracil also was not that good thing, not a good pyrimidine. Okay, not a perfect one. To make it more stable, okay, to make it more stable, we added a methyl group over here, and now we got thymine, which is going to be more stable. Now DNA is something that is considered as a good genetic material. That is why we say that in the case of DNA, thymine is being used, or basically I can say DNA is considered as a good genetic material. Why? Because already it used a deoxyribose sugar. Apart from that, it said that uracil, you move whenever a person does not work properly, you lay them off, right? That is how it usually works over here, right? So you lay them off, and now you have a new nitrogenous base coming over here, which is thymine, which has an methyl group over here in the 5th position. Most of things are going to be in the 5th position. So in the 5th position, suppose this is a carbon that we have over here, in the 5th position of this particular carbon, you are going to have what? You are going to have the methyl group. You also call thymine as 5-methyl uracil. What do you call thymine as? 5-methyl uracil, because it was uracil, but now in the 5th position you have added thymine. You have added methyl group. Got it? Always remember that. Where are we? Where were we? We are here. Okay, so it is going to be known as 5-methyl uracil. Got it? Perfect, ma'am. I got it. It's very clear to me. Okay, in that particular case, let's go ahead now. Now I think everything would be clear from base to nucleotide. Nucleoside formation. First, how does nucleoside formation happen? Nitrogenous base links to the first carbon of the sugar via the N-glycosidic bond. We know N-glycosidic bond is there. You will be asked about the name of all these bonds. Okay, so you should know. RNA, which is there. In RNA, when you have this being formed, whenever pentose sugar as well as the nitrogenous base is being formed, okay, suppose the nitrogenous base is adenine, you will now call this pentose sugar and the nitrogen base together as adenosine. Okay, if you have guanine in RNA, with the pentose sugar, you have guanine over here, guanosine. Okay, if you have RNA, you have cysteine over here, you call it cysteine. If you have uracil over here, you call uridine. So in RNA, you have the names for nucleosides. You have the name for nucleoside: adenosine, guanosine, cytidine, and uridine. Okay. In the case of DNA, you just have to add the term deoxy. Because deoxyribose sugar is being used, which is why you say deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. Now, if in your question they are asking you, what are the nucleoside that makes up the DNA? Options is adenosine, deoxyadenosine, guanosine, cytidine, uridine. What will be your answer? Deoxyadenosine. We need the pentose sugar to be deoxyribose. Get it? These type of questions have come. That is why I'm telling you, you have to be very careful. This is nucleoside. Okay, nucleoside only. Nucleotide we have not considered until now. A phosphate group attaches to the 5th carbon phosphate group attaches to the 5th carbon over here. Now this is how nucleotide is going to be formed. The entire structure is known as nucleotide, and this bond is known as phosphoester bond. What do you call it? Phosphoester bond. So sugar plus base plus phosphate nucleotide. Sugar plus base is equals to nucleoside. Just this is nucleoside. Adenosine, guanosine, cytidine and uridine are nucleoside of RNA. Deoxyadenosine, deoxyguanine and guanosine, deoxycytidine, deoxythymidine is an example of DNA. Okay, now over here, linking the nucleotides. Now we have formed the nucleotide and all. Now we have to form the linkage between the nucleotides. Nucleotide link via the 3,5 phosphodiester bond. Remember this. Okay, the 3,5 phosphodiester bond is there. Here you say 3,5, do not say 5,3. Okay, 3,5 because this is at the top, you can remember like that. So 3,5 phosphodiester bond is something that is going to make sure that the nucleotides are being stacked up together. The chain has a 5' and free phosphate group at the top. Above, at the top you are going to be having, so this is suppose the top. This is nitrogen base, by the way. It's just laying like this. That is why it looks like this. So here you have a phosphate group which is free. Okay, the 5' phosphate group is free, and you have a 3' free OH group. If at all you see this, this is going to be 3' free OH group. Okay, got it? Got it. Now, backbone. Backbone is formed by sugar phosphate unit with nitrogenous base projecting from it. So you might be asked, what is DNA's backbone? DNA's backbone definitely has to be the sugar and the phosphate thing that you see. Sugar phosphate is the backbone over here. Along with that, yes, the nitrogenous bases are also there, which is going to be stacking up things together. Got it? It's simple. It's not difficult. It's not difficult. We have understood the concept, so it is going to be simple. Guess? Yes. Okay. Cool. So that is also being done. What is a bond that we have? 3,5 phosphodiester bond. That is a backbone that we have. The sugar phosphate units which are there, they are going to be the backbone that we have in the case of DNA. Now, we studied about nitrogenous base, and we have two of them. As I said, purines and pyrimidines. So here is a trick. Pure as gold. Pure as gold as in purine, A for adenine and G for guanine. That is what you mean. Pure as gold. You can remember like that for purine. In purine, you have A and G only. Okay, next thing. Cut the pie. So there is an apple pie over here. You're going to cut it. Okay, you're going to cut it. So CUT, cytidine. Over here, you have uracil. And over here, you have thymine. Cytosine, uracil and thymine, you have CUT. Cut the pie. Pie is what? Pyrimidine. Pyrimidine. Pyrimidine, the spelling is PY only, no? That is why. Cut the pie. You can remember like that. So pure as gold and cut the pie is a trick that you can use. Pairing rules, A pairs with T with 2 hydrogen bonds, G base pairs with C with 3 hydrogen bonds. If at all you ever forget G, who has more hydrogen bonds, G and C look so similar, that is why they are going to be too close. They have 3 hydrogen bonds. Always remember that. Purine always pairs a pyrimidine. That is what keeps the 2 strands a constant distance apart. Always remember, purine always base pairs with the pyrimidine. KCET twist this every few years. Which base is DNA only? Thymine, you know. Okay, which base is going to be there in DNA? You should know it's going to be thymine. What is thymine known as? Thymine is nothing, but it is known as 5-methyl-uracil. 5-methyl-uracil. Got it? Got it, ma'am. Clear, ma'am. Okay. The Watson-Crick double helix structure. So, you know, there were 2 scientists, okay, Watson and Crick. Who are the people who came up and basically gave the whole thing? The thing that we were talking about until this particular point of time was actually given by Watson and Crick. They gave the Watson-Crick double helix model, and this can be a 5-marker question which could be asked to you. Explain Watson and Crick's double helix structure, or explain the double helical structure of the DNA. So, what do you have to go ahead is that, yes, there are 4 points over here, but before you go ahead with that, start talking about what is the DNA made up of. DNA is made up of a nucleotide. Nucleotide consists of this. When this and this comes together, you get a nucleoside, and you have a bonds between this nitrogenous base as well as pentose sugar has a bond which you call as the N-glycosidic linkage. Okay, phosphate group, and this has a bond. Carbon has a 5th carbon has a bond. You call this as phosphoester bond. 2 different nucleotides are going to bond with 3,5 phosphodiester bond. All of it you have to mention, and then you talk about these points. Okay, the first point that we have is anti-parallel strands. What do you mean by anti-parallel strands? I told you one is going to be there in 5' to 3' direction. Other one which is going to be there will be in 5' to 3' direction. Opposite. Okay, never together, opposite. So two polynucleotide. Polynucleotide means multiple nucleotides are going to stack up together. That is why you call it polynucleotide. Okay, two polynucleotide chains 5' to 3' direction and 3' to 5' direction with the sugar phosphate backbone is there. With the sugar phosphate backbone is there. These terms are very, very important. And the bases, nitrogenous bases, they project inwards. They are inside, no? They project inwards. Now the second rule is what? The antiparallel strands has been said. Second, we say that base pairing is going to, why they are anti-parallel in nature? Because they have base pairing. A base pairs with T with 2 hydrogen bonds, G base pair with C with 3 hydrogen bonds, keeps the strand distance uniform. That is also clear. The next thing, right-handed helix. So when they turn around, you know, they are not straight like this. Basically, they coil around. Okay, so they have a right-handed helix. They move in the right-handed direction. That is why you say right-handed helix is there. And we're going to talk about much more things. So over here, they have mentioned pitch is 3 point. What pitch? We are talking about cricket. No, listen to me very carefully. So basically, one whole turn which is there, you call this as a pitch. You call this as a pitch. And we say the distance is 3.4 nanometer. How much? 3.4 nanometer. Now, Jasmine Ma'am is telling you something. Okay, Jasmine Ma'am is telling you something very seriously. You have to listen to me very carefully. Over here, in this one pitch, okay, one turn, I am saying there are 10 base pairs. How many base pairs? 10 base pairs. As in, one adenine, thymine, one guanine, cytosine, like that, you have 10 of them. 10 base pairs are being there in one particular pitch. Now, the question that I am going to ask you is, if the pitch distance is 3.4 nanometer and you have 10 base pairs, what is the distance between each base pair? What is the distance between each of the base pair? Mathematics, I hate mathematics. That is why I am here taking biology. If at all there's a student like that, I highly relate to you. Listen to me very carefully. 3.4 divided by 10. 3.4 divided by 10 give you the answer for this particular question. It is going to be 0.34 nanometer. In between each of the base pairs, you have 0.34 nanometer distance out there in between each of the bases, and that is exactly what is written, and this is a very important thing. Pitch distance is what? 3.4 nanometer. Okay, in that one particular pitch, how many base pairs are there? 10 base pairs are there, ma'am. So in that particular case, in between each base pair, how much space is there? 0.34 nanometer. Done, macha. That's all. 0.34 that is a standard thing. Okay, they follow that, and you just imagine DNA is just a molecule. It's not the brain. It's just a molecule and it's being stacked up like that. It's crazy, right? Yes. So that is there. And now, obviously, after doing all of this, don't you think it is going to be stable? So yes, stability. Base pair stacking and hydrogen bonds provide the structural stability. Base pairs which are there, who provides the stability? Obviously, the other parts, you know, the deoxyribose and thymine and everything is there. But apart from that, we're talking about the double helix model. That means we have to be thinking structurally. So what gives the stability out there? Who provides the backbone? Sugar and the phosphate. Who provides the most stability? Obviously them. Along with that, there is going to be base pair stacking. The base pairs and also the hydrogen bonds between them are going to provide you the extra stability, which is very much important. So if you have been asked about the double helix model, as I told you, explain what is your DNA made up of and things like that. So 5-marker question, you have to explain that. You have to make the diagram. Please don't show conjusi over here. Okay, we have to give our best at this particular point. So there are anti-parallel strands that we have. That is the first point after explanation. Anti-parallel strands, one, they are polynucleotide strands which are going to be in one 5' to 3', other than 3' to 5'. Okay, and the sugar phosphate is going to be acting like a backbone out there, and the bases are projecting inwards. Next thing, ma'am, we're talking about this base pairing. We have seen that A base pairs with T and with 2 hydrogen bonds, G with C with 3 hydrogen bonds. Then we talk about the double helix model out there, and we talk about the pitch size 3.4 nanometer. In each pitch there are 10 bases, and in between the 10 bases, how much distance is there? 0.34 nanometer. So 3.4 divided by 10, 0.34. Okay. Lastly, does this provide stability? Yes, this provides a high structural stability. Why? Because the base pairs are being stacked up like this and they have hydrogen bonds. That's all. Okay, write these things and you are going to get your marks. This is going to be the Watson-Crick double helix structure which we have. Cool. Now, listen, apart from it, so we studied about the double helix structure, but it was not that easy. Before that, someone had to identify that there is actually something like DNA, right? So who were the people who went ahead and spoke about it? The first year that we need to know is 1869. 1869. Does anyone of you remember principles of inheritance and variation? I taught you for how many years, okay, Mendel worked on the pea plant. It was 7 years. Do you remember when did his experiment end? If you remember, put it down in the comment section right now. If you remember, when did his experiment end? And it's a very important KCET question, so you should be knowing it. Put it down in the comment section. So anyway, 1869, what happened? Okay, after Gregor Mendel finished his experiment, Frederick Mitscher was eating mixture. Okay, what is he? Mixture. While he was eating mixture in the bowl that he had, suppose this is the bowl that he had. Okay, in the bowl, in the middle, he found some acidic thing. Okay, some acidic, not so tasty thing out there. You can imagine like that, but it's actually not that like that. So Frederick Mitscher went ahead and was looking at the nucleus, okay, was looking at the nucleus and all. He identified an acidic substance within the nuclei, within the cell nuclei. He called this as, he didn't know that it was DNA. Why it is acidic, by the way? Because it has phosphate group. DNA has phosphate group that gives a negative charge, which makes it acidic. Okay, now over here, this acidic structure that we have in the cell nuclei is what we call as what? Nuclein. What do we call this as? This is known as nuclein. Very, very, very important. He didn't say it's a DNA because he didn't know about all of it. Okay, he called it as the nuclein. Got it, got it. Next thing, we have Erwin Chargaff. Before Erwin Chargaff, I think I should be going over here. Watson and Crick. Watson and Crick. Okay, so we got the nuclein, something acidic is there in the nuclei. Then Watson and Crick came. Okay, Watson and Crick came and they proposed that the double helix model of DNA is there. This is how it looks. And for that, they were not independent. They were not independent. For example, if at all I'm teaching you something, I was also being taught by some teacher out there. Generally, that is how it goes on, right? So over here, Watson and Crick also use some data. They use the X-ray diffraction data from two people. Okay, Maurice Wilkins and Rosalind Franklin. Maurice Wilkins and Rosalind Franklin. These were the two people who were doing X-ray diffraction data on the same DNA structure and things like that. And with the help of their data, Watson and Crick came up with the DNA double helix structure. Okay, so first thing, Mitscher was eating mixture, Frederick Mitscher was eating mixture, and he found something acidic in the cell nuclei in the middle that was known as nuclein. Okay, then we have Watson and Crick who said that. Watson and Crick, when he came together, he used the information from Maurice Wilkins as well as what? Rosalind Franklin. Rosalind Franklin. And basically with the help of that X-ray diffraction data, they came up with the double helix model structure. Now the last thing over here is Erwin Chargaff. What did he do? In a double-stranded DNA, the ratio of adenine, thymine and guanine cytosine are constant and it equal to 1. That is what he said. That means basically when I talk about adenine and thymine, you know adenine base pairs with thymine. So how many ever number of adenine is there, that means number of thymine will also be there, right? That ratio, okay, that ratio, adenine to thymine ratio, as well as guanine to cytosine. How many number of guanine will be there? That means number of cytosine will also be there, right? So adenine to thymine ratio as well as guanine to cytosine ratio is always going to be equal to 1. That is what Erwin Chargaff said. Now, ma'am, what is this? Okay, does it make any sense? Yes, it makes sense. Only when he got to know this, that's when he got to understand that A always base pairs with T. Arey Baba, you know it. They didn't know it. They did the finding and that's how they came up with this conclusion. Okay, adenine and thymine is equals to 1, so probably they might be base pairing, you know. So that is how it was there. So Chargaff, just remember his name for the base pairing thing. Rest of it, I told you Frederick Mitscher was eating mixture. That's when he found something acidic. So that's all. Now, this is the question that has come in your second PUC. As I have mentioned, this is a 5-marker question and I've told you how to write it, so I'm not going to repeat it again and waste your time. And we are going to solve some KCET question. Just taking a break over here. You guys make sure you just revise and be ready for solving some KCET questions. Okay? Okay, so let's go ahead with the KCET question, and here is the first one. Which of the following nitrogenous bases found only in the DNA? We studied a lot of things, now we have to use it over here. Which is found only in DNA is the question, and what are we talking about? We're talking about the nitrogenous bases. You have cytosine, you have adenine, you have thymine and guanine. 4 options. This is KCET 2019 question. Only in DNA. Let me decode. By the way, you can also try it out in the chat sessions right over here. This is a premiered video, so I might not be able to watch it. Okay, sometimes I may come and just say a hi and go. But my dear children, I really appreciate you guys watching the entire video. I am seeing your efforts out there. I'm pretty sure you are going to get the best. Okay, so anyways, cytosine is found both in DNA and RNA. Okay, adenine also found in DNA and RNA. Thymine only in DNA, because in the case of RNA, what do we have? Uracil. In the case of RNA, we have uracil. Now, guanine again found in DNA and RNA both, which is why the only answer for this question becomes what? It becomes thymine. It becomes thymine. Next question, KCET 2018 question. Okay, DNA present in the nucleus was named as nuclein by. Okay, the DNA which is there inside the nucleus, it was named as nuclein by whom? James Watson and Crick, Frederick Misher, Maurice Wilkins, or Rosalind Franklin? You have three options right over here and you can choose from it. DNA present in the nucleus was named as nuclein by. Nuclein, is it James Watson and Crick? No, Baba. They gave this DNA double helix model, right? It has nothing to do with the nuclein and all, okay? Nuclein basically was given by the person who said that there's something acidic inside the cell nucleus, if you remember. It was none other than Frederick Misher. While this particular famous personality was having Misher, he found in the middle, in the center of the cell, in the nuclei, basically there's an acidic thing which he called as nuclein. We studied about it, right? So this is going to be the answer. Maurice Wilkins and Rosalind Franklin, Baba, they were responsible for the X-ray diffraction data, with the help of which Watson and Crick were able to deduce the double helical structure. Okay, so we have got this answer also, and I hope this makes you a little bit happy out there. Right? Going ahead, another chemical name for the nitrogenous base thymine is. This is not a KCET question. This is a model question paper question, MCQ. Another name, another chemical name for the nitrogenous base thymine. What is the other name for thymine? 2-methyl-uracil, 3-methyl-uracil, 4-methyl-uracil, or 5-methyl-uracil. We all know that 5 is a number that is very, very common in this particular chapter. So it has to be 5-methyl-uracil. And Jasmine Ma'am, you spoke about it right over here. You taught us this particular diagram, no? Where this is uracil. Inside, over here at the 5th position, if at all you add methyl group, it becomes 5-methyl-uracil, and it was structurally more stable. It was structurally making the DNA more stable than the RNA, right? We studied about it. So the answer for this question is going to be this one. Kudos to you. Next one. The components of deoxyguanosine is. This is an important question, a tricky question. The components, okay, what is deoxyguanosine made up of? That is the question. Guanine plus ribose, guanine plus deoxyribose, guanine plus deoxyribose plus phosphate, guanine plus ribose plus phosphate. These are the options that is given to you. Guanine is what? Nitrogenous base. Ribose is a sugar. Deoxyribose is also a type of sugar. Deoxyribose and phosphate is given over here. Here ribose and phosphate is given over here. First of all, is this the part of DNA or RNA? Deoxy term is coming. That means it has to be a part of DNA. If it's a part of DNA, can it be ribose sugar? No. Cut it off. Okay, cut it off. Here also you have ribose. Here also you have ribose. Now you have guanine and deoxyribose left. Here you have guanine, deoxyribose, and phosphate which is left behind. What do you call this as? What is it made up of? You saw the answer. So listen to me very carefully. Deoxyguanosine is a nucleoside, not a nucleotide. Not a nucleotide, it's a nucleoside. We studied about it. If you remember, over here, just give me a second. If you remember, we studied in RNA, we call the nucleoside as adenosine, guanosine, cytidine, uridine. And we call the, the same nitrogenous bases as well as the sugar combining together as deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. When you add phosphate group, the whole thing changes. It becomes a nucleotide. As of now, when you say the term deoxyguanosine, cytidine, or whatever it is, it means just one thing. That means only a sugar and the nitrogenous bases together. There is no role of the phosphate group, because if it was, it would have been said as deoxyguanosine phosphate. Okay, or deoxyguanosine. If 3 phosphate groups are there, triphosphate. But anyway, only 1 phosphate is generally there. Okay, but that is not the question over here. The question is, what do you call this? What is it made up of? It is made up of just this and this. There is no phosphate, because if at all phosphate is there, that term would come. That term would anyway come at the end. That is not there. That means, understand, it's just this, a nucleoside. Okay, if you're asked what is this, this is going to be a nucleoside. Nucleoside, not a nucleotide. Got it? Yes, ma'am. Next question. Okay, next question. In a DNA strand, if there are 100 nucleotides, this is a very different question. Okay, in a DNA strand, if at all there are 100 nucleotides and 30 are adenine and the number of thymine, guanine, cytosine respectively are. in a DNA strand, if 100 nucleotides are there and 30 are adenine, what do you mean by that? Suppose this is your DNA. Okay, and I am saying there are 100 nucleotides. How many? 100 of them. 30 of them are adenine. You have been asked about the number of thymine, guanine, and as well as cytosine. How will you come to a conclusion about this? How will you give your answer? Listen to me very, very carefully. If A is here, T will be here. If T is here, A will be there. Which means what? Which means what? If this is a strand, here A is there, T is there, here T is there, then there will be A. The number of A will be equal to T. The number of adenine will be equal to T, right? If this is 30, this is also going to be equal to 30, right? Right. So we got to understand thymine is definitely 30. What about guanine and cytosine? How do we know that? Total is 100. Out of that, 30 is adenine and 30 is thymine. So what do you do? First of all, 100 minus 30 plus 30 is equals to what? 60. What do you get? 40. 40 nucleotides are still left. In that 40, Baba, again, how many number of G will be there? That many number of C will be there. How many number of C will be there? That many number of G is also going to be there. Whatever you do. Whatever you do. If I see 2G, that means 2C is also there. So this is 40. If this is 40, I have to divide equally amongst both of them. This is going to be 20. This is also going to be 20. You get it? Take your time. 100 nucleotides overall. You have been given the number of nucleotides over here of adenine, 30 you are saying. Okay, nucleotide containing adenine, 30 you are saying. Then thymine will also be 30. If both of them are 30, out of 160 of them already decoded. Now what is left with? What are we left with? 40 of them. In 40, Baba, if I say guanine which is there is 20, then cytosine also has to be 20. It cannot be something else. Okay, it's a double-stranded DNA that we're talking about, right? Okay, got it? In that way, you have to find the answer. And here they have mentioned the number of thymine first. What's thymine? How many number of thing are there? 30. Guanine second. Guanine is basically 20. Cytosine is also 20. 30, 20, 20 would be your answer. 30, 20, 20. I see that this is my answer. Please know the order of it that the question is asking you accordingly. Sometimes children do all of it. But while marking it, they go like guanine, 20, 20, 30, 20. Don't like that. The question is asked thymine, guanine and cytosine. You have to go like that. Got it? Clear? Clear, ma'am. Perfectly clear. Okay. Now try to solve this question. In a single-stranded DNA, if the ratio of A is to G is 60 to 40, then the ratio of T is to C is equals to what? If they're asking you, the ratio of A is to G is equals to 60 is to 40. Then the ratio of T is equals to C is what? That is the question. Okay, how do we get to know the answer for this? If G is 40, this is also going to be 40. If this is 60, this is also going to be 60. That is what you usually assume. Listen to me very carefully. Here, they have twisted things up. What are they asking you? They are asking you about a single-stranded DNA, not a double-stranded. They have clearly mentioned single-stranded. In a single-stranded DNA, if A is 1, 2, 3, 4, and T can be 1, 2, 3, 4, 5 because they are not base pairing amongst. Base pairing happens over here. So in a single stranded, can you predict anything? If it is given about A or G or C or D, can you predict anything? No, Baba, you cannot predict. You cannot predict. Over here, when you say single stranded has A this much, then T over on that side would also be 4. Okay, if T is this much, then A would be like this. Okay, I'm talking about double-stranded, then I can talk about numbers of A and T. But here, that's not the case. Here, that's not the case. This part is totally missing. You do not have this particular portion altogether. So there is no point of talking about it. The answer for this question is going to be what? 60 is to 40, 40 is to 60, 20 is to 80, none of the above. None of the above. It is going to be what? None of the above, because Chargaff's base pairing rule applies only to double-stranded DNA. Not to single-stranded, but to double-stranded DNA. Got it? Clear? Yes, so they can make the question twisted. Read it very, very carefully. Here in a DNA strand they have mentioned. We do not know whether this DNA strand is double or single, but in the options there is no none of the above or none, which is why we can go with the perfect one, which is there according to our understanding. But here none of the above is there, which is why you have to go with that. Got it? Clear? Yes. Now, my dear children, we have completed the first part of the chapter where we studied about the structure of the DNA as well as the different genomic size of different organisms. The lowest amount of genomic size is there in bacteriophage 5x174. Remember, it was in 5,396, something like that. Yes, yes. Now, one more very important thing before I go ahead with anything. If you check our videos, not this one. If you check generally our videos, by the way, one-shot videos I have done for a lot of chapters now, 4 chapters already done. Over here, there might be something like this. Just a second. Just a second. Join the free ACE KCET 2027 course. This is what you know. This is a website we have made over here, where you just have to go to buy now, and it's available for you in 0 rupees. Basically, all the academic lectures which are there, they are stacked up together in the order of the subjects. So you can see subject wise what all acad videos, only acad. There is no counseling in this because I know that could be very disturbing for you. So you can watch it there only. All the acad videos that have been completed for a subject would be there along with the assignments. Assignments are gonna come. You will start getting PDF also there slowly. Okay, so make sure you check it out. This is something that you have to do from your end. And by the way, join the WhatsApp channel compulsorily. That link is also there in the description section. The reason being that that's where you'll get all the updates, what class is coming, when, what day, what time, and things like that. And sometimes I come to talk there also. Okay, now listen to me very carefully. We spoke about the DNA. We know that DNA is going to be very long. How long? I have mentioned it before. Yes, it was 2.2 meters long. How long it is? It is 2.2 meters. Now just imagine. Take a thread 2.2 meters long and stuff it inside a ball 1,000,000 times smaller than a meter. Very small. Very small. Without a single tangle. And it must still unwind on demand. That means it should not get tangled around. It should unwind. It should be straight whenever we want it to be. And still you have to put it inside. Not in this, okay, onto a ball that is 1,000,000 times smaller than a meter. So small, microscopic structure. Very difficult, no? You feel it's impossible almost. But you know what? In our body, that's not the case. You are going to see something very unique, which you call as packaging of DNA, my dear children. And this is going to be concept number 3 out of 13. Over here, this is what you need to know. Our nucleus has a dimension of 10 to the power -6. What is it? -6 meters. That's small. Okay, and what is our length of the DNA? What is length of our DNA? 6.6 into 10 to the power 9. Okay, this is 10 to the power -6. This is 10 to the power 9 base pairs into, listen, these many number of base pairs are there. If you remember now, okay, if at all you look at this particular thing now, everything will become clear to you. Here when I mentioned base pairs means how many number of stacks are there. So these many number of stacks are there in a diploid cell. 6.6 into 10 to the power 9 base pairs. Okay, 9 base pairs. That many number of stacks are up there. Now where is it? Yeah. So, and in between each of them, how much space do we have? 0.34. We have 0.34 if you remember. Total pitch size was 3.4 nanometer. 10 base pairs were there in one pitch. To divide it, 10 divided by 3.4 is equals to what? 0.34. That is the distance that we have. So in between these many base pairs, 6.6 and 10 to the power 9 base pairs, we have 0.34 space. Okay, and we have to convert it. Okay, we want, that was nanometer. Okay, 0.34 nanometer was there. See, we would want to convert it to meters. Okay, so 10 to the power -9 meters. So basically at the end of the calculation, what do you get? That's more important. 2.2 meters. Okay, what do you get? 2.2 meters. This is in meters, so we are talking in meters. A thing which is 2.2 meters long, you have to fit it inside something which is 10 raised to the power -6 meters in dimension altogether. How do you achieve it? You have to do something. You have to wrap your DNA by doing something. So you are going to do that. You're going to wrap it around what? Around what? 2.2 meters of DNA inside a 10.-6 meters, not m, meters nucleus. How, ma'am? How is that possible? It is possible with the help of this particular structure, my dear children, and this structure is going to be super important for us. So listen to me carefully. Okay, your DNA, there are some features about your DNA that we need to remember. The features are what? It is negatively charged. It is negatively charged because it has the phosphate group and everything. It is acidic because of the same reason and all. Okay, because of the phosphate group and stuff like that. We know about it. We know clearly about it. Okay, my handwriting over there is really bad now. I'll try to be a good girl. Okay, listen to me. If I want to wrap around my DNA and make it really compact, what will I wrap it around onto? Will it be negative thing or will it be positive thing? If I wrap it around a negative thing, negative is going to repel. It's not going to attract, it is going to repel. And we do not want that. We want it to stay like that itself. So my dear children, over here, I am going to use some very important thing. Okay, I am going to use something that is super important for me. And what is that? That is going to be a histone protein. This histone protein which is there, Baba, okay, P for protein and P for positive. It's going to be positive in nature. Proteins are made up of what? Proteins are made up of amino acids. So over here, the reason why it is positive is that it is made up of positive amino acids. Okay, there are different type of amino acids, but here majority of it is what? Positive amino acid. Ma'am, the name of the amino acid, if at all you can tell, it will be really good. Okay, then I'll tell, because anyway, you and me have the same mission to make sure that we study this chapter nicely. One is going to be lysine. This is an amino acid. And the other one which is there is going to be arginine. Is it? Lysine and arginine. Okay, you also can remember it as Los Angeles. Very important thing. Very important thing. Lysine and arginine are the amino acids that you have that makes up this particular histone protein onto which your DNA is going to be wrapped around. Now, ma'am, how does the DNA wrap around this thing? And is there only one histone protein out there? The answer is going to be no. There are multiples of them. How many? Let's check it out. So, one histone protein, second one, third one, you count with me, okay? Fourth one, fifth one, sixth one, seventh one, eighth one. So the back side also it's there. My diagrammatic skills is not that great, okay? That is why it's like this. But please do understand, there are 8 histone proteins. How many? 8. Okay, I'll write 8 of them are there. Now, 8 of these histone proteins onto which what is going to happen? Your DNA is going to come. Now look at this DNA, okay? The DNA is coming, wrapping around, coming again. So if at all I have to check, how many times does the DNA wrap around this? DNA came, wrapped around once, and then again coming, wrapping around it once more. Can I say it wraps around it twice? Yes. And when I say twice, you need to remember one more very important thing. There are almost 200 base pairs covered over here. So we have 6.6 into 10 to the power 9 base pairs of DNA. Out of that, this particular structure, the histone, okay, 8 of them together, when they come together, or your DNA wraps around it, and 200 base pairs are covered there. Similarly, you are going to have multiple sets of these things. You are going to have multiple sets of these things. Okay, ma'am, I have a doubt over here. What do you call this? And also, this is just this histone that is there? No. Listen to me carefully. So, just a second, children. I'm going to remove this thing over here. Okay. Over here, you would be having one more histone protein right up. Okay, it's like a linker one which is there. And then again, you would be having what? You are going to have your other set of histones. 8 of them. Again, the DNA is going to wrap around it twice. That is how it generally works. Now listen, this particular structure that we have from here to here, this is generally known as, okay, this thing that you see over here is known as a nucleosome. What do you call it? It's a nucleosome. A nucleosome is made up of how many histone proteins? 8 histone proteins. 8 histone proteins. Apart from it, what do you have? You have one H1 histone. This is another type of histone only, but this is H1 histone that we have. H1 histone is also there. Please do not forget it. Okay, and what happens over here? DNA wraps around twice. DNA wraps twice. And what? 200 base pairs are going to be covered up over here. Will you be able to remember this thing? Okay, now you imagine over here. This is something that is important. If now I look at under the microscope, how does it look to me? These are what? These are nucleosome, made up of so many 8 histone out there. This looks like a bead on a string. These are beads. Bead on a string like situation, right? Well, this whole thing that is being formed, when your DNA is going to get compacted and is going to roll around and is going to have a look of bead on a string like, what do you say, look out there, you call this as chromatin. You are going to call this as chromatin. Now the chromatin which is there, it is going to condense even further. It is going to get compacted even further, and that's how you are going to get what? The next thing out there which you call as chromosome. So this gets more condensed, and that's how you get your chromosome. Initially, you had the loose DNA. The loose DNA is trapped by the histone proteins and all. Okay, then you get the chromatin, which is bead on a string-like structure. Then you compact it further, and finally you get the chromosome. That is how things generally work. And I also want to tell you one more thing. These 8 histone proteins are also known as histone octamer. Okay, it's also known as histone octamer. Octamer means a group of 8. Histone octamer. Works. Now listen, one more thing. Okay, one more thing. So before I go ahead with everything else, I just want to repeat whatever I said because I know it's too much for you. So your DNA is negatively charged. It is 6.6 into 10 to the power 9 base pairs long. Okay, that means basically you have that, and if you talk about in meters, 2.2 meters long, and you have to fit it inside 10 raised to the power minus 6 meter dimension wala nucleus, right? For that, you have to make things compact, and how do you do that? You take the help of histone proteins. How many histone proteins are there to help? 8 of them. They say that we are going to group together. The DNA can wrap around us twice. That means 200 base pairs will be covered in one particular octamer itself, and then you would be having one linker histone protein which you call as H1 histone, and further again next histone octamer. Together this structure that I have over here is known as what? A nucleosome. How many nucleosomes will be there? So many nucleosomes will be there, and the arrangement of these nucleosomes looks like a bead on a string. Bead on a string like structure. This is now known as what? Chromatin. This strand which is being formed over here is known as chromatin. It is going to condense even further to form what? The chromosome. Okay. Now the histone protein, why DNA wraps around it? Because DNA is negatively charged. Histone protein is going to be positively charged. Why? Because of 2 important amino acids, lysine and arginine. Lysine and arginine. LA, Los Angeles. Cool. Now, to make the chromatin, to make the chromosome, there are also some non-histone proteins which are required. Just understand it. Apart from this histone proteins and all, there are some non-histone proteins which are also there. Just have an understanding of it. Just remember it. Non-histone proteins are also there. Okay. Cool. Now let's read it. Have a quick read. Histone octamer. 8 histone proteins rich in positively charged amino acid, arginine and lysine. Los Angeles. Okay. The DNA wraps twice around the octamer with 200 base pairs per nucleosome. Okay. H1 protein. H1 sits between the 2 nucleosomes, sealing the DNA at the entry and the exit. So basically, when we make the diagram of, the reason I told you that H1 comes here together is because whenever you are asked to make the diagram, you have to make it like this. Okay, here basically, here, so suppose here it is entering, here it is going to exit. this touches H1 histone which is there, it is going to touch the entry and the exit points. Please to remember it. And this whole structure is going to be known as nucleosome. So it is said that H1 actually comes in between two nucleosomes, but okay, you can just remember it like this because this diagram is important. Diagram based question usually come from this particular concept most of the times. Most of the times you are asked to make this particular diagram. Now you can make the 8 ones also. You can make something like this and mention that it is histone-octamer as well. That's your choice. Cool. Cool. Now, chromatin. Nucleosome units come together to form beads on a string structure which you call as chromatin. Nucleosomes, they come together to look something like this. This is actually a microscopic image. Okay, it looks something like this. Non-histone chromosomal proteins are also required in chromatin formation. For the formation of chromatin or for the formation of chromosomes, you will require some non-histone. That means not histone, but there are some other things also, there are other proteins which you might require in the whole process. Done, ma'am. Okay. Now, there are two types of chromatin. One is euchromatin and the other one is heterochromatin. Euchromatin and heterochromatin. What do you mean by that? Very, very simple. Listen to me. Just use your imagination, okay? Just use your imagination. If the chromatin is not stacked up very close to each other, if the nucleosomes on the chromatin are not very stacked up, they have some space, they have some breathing space, you call them as euchromatin. If they are stacked up together, you call them as heterochromatin. Now suppose I put a stain over here. I put a stain, coloring stain. Will it get darkly stained? No, it has enough space and all, which is why it might be having a light stain. But if at all I put a stain over here, it is going to get darkly stained. It is going to get darkly stained because that is how it is. It's in compact nature. Okay, light stain, dark stain, loosely packed, tightly packed. Okay, got it? Got it. Now one more thing. Listen to me very, very carefully. We said that DNA is important. DNA. We are talking about DNA from a very long time. This DNA which is there, why is it important? Because it is responsible for formation of protein. Can it directly make the protein? No, Baba. It needs the help of another thing which is known as mRNA. It needs the formation of mRNA. Only the mRNA can directly give information to provide proteins. DNA directly cannot make a protein. Okay, it needs to go by this particular step. This step of basically making an mRNA from the DNA is known as what? Transcription. Transcription. Transcripted, it's written sometimes when you open WhatsApp, encrypted, transcripted, some things like that will come. Okay, so it's transcription where whatever information is there in this very important chip of ours, DNA, is going to be transcripted to an mRNA. That mRNA later will give information to form protein. Right. Now, your DNA has to get transcripted. Where is your DNA? Your DNA is right over here. Okay, obviously it has to get transcripted, then only it can make protein. Suppose this wants to make a protein. Now what? Can it make a protein? Yes. Why? Because there is space, no? You see there is space where the enzymes can come and attach. Obviously, they cannot just start off. You need some enzymes to come over here. So only if you have a route, then only only vehicles can come in, no? Similarly, enzymes which are ready for the process of transcription are gonna come only if at all there is space. So euchromatin, we say, they are going to be transcriptionally active. That means transcription is happening up there. What about this guy heterochromatin? It is going to be transcriptionally inactive. Transcription cannot happen there. The reason being, it's highly stacked up together. That's the reason. It's highly stacked up together. That is going to be the reason. Okay, so euchromatin, loosely packed, stains light. Transcriptionally active chromatin. Okay, loosely packed, stains light, it is transcriptionally active chromatin. Heterochromatin, more densely packed, stains dark, transcriptionally inactive chromatin. That is heterochromatin. Okay, so you remember euchromatin as you are a green flag. You are a green flag. For you, everything is fine. Okay, if at all you ever forget the transcriptionally active, which one is it? Is it euchromatin or heterochromatin? I hope you will not forget now. It's you who are the green flag. Let's not forget that. Yeah? Yes. Perfecto. Children, drinking water? I need some water. Let me drink, okay? I hope you are also getting yourselves hydrated. Liking the class? Are you liking the class? Okay? As I said, if you're loving the class, you have to hit on the like button first. Second, hit on the subscribe button and share it with at least 5 of your friends and say that this teacher actually is okay. You can probably trust her with teaching. So if you feel like that, definitely do so, okay? Only if you feel, otherwise don't send to anyone. Okay, let's go ahead. Draw a label diagram of nucleosome. This is a 2-marker question that have come, and this was in model question paper 2, model question paper 3. This has come in the examination also. Very important, that's what I told you. Please practice the diagram. Okay? Okay. Now, this is a KCET 2022 question. Very easy question. The given diagram represents ball. You know, there is something like we do like this and then it goes. Okay, so I don't know why they even gave this question. Nucleosome, chromosome, mesosome, ribosome. But I do understand when I saw the options. The children, when we do ratification, we just remember some. It can be nucleosome, samosasome, anything some. So nucleosome, chromosome, mesosome, ribosome. What is the answer? Nucleosome. We are talking about nucleic acid, DNA, deoxyribonucleic acid. Okay, so we have to talk about the nucleosome right over here. Done, done. Next question. Okay, question, this is okay. In 2022 also you got a question based on this. 2023 also you got a question based on this. Let's go. Histone proteins are positively charged because they are rich in the basic amino acid residues. They are rich in some basic amino acid residue. What do you mean by that? Arey Baba, DNA was negatively charged, acidic. Right over here, when you say positively charged proteins are there, okay, things like that, they are going to be basic. They are going to be basic. That was acidic, this was basic. Okay, anyway, so why is positively charged? Why is it positively charged? Because of which amino acids? Arginine and lysine, arginine and phenylalanine, arginine and proline, arginine and alanine. Los Angeles, I told you it is going to be this one. Okay, now some people, they will learn lysine or probably leucine, and they will go with alanine. There are multiple amino acids, please do know that, okay? There is alanine, there is leucine. So again, LA ma'am said, hurry, just have, that's just short trick. This is something that you have to remember. Lysine and arginine. Okay, lysine and arginine, both of them are responsible for this. Cool, cool. Now, DNA is genetic material in both prokaryotes and eukaryotes, yet the packaging process in the cell is not the same. Explain. This was a model question for one question. Okay. So, basically DNA is found in prokaryotes, Escherichia coli, a prokaryote, right? A bacteria. What is prokaryote? Prokaryotes are the one which, for example, in the genetic material, no, there is no membrane around it. Okay. Similarly, they do not have membrane-bound organelles also, such as mitochondria and things like that. They do not have all of it. They are prokaryotes. They are very primitive organisms. We are eukaryotes. Each of a cell is eukaryotic cell, where nucleus is having nuclear membrane. All the other organelles are having membranes around them. We are very complicated. Okay, we want to be complicated, which is why we are complicated. Anyways, it's a good thing only. That's why we are we. Okay. Question is, DNA is genetic material in both prokaryotes as well as eukaryotes. Prokaryotes as bacteria and eukaryotes as us. Yet the packaging process is going to be different. Why is it not same? Explain. So how do you explain this? Listen to me. What are the stark differences that we have? First thing, E. coli, the genetic material was 4.6 into 10 to the power 6 base pairs long. Ours 6.6 into 10 to the power 9 base pairs long. Whose genomic size is more? Ours. Theirs is less. So prokaryotes basically has lesser genomic size. First thing. Okay, first thing, that is itself is different. So packaging is going to be different. They might not need this type of complicated packaging. Second thing, their DNA is going to be, in the case of prokaryotes, do they need to be compact? Why do we need to compact? Why do you think that DNA over here, in our case, need to be in compacting? Because we have to put it. We have to make sure that it is safe inside the vault over here, that is your nucleus, which is 10 to the power minus 6 meters. Okay, if you remember, we have to put it over here. That is why we are compacting it. Arey Baba, in the case of prokaryotes, they have the whole area lavish. Okay, their genetic material does not have any membrane around it. So why do you think you will need histone proteins? Do they have histone proteins? No, they do not have histone proteins. Got it? Now we're going to read it and probably you'll understand it even more better. DNA is genetic material in both prokaryotes and eukaryotes, but its packaging differs due to difference in genome size as well as cell organization. The size of the genome and also the cell organization, how the cell is. Does it have membranes? Does it not have? In prokaryotes, DNA is going to be naked, circular. The DNA in the case of prokaryotes is going to be circular. It looks like this. And present in the nucleoid region without histones. Nucleoid region is a region, a free region where you have your genetic material being intact. Okay, it's not bound by any membranes. Small genome is also there and no nucleus is there. Small genome is there, no nucleus is there. Okay, two reasons. Small genome, no nucleus, which is why they do not need histone proteins for packaging. It's just there in the nucleoid region. When you talk about eukaryotes, it associated with histones. About 200 base pairs wrap around a histone octamer that forms a nucleosome. Nucleosome is called to form chromatin, which condenses into chromosome, packing almost 2.2 meters of DNA into the nucleus, which is just 10 to the power 6 meters. Got it? So genomic size is more. The cell structure is also going to be more different. It is having a nuclear membrane and things like that. Got it? Will you be able to answer like this and make a diagram? Okay, you will get your 5 mark. This was a 5 marker question. So diagram also carries 2 marks, and probably all this thing carries 3 marks. Okay? And always write your answers in points. Yes. Fine. Now write the difference between euchromatin and heterochromatin and name the virus used in Hershey and Chase. Okay, so Hershey and Chase experiment, we're going to talk about that. First of all, we are over here. Difference between euchromatin and heterochromatin. 2 marker question. Euchromatin, loosely packed, light stained. Loosely packed, light stained. Here dark. Here basically densely packed, dark stain. Okay, DDLL. Okay. Now euchromatin transcriptionally active. It is transcriptionally inactive. That's all. 3 points. 3 points if you put. Done. Okay, in Hershey and Chase experiment, which virus did you use? We are going to talk about it really soon in this particular thing. That is going to be the search of DNA. That's the next concept. So I hope the structure was clear, the packaging is clear. Now we go ahead to the next thing. I'm just curious, but who really found that DNA was the genetic material? Why are you even talking about it? Who was the person who said that it is the genetic material? Because I feel it's very important, and you are saying it's a genetic material, it is used to form proteins and things like that. Was there anyone who said that? Yes, DNA is a genetic material. The answer is yes, not just one person, a group of people out there. And I'm going to talk about each of them over here. Concept number 4 out of 13. Search for the genetic material. Okay. So over here there was a guy whose name was Frederick Griffith. Okay, Frederick Griffith. And by the way, I'm really famous for this thing. I mean, I'm not famous. My co-guest over here, Puchu. Okay, Puchu. This is 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 Puchu. All of them are Puchus, and they are very famous. Okay, because on Puchus, Frederick Griffith did his experiment that is known as transforming principle experiment. Very famous. All the experiments over here, all the 3 experiments that I'm going to talk about over here in the next few minutes are something that could give you easily 5 marks. Okay, so listen to me very carefully. Okay, so what happened is that there was a bacteria. Okay, there is a bacteria, not was, there's a bacteria, Streptococcus pneumoniae. Streptococcus means they are round shaped bacteria, and they cause what? They cause pneumonia. You know pneumonia, where your lungs and everything is being affected. Okay, you cannot probably breathe properly, it's filled with fluid and stuff like that. And organism, if not treated, it will be dying, right? That is how it works. So there is Streptococcus pneumonia, pneumoniae, which causes pneumonia. Okay, and there are 2 varieties of it. Okay, one is going to be the rough strain. You know, the varieties, you call them as strains, okay, in the case of bacteria. And then there is a smooth strain. And then what do you have? You have smooth strain. In the rough strain, Baba, this is the bacteria, suppose. This is the bacteria, that's all. Okay, this is the bacteria, that's all, khatam. In the case of smooth strain, you have an extra coating over here. A polysaccharide coating around it. Polysaccharide means it's made up of carbohydrates and stuff like that, which makes it smooth, Now, people in general who look so rough to us, probably from inside they are papa people. People who look so nice to us might not be that good. Okay, even though I'm not in that particular logic, but still I'm telling you. Okay, so this is how it works. This is how generally it works. My dear children, listen to me. The same thing is happening over here. If at all an organism is affected by the rough strain, it will live. The organism is going to live. Why? Because the rough strain is the non-virulent one. Means it does not cause any trouble. Non-virulent means it is not harmful. Whereas the smooth strain, which has an extra mucus polysaccharide covering over here, is the one that is virulent in nature. That means if at all an organism is affected by this particular strain of bacteria, then got it? So Streptococcus pneumoniae is the bacteria that we have. It is a bacteria, it's round in shape, that is why coccus. It causes pneumonia. You have two strains of it, rough and smooth strain. The rough strain is rough because it does not have the covering. Smooth strain is smooth because it has an extra polysaccharide covering over here. Since it does not have the covering, we call it non-virulent form. It does not cause any trouble. Over here, this is a virulent one. This can lead to death of the organism. Okay. Now, S strain versus R strain. S strain has a mucus polysaccharide coat that makes it virulent. R strain lacks it, making it non-virulent. The conclusion. Okay, now let's talk about the conclusion a little late. Let's talk about the experiment first. So this is my puchu. Okay, this is puchu. Onto puchu, Griffith went in and put the live R strain. What happens if you put the live R strain? Does it affect? No. Puchu lived. Puchu lived. We are all happy. Okay. Again, Griffith was like, took another puchu and then put live S strain over it. Okay, probably this puchu, same puchu also, probably put S strain, live S strain. What is going to happen? Puchu is going to die. What's the next thing? Puchu is going to die. Papa puchu. Okay, now he was like very sad. What did I do? So sad. Okay, I'm going to do something. Okay, I'm going to treat it with heat. Let me treat the live S strain bacteria with heat. Let me see what happens then. So he treated with heat. When you treat with heat, know what happens? The cell, basically the organism, generally the outer part of it and everything just goes off. Genetic material can also be hampered, but something might be still left. Some genetic material could be still left, or it could be just hampered altogether. Anyways, he took the heat-killed S strain and he put it inside the mouse. Puchu lived. Surprisingly, he lived. Which means that heat was having some change over here, which is why it happened. Otherwise, the organism was dying. So now, Griffith was like, this did not cause any trouble. This also did not cause any trouble. Why not mix both of them and put it inside puchu? Let's see what is going to happen. So he put heat-killed S strain and he put live R strain. Heat-killed S strain and he put live R strain, mixed both of it and put it inside the mouse. What happened? Puchu died. Puchu died. And not just that, puchu didn't just die. Inside puchu, you got live S strain. Did you put live S strain? No, you put heat-killed S strain and you put live R strain. Then how can you get live S strain over here? Inside puchu, inside of that puchu. Baba, it's because something was moving from here to here. When you mix both of it, and I told you, if heat does not damage the entire thing out there, if at all there's some genetic material left behind, probably it can pass through the live R strain out there and can transform it. Now the live R strain which is there has the DNA, or probably has the genetic material which was required, has the genetic material which is required to convert into or to make the polysaccharide around it and cause trouble. The superpower it received from somewhere. Okay, we do not know what is superpower, whether it's a DNA, whether it's an RNA, whether it's a protein, we have no idea about it. We just know that something has passed from here to here, such that there is a transformation that have taken place from live R strain to live S strain. This is known as transforming principle experiment. It was given by Frederick Griffith in the year 1928. 1928, he gave this experiment. Some transforming principle, he said that, I do not know what is transforming the organism, but he called it transforming principle. He called that particular thing as transforming principle. He said some transforming principle from the heat-killed S enabled R to build a smooth coat and turn virulent. From the heat-killed S, something went over here, some transforming principle went over here and transformed this such that it basically formed a mucus polysaccharide coat around it and made itself virulent, which is why the organism died. Okay. That's the end of the experiment. That's the end of Frederick Griffith's experiment. Now to continue that experiment, some other people also came into the picture, because obviously we have to talk more about it, right? I hope this is clear. You have to, when you are asked to write about Griffith's experiment, you have to mention it. He took a mice and he inserted the live R strain. He found that the mice lived. You just put live R strain, put inside the mouse, and then put it arrow mark, mouse lived. Live S strain, put it in the mouse died. Okay, heat-killed S strain, mouse lived. Okay, heat-killed S plus live R strain, the mouse died. And then he found live S strain from this particular mouse also. With that, he understood some transforming principle is getting transferred from heat-killed S strain to live R strain. Got it? That's all. That's all you have to write. Making it produce polysaccharide covering, making it virulent. Griffith's transforming principle experiment, explain it. This is a model question paper 1 question of the previous year, and it's a 5-marker question also. Cool, cool. Now listen to me. Over here. Okay, over here I have a KCET question also on the same topic. Following representation PQR denote few steps of Griffith experiment. Identify the correct steps. R strain, inject into mice die. If R strain, rough strain I put inside the mice die, no baba, it's Papa strain, my mouse will not die. Okay, S strain, heat killed one. If at all I'm injecting in the mice die. No, it is going to live. It is going to live because it is heat killed. If I'm putting R strain inside the mice live. This is correct. So R is the only one which is going to be correct, which is why the answer for this question is going to be what? B. It is going to be what? B. This is KCET 2024 question. KCET 2024 question. Okay, there is one more question. In KCET 2022 it had come. A series of experiments were conducted by Frederick Griffith in 1928 on transforming principle with what? With what? A series of experiments were done by Griffith with the help of which organism? Salmonella typhimurium, that causes typhoid, Escherichia coli, that is E. coli, Streptococcus pneumoniae, or Bacillus thuringiensis. By the way, you know what? In your 12th grade, you're going to study about all of them, in different chapters. So anyway, Streptococcus pneumoniae, that causes pneumonia, is the one that is the answer for this. Got it? Got it, ma'am. We know that. We studied about it. So that is also done. Okay. Now we come to the next experiment in continuation. Okay, just understand this is continuation. After Griffith went, Avery, MacLeod, McCarthy came into the picture. Avery, okay, MacLeod and McCarthy came into the picture. They said that, okay, we do not know whether it's a protein or a DNA or it's an RNA that is getting transferred. Let's do an experiment. Okay, listen to me very, very, very carefully. All your attention over here. Okay. Heat killed cells are here. Heat killed. I treated with heat, killed it. Okay, heat killed with cells, S cells. Smooth strains are there. That does not usually cause a problem, right? Yes. I am going to add some proteinase into it. Proteinase. What is it? It's an enzyme that is going to digest all the protein. When I add proteinase into it, all the protein gets digested. Okay, if at all heat killed S cells has some proteins, those gets digested. Proteins could be the one that is getting transferred. That could be the genetic material that is getting transferred and basically making the organism have a coat around it, right? So what did I do? I put proteinase. Then I checked. So no proteinase there now, okay? Then I mixed this with this one, the R cells. I'm going to mix it with the R cells, the rough strains, and I'm going to see whether it's going to change or not. S cells still appear. That means the changing still was there. The thing that we wanted to kill is basically still there. That means it's not protein. It's not a protein, because if it was protein, this change would not have happened. If protein was the reason behind the change, then this change would not have happened because we have already digested all the protein with the help of proteinase enzyme. Okay? Okay, got it. Next thing let's do. Let's take again the heat kill S cells. Let's add RNAs. Let's add what? RNAs. Now, if at all I am adding RNAs, what is going to happen? The RNA is going to get digested off, right? Yes. Now, everything is digested from here. I'm going to mix it with the R cells again. We'll see whether transformation is happening. Transformation is still happening. S cell still is appearing. That means it's not RNA also, because RNAs could digest all of it. It's not RNA also. Now what do we have? We have the next thing which is left, which is going to be what? DNase. Proteinase we used. All the ACE means it's enzymes, okay? Proteinase we used, RNAs we used. Now we're going to use DNase. I'm going to check it. When I put DNase, all the DNA is going to get digested of the S strain, of the heat-killed S strain. Now when it is mixed over here, what is going to happen? No S cells appear. No S cell, no transformation is happening over here, which means that DNA is the genetic material. Now, did they say that? Avery, MacLeod, McCarthy. Avery, MacLeod, McCarthy. I know eclairs come to my head. I don't know why. But Avery, MacLeod, McCarthy, these three people, tell their name together only. It will be easy for you to remember. Avery, MacLeod, McCarthy. Okay. These three people said that DNA is the genetic material. Did people believe him? Believe them? No. People wanted one more proof. People said that we wanted one more proof. So method is what? Isolated components, proteins, DNA, RNA from the heat kill strain bacteria. Introduce each component to live R strain bacteria. Okay, wait. Just a second, okay? The method was not this. The method was basically, just a second. Sorry. Okay. The method was basically you had treated the heat-killed ones with proteinase first, then RNase, then you treated it with what? DNase. Proteinase and RNase, when you treated with that, it did not prevent the transformation. Okay, it did not prevent the transformation. Still the heat-killed S cells were converting into the R cells. Okay, DNase treatment inhibited transformation. When you were treating it with DNase, it inhibited the transformation. DNA is the transforming principle. This was being proved by Avery, MacLeod and McCarty. Okay, you have to write this. The first we have to take this heat-killed S cells, strains out there, treat it with proteinase. Then in that particular case, the protein is going to be digested. Then you're going to mix this one with the R cells. We see that still S cells are appearing, which means that this is not, protein is not the transforming principle. Second thing, same thing we have to repeat again. That's all. So this one would be more complicated. This is not wrong, but this would be complicated for you to understand. That's why I just cut it off. Cool. So this was almost around 1933 to 44. Okay, and you remember that was 1928. In case that you never get year-based questions from this particular concept, but if at all you're writing NEET and all, you could be there. Okay, so 1933 to 44. Over here, Avery, MacLeod, McCarty did the experiment. People were like, we need more proof. And finally came the last proof for the genetic material, and that was Hershey and the Chase experiment. Hershey and Chase experiment, you call it Alfred Hershey and Martha Chase experiment in the year 1952. So many years after 1944, over here 1952, this experiment was done. Now, they only had a confusion whether it's a protein or a DNA. That's all. We have to find that out. Okay, so for this they used an organism which you already know about. Okay, two organisms about which you already know. One is going to be the bacteriophage. Remember, the ones those eats or affects the bacteria, they are known as bacteriophage. What are those? They are nothing but the virus. They are actually virus. Okay, so which is the bacteria that they are going to affect? It is going to be Escherichia coli. So going to be Escherichia coli, or you also call this as E. coli. Our famous thing for all the experiments, we are going to use Escherichia coli. So over here we're going to take phosphorus also, P32 phosphorus. We know phosphate is a part of what? Your DNA. We already know about it. So it is going to make the DNA radioactive. Okay, now you have to remember these numbers. That is very, very important for you. Okay, S35. So 5 over here looks like S, so you can remember like that. And then there is P32. This is going to tag the protein, and this is going to tag the DNA. We know that. Hershey and Chase also knew that. Okay, now we are going to allow it to go and infect the E. coli. It infects over here, it infects over there. What happens in infection? What happens in infection if at all the virus and all is infecting? Basically, they put their genetic material inside the organism. Okay, they are okay to let go of the other things. They put their genetic material inside such that, okay, using this particular organism's machinery, the genetic material, this organ, so listen to me very carefully over here, I'll just repeat it so that there's no confusion. This organism wants to introduce its genetic material over here in Escherichia coli. Why? Because it wants to use the machinery that this organism already has for replicating, for increasing its own genetic material. So I am using someone else's machinery to basically increase my own genetic material. That is how it works. That is how any infection is going to work. Okay? Okay. And then they can make the baby viruses out there. You know, if you get genetic materials out there, basically you can say that the baby virus is also going to be formed later on. Okay. Now when it infects, it is going to pass the genetic material. So we do not know whether it's a protein or whether it's the, what do you say, is the DNA that is going to be passed down. Infection is being done. Second is blending. Blending is done such that in a way that after sending the genetic material, everything else which is there, it will be separated, it will move aside. Okay, it is going to be moving aside from the E. coli. That is known as blending. With the help of a blender, with the help of a machine, we do that. Next is centrifugation. With high speed, we make sure that everything is being rotated like this, such that depending on the density, they will be segregated. They will be moving down, moving up and things like that. Cool? The one that has higher density will move down. The one that has low density will be there at the top and things like that. Got it? Got it. So we had infection first, where we infected with the help of bacteriophage. Then we had blending. Then we had what? Centrifugation. Okay, okay. Now listen to me very carefully. Over here, after doing this, we looked out for radioactivity. Here you are going to get two very, very important things. Always remember, two very, very important things. The thing that settles down is known as pellet. And the thing that is going to be left behind is going to be known as supernatant. It is known as supernatant. Okay, so we have pellet and supernatant. In the pellet, you are going to have the cells because obviously the E. coli cells are going to be larger in size, which is why they settle down. Supernatant will have all the leftover things which is there, which probably is not being passed into the E. coli. Okay, okay. In the first scenario, we saw that there is no radioactivity seen in the case of pellet. In the first case, what did we use? We used protein. We used S35 to label the protein. We could not see that radioactivity being passed down to E. coli. That means the protein didn't go inside the E. coli. You get it? It was not found in the pellet. That means it's not there inside the E. coli. It is not being passed on. What about the supernatant? The supernatant was radioactive, means whatever was left behind was actually the protein which was left behind. That was not being taken inside the cell, that was being left behind. This organism didn't send it through. So that's not a genetic material. That is not genetic material in this particular, we have got it. But still we need to see this scenario also. When we checked this scenario, we found that radioactivity was seen in the pellet. What was radioactive over here? The DNA was made to be radioactive with the help of P32. If that is seen in the pellet, that means this particular DNA has been put inside the E. coli. That's the genetic material. That is the genetic material that is there, that is passed into the E. coli. You get it? So that means that DNA is going to be genetic material. What about supernatant? In supernatant, there was no radioactivity. In supernatant, there was no radioactivity because we didn't tag the proteins out there. Got it? Okay, so there are two different scenarios, and this proves that since you see radioactivity in the pellet that is inside the E. coli, that means DNA is going to be your genetic material, because the one which was tagged by DNA is showing this particular thing. This is known as Hershey and Chase experiment. I'm just going to revise it once more. What did we have? We had bacteriophage. We made this bacteriophage affect the E. coli, Escherichia coli. And we used two things. P32, phosphorus 32, it's a part of your DNA, so it goes and combines with your DNA molecule. Then S35, it combines with your protein. Both of them were allowed to infect. That's the first is infection. In infection, they are going to pass it genetic material. We do not know whether protein or the DNA. Then there is blending, where we make everything separated. Okay, and then there is centrifugation, where according to density, they are going to settle down. They're going to differentiate. Okay, now when it happens, what is going to be seen, Baba? You will see over here, when you see the centrifugation, in the pellet where you have the E. coli, you do not have the proteins. That means proteins have never been passed on. But in the supernatant, you have the proteins. That means they were left behind. They were left behind. They are not inside the E. coli. That's not genetic material. In this particular scenario, we see in the pellet, that is in the E. coli, we have radioactivity. That means your DNA passed into. Otherwise, there is no radioactivity that would have been seen. If DNA didn't pass into, then basically it will be there in the supernatant. Radioactivity would have been seen in the supernatant, but that didn't happen. Radioactivity was seen over here. That means DNA is a genetic material which is going to be passed through. This is known as Hershey and Chase experiment, and they are the ones who finally proved that DNA is going to be the genetic material. So we have three experiments, okay, Frederick Griffith's experiment, second is Avery McLeod-McCarthy, and the third one is going to be Alfred Hershey and Martha Chase. Three of these experiments are equally important, and three of them can be asked in different ways. So Yes. Got it? Clear? Experiments, all of it. I hope it's super clear to you. Done? Done. Now let's go ahead with the next thing which is there. Ma'am, why DNA? Why not RNA? Ma'am, why DNA and why not RNA? That is another very important question that we have. What could be the reason? Let's check it out. DNA versus RNA as the genetic material. So, listen. First, we need to know the features of a genetic material. That if you need to be a good genetic material, these are the things that you need to have. Then let's talk about whether it should be DNA or RNA. Okay, let's talk about whether it should be DNA or RNA. The first feature that it should have is replication. It should be able to replicate, as in it should be able to make its copies. You know, DNA makes its copies, RNA also can create its copies. Okay, so there is replication that is there for sure in both. Okay, second thing, stability. Chemically and structurally, the genetic material should be highly stable because it's a genetic material. It is the one that contains all the information regarding what proteins have to be made, and those proteins decide each and every character of our body. So it has to be stable. Now, if you had a talk about DNA and RNA before, and if you remember, RNA had ribose sugar with the OH group at the second position, making it highly unstable. Other than that, there's a nitrogenous base that is uracil, making it unstable. On the other hand, when you talk about DNA, you have deoxyribose where that oxygen is being removed, making it more stable. And you also have 5-methyl uracil. You are adding methyl group at the fifth position of the uracil, making it thymine to make it more stable. So we already know that in this case, so definitely DNA is the one. So replication is one property, because without replication, why is this a feature of genetic material? Listen, a cell is there. A cell is there, and the cell has to divide into two. When I say I have the DNA, okay, when the cell has to divide into two, but this DNA has to be copied also, no? This DNA has to be copied and it has to be sent in both the cells. Otherwise, who will give information within the cells? There has to be someone to inform, to take charge of everything, right? So you need replication to happen. That is why it's a feature and it's there in both. Stability, we know it's more in DNA. The third one is mutation potential. That means it should be, genetic material whichever is there, it should be able to mutate, have some changes. You should not be rigid. For example, I am like this from my 18 year, when I was 18 years old, I was like this, and still I am like this only. There's no change in me. Will that be okay? No, it's not okay. We as human beings have to change according to the situation. That is how we grow, right? It's a part of the growth. Similarly, when you talk about genetic material, if at all there are no changes, Baba, evolution is not going to happen. Evolution is not going to happen. And we cannot take that chance. We need evolution to happen, to make things better. Otherwise, we might not be able to survive. We might not be able to survive. We get it? So mutation potential should be there, but in a limit, not too much. So that the entire thing is mutated and not the same type of proteins are being made. So that is thing. In that particular case also, DNA is good. DNA allows mutation. Okay, in DNA we have changes. In RNA also we have changes, but RNA mutates even faster. Why? Because it is highly unstable. So in this case also, I feel DNA is far more better. Okay, last is expression, able to manifest trait as per the Mendelian inheritance. That means basically it should be able to express, in simple words, it should be able to express itself in the form of Mendelian inheritance. You know, Mendelian is a dominant trait, recessive trait. The dominant allele is there, it should be able to express itself. So it should be basically all the genes over here which are there, they should be able to express itself. Whatever genes are there in the genetic material, they should be easily able to express itself. That is expression. Both DNA and RNA are good at it. Okay, DNA carries the information, it transforms into the RNA makes the protein. That's the only thing which is there. Where you have a DNA has a dependence on RNA. DNA cannot directly make the protein. I told you. Yeah, transcription, remember? Right? It has to make an RNA, then only it can make a protein. That was the only thing that is there. But DNA is able to express itself at the end. Yes, RNA is able to do it even better. But okay, if at all we talk about the overall sense, who is better? Obviously, it's going to be the DNA. So what are the features of a genetic material? Important question, by the way. It's a 2-marker question. It's a 2-marker important question. It's put over here, right over here. Okay, replication should be able to replicate by itself such that basically there's a continuity. Second thing, it should be stable chemically and structurally. Third thing, there should be some mutation potential allowing changes to happen. And the fourth thing, it should be able to express itself in the order of Mendelian inheritance pattern. Okay, Now, RNA came first, so why DNA? What is the, okay, just a second. So, who is the better genetic material if at all we talk about it? Stability, DNA wins. Complementary strands rejoin after heating, lacks reactive 2OH group, has thymine instead of uracil. These two things you know. What is this? Complementary strand rejoin after heating. So, DNA has two complementary strands. If at all, if I heat it, they are going to separate. But after some point of time, they can coil back again with each other. Okay, they can coil back again. But these 2 points are super important ones. Okay, in terms of mutation and evolution, RNA mutates faster, but that's unstable. We do not want that. RNA viruses evolve rapidly. We know that. So in that case also I'll say DNA wins. Because DNA allows slow mutation. DNA allows slow mutation. This term is important. It allows slow mutation. That is the criteria actually. Next thing, expression of information. RNA directly codes for proteins. DNA relies on RNA for protein synthesis. Agreed. That is true. That is true. So in that wise, you will say RNA could be there. But two very important things are still lying, which is why DNA definitely is going to be the better genetic material. So we study the criteria of genetic material: replication, stability, mutation potential, expression. Replication means capable of self-duplication. Stability means it has to maintain the integrity across the life cycle. Okay, it has to be like that. Okay, mutation potential allows gradual means slow changes to happen that favors evolution. And last, expression manifests traits per Mendelian inheritance, as in the form of Mendelian inheritance. Conclusion: DNA is preferred for storage of genetic material because of its stability. DNA is preferred as a storage, as an actual genetic material because of its what? Stability. But when you talk about RNA, it's definitely good in terms of transmission of information. To make the proteins, at the end we want to make the proteins, that is the whole thing about. And RNA is going to be better in that. Both of them are equally good, but definitely DNA is a little bit on the better side for storing information, and that is very important thing. If at all you do not have your information secure, how can we make everything possible? So for that, you have DNA, and for transmission purpose, we say RNA is better. Cool? Cool. So it's like whenever you ask your mother, whom do you like more, your brother or me? My mother used to say both of them. So it's just like this. Okay, both. So now listen to me. So who came first? Was it DNA or RNA? You know that uracil had come first, then you made thymine. So obviously it has to be RNA. RNA was there from the starting point onwards. So was it only a genetic material or anything else as well? Listen, because of its reactivity, because it has 2 OH group, or like its second position, it has OH group, right? So that is what you call as 2 OH group. And also because of uracil and things like that, it used to act like a catalyst. Okay, as a catalyst, as a riboenzyme, which helps in conversion of A to B, okay, helps in increasing the pace of things and things like that, which is also the reason why it is not very stable, okay, which is also the reason why it's not very stable. So yes, it was a genetic material, but apart from that, it was also acting like a catalyst, which made things all a little bit trickier up there, which is why it was restricted from its position. Now, is that a problem? The answer is yes. RNA being a catalyst was reactive and hence unstable, which is why we thought, let's give the duty of storage and all to DNA only. Let it take care. Expression part, yes, you'll need the help of RNA. You please be there also. Okay, done. Now, comparison between DNA and RNA, this is just for your reference, is not there. Okay, sugar deoxyribose over here. In the case of DNA, so in the case of RNA, ribose has an extra hydroxyl group as an OH group. Here you do not have that oxygen. Okay, nitrogenous bases, thymine is present. There uracil is present. Function mainly acts as genetic material. Here it is involved in protein synthesis and also transmission, passing of the information. Types, only one main type is there in DNA. In the case of RNA, you will find multiple types. There is mRNA, tRNA, rRNA. We are going to talk about all of it right now. And stability, obviously, deoxyribonucleic acid is going to be more stable. It resists degradation. On the other hand, RNA, which is there, it's going to be less stable. It degrades very, very quickly. We studied about it, right? Right. Now, ma'am, how does DNA do something crazy? How does DNA do something crazy? The question was actually that I was planning to put was, does DNA do something crazy? So the answer is yes. If at all you are asking, does DNA do something crazy? Yes, it undergoes replication, and you might be in the state of awe when you see that. It's very, astonishing for just a molecule to do all of it. I'm going to talk about all of it just a second, okay? We studied DNA is genetic material, RNA is not. DNA is genetic material, RNA is not going to be something that is there for storage, right? Right. Okay. Just give me a second. Okay, cool. Did DNA do something crazy? Yes, DNA does something crazy, and that is semi-conservative DNA replication. This is going to be the 6th concept out of 13. Come on, we are halfway up there, okay? Semi-conservative replication, what do you mean by that? Listen to me very carefully. Over here, you have the DNA. Now you have 2 cells. You are having 2 cells and you want to make copy. What do? You give 1 DNA strand over here, 1 DNA strand over here. Ma'am, it should be having 2, right? Yes. So what exactly happens is that this strand which is there, I'll call it parental strand. I got it from the parental cell. This is the parental strand. This strand which is there is going to be the new strand. Depending on what's up over here, A, T, G, C and things like that, a new strand is going to be made. Okay, a strand would be containing T over here, T would be having A, C would be having G, and like that. A new strand is being made over here. Okay, so this is a parental strand and this is a new strand which is there. This is a new strand, which is why, because of this particular reason, Baba, I'll call this as semi-conservative mode of replication. I will call this as semi-conservative mode of replication. Why semi-conservative? Because half of it is being conserved. You're taking the parental trait, half of it is going to be new. That is why you call it semi-conservative mode of replication. Okay, now this was being found out. After replication completes, each DNA molecule has 1 parental and 1 newly synthesized strand. The scheme was termed as semi-conservative DNA replication. Remember, it was proposed by Watson and Crick only. They were behind the DNA double helical model they gave. They spoke about the semi-conservative replication model also. But obviously, when you talk in terms of that, my dear children, you need to know, again we need a proof. Without proof, we cannot believe whether it's semi-conservative or not. So people were behind the proof. Okay, so there was a guy whose name was, actually there were 2 people, Michelson and Stahl. Oh my God, so many experiments are there. My dear children, I feel bad for you. But you know what? Once you are thorough with this chapter, everything will be like a piece of cake for you guys. Listen to me, I promise you that. First experiments that we saw was Griffith, Avery, McCarty, and then Hershey and Chase. These guys are totally different, Michelson and Stahl. Okay, they were trying to prove the semi-conservative mode of replication experiment. That's all. They were trying to prove the semi-conservative mode of replication. That's all. Nothing else. So what did we do over here is suppose. Okay, you have to listen to me very, very carefully. So I had my DNA. Okay, should I be making like this? I'll be making like this. Okay. This is the DNA strand that I have. Okay. And I am actually growing this particular organism which is having this particular DNA strand. This organism is what? This is going to be E. coli. This is E. coli. Escherichia coli I am using for the experiment of Michelson and Stahl. Hershey and Chase also used Escherichia coli, if at all you remember. But along with it, you had bacteriophage, right? Okay, anyways. The medium in which I was growing was having N15 isotope of it. What isotope? N15 isotope. Nitrogen 15. You know that DNA has nitrogen. You know DNA has nitrogen, which is why you need to understand that it is going to take that up. I'm just sorry. I'm so sorry. Just a second. I'll write it this way. I was growing in N15 medium, which is why the DNA took that and both the strands were N15. Both the strands took that up. You had N15 nitrogen in both the strands. Cool. Now, I am going to centrifuge it. I am going to centrifuge it inside a medium which is of cesium chloride. Cesium chloride medium. Cesium chloride helps with actually, centrifuging or separating things on the basis of the density. Okay, so I can use cesium chloride medium where I am going to centrifuge this particular nucleic acid. This is DNA out there. When I try to centrifuge, what happens is that basically I will be seeing a band over here. I am going to see a band over here, which means that it's a heavy band. It's going to be a heavy band. Why? Because actually the N15 isotope which is there, it's actually a heavy one. And both the strands are of N15, which is why it's going to be heavy band that is being formed over here. It's sediments down. This is basically seen after centrifugation. After centrifugation. So what I'm trying to say is, there is E. coli, which I am growing in what? N15 medium. Obviously the the DNA which is there, it is going to take up that particular thing. Now, when I centrifuge this particular E. coli and everything out there, basically I can clearly see in the cesium chloride medium, they're going to settle down. Why? Because they have a heavy strand of DNA. Okay. Now, I'm going to do something crazy. I had this, okay. I had this, the same thing, E. coli itself. N15, N15-1. Now I'm going to change the medium. I'll make it N14 medium. I'll make it N14. What is going to happen in that particular case? If I change the medium to N14, what is going to happen? Let's check it out. It was seen that the DNA strand which were there, the parental strands, they separated. The new strand which was there, so the separated ones were N15, N15-1, N15, N15-1. Now the new strand which was there, obviously will use the nitrogen which is there in the medium. So they use this one, the nitrogen 14-1. They use this one, this is going to be what? The nitrogen 14-1. Over here, do you get to see the same strand that you found over here? Over here, did you see the same strand that was there? No. Okay, let's centrifuge it. Oh my god, my tongue is getting twisted. Centrifuge it and let's see what is going to happen. When I centrifuge it, okay, again, I am going to find a band over here. This shows that basically it's like a medium density thing that I see. Medium density. So probably it could be hybrid. One strand is 15, which is why it's a little heavy. One strand is N14, and N14 one is actually light. Okay, it has light density, which is why somewhere it is found in the middle. Cool. Now we are doing the experiment, and I'm pretty sure you will get to understand things. By the way, this E. coli divided into 2 other E. coli, and for that it required 20 minutes of time. It required 20 minutes of time. For every division, no, E. coli dividing into new E. coli babies out there, you need 20 division. Okay, 20 minutes. Fine. Now what do? We take this. We're gonna take this. So you have 1, 2, and I'm going to put it right over here. 1, 2. This is going to be N14. This is again going to be N14. This one which is there is going to be N15. This is going to be N15. I am going to grow them in a medium again containing N14 itself. N14 itself. The same thing I'm taking, and basically I'm making it divide. Again, how much minutes I require? 20 minutes. So if at all I'm counting from the start of the experiment, I'll say 20 minutes I required over here for division of this. Then now I require next 20 minutes, so I'll write it 40 minutes. I'll write it as 40 minutes. Okay, after 40 minutes, what I'm seeing over here. This is what I'm going to see. This strands divides into 2. One is going to be the yellow one. Okay. The other one which is there is going to be the blue one. Right? Similarly, this strand divides into 2. One the blue, and the other one which is there is going to be the yellow one. This strand divided into 2, this strand divided into 2. The new strands which are going to be formed over here are going to be what? Blue. All of them N14, here it is N14, N14, N14. This yellow ones which are there, they are going to be what? N15, N15. What do I understand? I don't understand anything, ma'am. Listen to me. Again, we're going to centrifuge it. What are we going to see? We are going to see that some of them are here. Some of them are over here. What do you mean by that? It means that basically some of them are going to be hybrid and some of them are going to be purely light. They do not have N15 at all. They're going to be the light ones. Okay, it is going to be the light ones. Have a lighter weight, lesser density. out there. What do you understand from this experiment? You understand one very important thing. That is, ma'am, if at all I would see a hybrid band over here, it means that the parental character is being passed on. The parental DNA which is there, it is being semi-conserved. Half of it is being conserved throughout. Because if not, how will I see something like this over here? I see it because one parental strand, both of them, they came over here, new strand is being created. That is why I'm seeing hybrid strand. If at all there was nothing like this, I will not see a hybrid strand. There will be, if at all, it's just new DNA that is being formed altogether without any parental strand. Without any parental strand, it will be what? Light. But there is no light over here. I see a hybrid one. Similarly, when I do this, when I again try to grow it, after 40 minutes, what I see? After 20 minutes, I saw this. After 20 more minutes, that is basically a total of 40 minutes, what do I see? I see that there is still hybrid band and there is still a light band. Why do I see the hybrid band? Because still it is being down, and again it's getting replicated semi-conservatively itself. That is the reason. That is the reason why I am able to see all of it. You get it? You get it? That's why I'm getting hybrid strand as well. So this was the experiment. It looks something like this. Okay, you had the N15, it's the easier way. I know I made it a little complicated out there. Here when you try to centrifuge it, it was N15 itself in the cesium chloride media. You try to centrifuge it. Then after 20 minutes, you used basically what? N14. You used N14 over here. What did we find? Basically the new strands which are going to there, they're going to take up the new N14 one, which is why you're going to see a hybrid strand over here. Now, here in this particular case, what happens? Again after 24 minutes, that is 20 minutes, that is basically 40 minutes in total. What do you see? Here they get separated, but the parental strand is still being there. It's still being there, which is why you see a hybrid band and you also see a light band. Very, very, very important. You have got so many questions from this. Please do not forget it. This is Michelson and Stahl experiment, and this is the proof for semi-conservative mode of replication. This is actually the proof for semi-conservative mode of replication. So was this the only experimental proof? The answer is no. Taylor and his colleagues, so there were a few people, Taylor and also his colleagues, they used radioactive thymidine. Over here you are using nitrogen isotopes, N15 as well as N14. There they used radioactive thymidine to trace the newly synthesized DNA in chromosomes of Vishya faba beans. I'm going to show you the picture because there's a question that has come from here. So it is not just Michelson and Stahl who did the experiment, but the first people or the first set of people who were there to prove it were them. Cool. And for that they required E. coli divides after every 20 minutes. Do remember that. Then apart from it, Taylor and his colleagues did an experiment with the help of radioactive thymidine, which was used to see the replication in the case of Vishya faba or faba beans. Okay, now we'll move on to the question. Okay, just a second. I really want you guys to solve the question first. Yes. Okay. DNA replicates semi-conservatively was first shown in. DNA replicates semi-conservatively was first shown in plants, Escherichia coli, higher animals or human cells. What do you think is the answer? Where did you see the semi-conservative mode of replication first in the form of experiment? It was E. coli. It was E. coli. The answer for this question is going to be this. This was the experimental proof which was being done. Next thing, experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in chromosome was performed on which plant? Experiment using radioactive thymidine by Taylor and his colleagues was done on which particular plant? This is the Vishya faba plant that you see, and Vishya faba, or you call this as faba beans, and on this plant you did the experiment. On this plant you did the experiment. Cool. Now, there are more questions. Don't be worried. Okay, it's not ending up there. This is again a KCET 2021. That was KCET 2021 question again. And this was KCET 2022, the first one we did. So many questions are coming from here. Okay, E. coli, important. Okay, we have to solve it together. E. coli fully labeled with N15. Fully labeled with N15 means both the strands are N15. okay, is allowed to grow in N14. Is allowed to grow in N14. The two strands of DNA molecules of the first generation bacteria will be, at the end of it, the two strands that are going to be formed, they will be like what? Same density and resemble with their parent DNA. Do you think they are going to be same density? Density is going to be hybrid, no? They cannot be same density, because if you're growing in N14 now, it's going to be hybrid strand that you're going to get. Just similar to the experiment, remember? You're going to get a hybrid strand. A new strand is going to be formed over here. It's not the same N15 that we used. It was N14, which is why the density is going to be changed. The density is going to be changed. So same density and resemble with the parent would be wrong statement. They do not resemble with the parent, and they are not of same density. Same density but do not resemble with the parent, they are not same density. Different density but do not resemble with their parent. They are different density, yes. Do they resemble with their parents? No. Their parents were N15, N14, N15, N15, but now you have N15, N14. They are not going to resemble their parents. Different density but resemble their parents? No. If you have different density, they do not resemble their parents. As simple as that. So the answer for this question is going to be what, Baba? It is going to be C. I would love to show you this also. If you have not understood this particular thing clearly, I'm just going a little fast over here. The reason being, we just spoke about it, right? This was the case. N15 when you're using, it was heavy density. Okay. When we are now growing in N14, the new strand that is going to be formed is green, which is why you're going to get a hybrid strand. Will it have the same density like the parent? No. Does it look the same? No. So that was a question. Okay. Okay. Now, next question, KCET 2017. If E. coli is allowed to grow for 40 minutes in a medium containing N15. If E. coli is allowed to grow for 40 minutes in a medium containing N15, then the number of N15, N14 containing DNA would be. If a E. coli is allowed to grow for 40 minutes, first 20 minutes to divide, then next 20 minutes to divide, containing what? N15. The medium is containing what? Sorry, you saw the answer. The medium is containing what? N15 itself. The number of N15, N14, how will you get N15, N14? When you are growing in N15 only, how will you get two different strands? You will get the same type of strands, no? So you are asked how many of them are going to be N15, N14? None of them. None of them. It will be 0. Why? Because you are not changing the medium. It's still N15 medium that you are using right up over here. Got it? Yes. That's all. Okay? 0. Now, okay, now the other questions are all related to DNA replication. So let's just go up at the top. Where is it? Okay. So that was about Michelson and Stahl experiment, and I hope you have got to understand the questions also. By the way, children, I hope this is helping you. I hope this particular series of one shot where we are doing questions and all type of questions are really helping you well. As I said, if at all it is, definitely let me know. I deserve to know, right? Yes. We are going to go ahead. Okay. Steps of DNA replication. Let's see that. Steps of DNA replication. What happens in DNA replication? So suppose over here, you have the DNA. Okay, one is in 5' to the dash, other one will be in 3' to 5'. And they will be having what? They will be having base pairs amongst here. If you want to do DNA replication, the first thing is that you would have to break this open. You would have to break this open and make sure that they are now separated. If this is a strand, you have to separate both the strands from here. You have to separate both the strands. This is the 5' 3' one and this is the 3' 5' one. This process involves an enzyme which has a name. It is known as helicase. It is known as helicase, an enzyme that is going to breaks the hydrogen bond. Breaks the hydrogen bond over here. Done? Now, that's the first thing. Second thing is that now when the hydrogen bond is being broken, this single-stranded DNA which is there, it has to stay intact like that. Otherwise, they will come again and basically they will bond back. So there are some single-strand binding proteins which are added up. Okay, there are some single-strand binding proteins which are going to be added up over here such that they do not bind back. Okay, we want replication to happen. For that, this thing has to be done, right? Right. Okay. Now the third thing is that, listen to me carefully, very, very carefully. You need an enzyme which is known as DNA polymerase. Okay, DNA polymerase means DNA polymer. Polymer means small unit of nucleotides come together to make a polymer of nucleotides. Monomer of nucleotides come together to make a polymer, means a chain of nucleotides out there. So, to make the DNA, you need DNA polymerase enzyme. Very, very important. Okay, very, very important enzyme that is known as DNA polymerase. And from here, I am going to have an enlarged picture. From here, I'm going to have an enlarged picture. So this is how it looks. Okay, we have put the helicase, we have used everything. This is going to be the 5'N, this is going to be the 3'N, this is going to be the 5'N, this is going to be the 3'N. What did you need? I told that you need DNA polymerase. What does it do? It is the one that is going to add nucleotides, that is going to add the nucleotides one by one, and make a complementary. Complementary means opposite. Okay, complementary means basically it's pair. It's pair. Make a complementary DNA. Okay, suppose over here when I say it's A, G, C, T, and then A, G, C, then you need to make a new pair, new strand over here. This is going to open up. Your DNA is going to keep opening up, and that's how you're going to get 2 copies. That's how you're going to get 2 copies. That copies basically can be given, can be passed down. At least you get it, right? That's the basic logic, right? That's the basic logic that we want. So over here, when we make a new strand, that is, for example, a new strand will be having T over here, A goes base pairs with T, G base pairs with C, so you will have C over here. So who brings 1, 1, 1, 1, 1? It's basically this DNA polymerase. So DNA polymerase says that I am basically going to work only in one direction. I will be working only in one direction. I basically cannot work in multiple directions out there. By the way, I did something wrong. Just a second. Here. I work in only one direction, and that direction is going to be, I'm just changing the polarity a bit. Okay, that direction is going to be, listen, so this one is in 3', this one is going to be 5', this one is going to be 5', this one is going to be 3'. A new DNA strand is always made in, always made in 5' to 3' direction. As in, if at all it is made in 5' to 3' direction, then this should be 3'. Then this should be 3' because always the DNA strands are going to be anti-parallel. Okay, so here it's going to be very normal to make a DNA. To make a new DNA, it's going to be very, very easy. Just know that. Okay. We're going to talk about the other strand also. But anyway, I was just telling the DNA polymerase is required to do so, and it is going to make only in 5' to 3' direction. Now, DNA polymerase says that, Macha, I am ready to make the DNA and all. Okay, you have already broken the hydrogen bond with the help of helicase. You have made it stable with the help of single-stranded binding proteins. And basically, I have myself also. But to start off this process, I have some tension. I need a set of nucleotides already up here. Basically, I'll say this particular structure which is going to be there is a primer. It cannot start by itself. DNA polymerase cannot start by itself. It needs a help. It needs a help over here. Initial segments should be there, then only it can take it along. Okay, this is known as a primer. And this primer is added up by an enzyme which is known as primase. These two things are very, very important. With the help of primase, you add the primer, then the DNA polymerase is going to elongate it, In which direction? From 5' to 3' direction, Okay, ma'am. So, ma'am, it has reached over here. Now what happens? It opens up again. It will be opening up again. So every point of time, only little portion will be opening up because it's a very high energy taking process. You will not open everything up, invest too much of your proteins for a single stranded binding proteins and stuff like that. This particular area where everything is happening right up there, you call this as replication fork. You call this as replication fork. It looks like a fork, right? This is a replication fork that you're having. And every single time when it reaches over here, it opens up and it moves on. Right? If you see, it's very smooth over here. In this particular strand, it's very smooth because it's in 3' to 5' direction, and basically your DNA polymerase is able to do everything. What about this strand? Have you ever thought about it? It's in, okay, what about this strand over here? Suppose this is a replication fork at this particular point of time. If I want to make a new strand of DNA, what do? I add a primer first. Here, why? Because DNA replication happens from 5' to 3'. So this is from 3' to 5'. So this side, from here only it can begin. So suppose it began. From here, it was able to make. Suppose it was able to make from 5' to 3'. Now when it opens, what happens then? When it opens, what happens then? So suppose it opens. Okay, suppose it opens. Over here, you had the primer on this side, you made it. Now you had the primer, you made it. Again a gap is there. What do you do with that? Because it has got opened. Again, Baba, you need to add the primer. Again, it has to start the process of replication from here. So this strand where everything is going on really smooth, okay, you call this strand as leading strand. What do you call this? You call this as leading strand. It is leading the game. Here where everything is going to be slow, I am going to have a name. It is known as lagging strand. What do you call it? Lagging. So there is a leading strand and there is a lagging strand right over here. And these fragments, if at all you see, the DNA fragments are going to be small. These small DNA fragments which are going to be formed because of like multiple distributions out there, you call these fragments as Okazaki fragments. Okazaki fragments. Later on you have to remove the primers. Later on you are going to remove the primers out there and you have to join the DNA things. Always remember, the one that is responsible for joining all the nicks and breaks out there, you have one more enzyme that is responsible for that. That is known as DNA ligase. It is known as DNA ligase. It is the one that joins all the break points and everything out there. Cool? Very important. So what did we study until now? We have studied that DNA replication is going to be a semi-conservative mode of replication, and when it does, Baba, you will be seeing a leading strand and a lagging strand. First of all, you have to separate them using helicase enzyme, which is going to break the hydrogen bonds. Then you are going to bind them with the help of single-stranded binding proteins, which is going to make it stable such that it does not bind back. Okay. Then further, what you're going to do is you would need the help of primase to add a primer over here. This primer is usually RNA primer. Later, you have to remove it. But anyways, such that it helps in the starting point for DNA polymerase. The DNA polymerase will make a new strand in only one direction, 5 to 3' direction. That means the other strand would be 3 to 5. Got it? Here in this particular strand, that does not take place because every time the replication fork open, you have to make it again and again and again. That is why you are going to get multiple fragments of DNA, which is known as Okazaki fragments. Okay? Later on, Okazaki fragments, the primers are being removed and the DNA polymerase extends everything. If at all there is any nooks and cricks out there, DNA ligase is going to join it. Okay. Yes. One more very important thing. This DNA polymerase which is there, I'll call it DNA dependent DNA polymerase. DNA dependent. It is dependent on a DNA to make a new DNA strand. That is why I call it DNA dependent DNA polymerase. Okay. Okay. Can we read it once? Can we read it and try to understand it? Initiation, the starting of the process. If at all you have been asked, write the steps of DNA replication, because this is also an important question. Every part of this particular chapter is important, okay? So DNA replication, initiation, how do you write? DNA helicase unwinds the double helix at the origin of replication. Ayyo, I didn't mention that at all. So listen to me. The whole process can only happen at specific location. The whole process of starting of the replication process can only happen at certain areas where your DNA polymerase can come and bind. Otherwise, those other areas it will not come and bind and all. Okay, so this areas where a DNA polymerase will come and bind and do the process is known as ORI. It is known as what? The ORI site. ORI stands for origin of replication. It's known as origin of replication site. Very, very, very important. Cool, ma'am. Got it, ma'am. Okay, so initiation, DNA helicase unwinds the double helix at the origin of replication. Single strand binding proteins keep the strands apart. Primary side RNA primers to start the replication. Primers, yes, done. Elongation, after initiation, after everything is ready, elongation. DNA polymerase adds new nucleotides in 5' to 3' direction. Leading strand is made continuously. Lagging strand is made in Okazaki fragments. DNA ligase joins the fragments together later on. Done. Last is termination. When does termination happens? Suppose from there replication is happening, from here also replication happening. Two replication forks, when they come together, that's when termination can happen. Or basically at the end of it, when this particular whole thing gets completed, that's also when it can end up. So replication stops when two replication forks meet or at specific termination sites or at specific termination sites. These are what the steps of DNA replication that you need to know. Got it? Clear? And we had revised it also, so I don't think we have to go again and revise. Origin of replication, please not forget that, huh? Okay, done. Now, which enzyme replicates DNA and what does it need? What does it need? So enzyme, we know it's DNA dependent, DNA polymerase. Catalyzes polymerization of deoxynucleotides using a DNA template. Your template or DNA template only has been used to make a new DNA strand, right? So that is absolutely correct. Let's study about it. Efficiency. So efficiency, I'll come to that, wait. Direction, DNA polymerization happens only in 5' to the right direction. Done. Okay, origin. Replication requires a definite origin of replication. I told you ORI site. That is there. Only then the DNA polymerase is going to attach. Okay, and only that point. Accuracy, high precision is required. And usually it is highly precise. If at all not, your body has DNA repair mechanisms where it repairs things. If it is not able to do that, that's when we have problems. That's when we have diseases and issues like that. Okay, this is very, very important. Cool. Now over here, energy source. What do you mean by energy source? Here it is written deoxyribonucleoside triphosphates act as substrates and energy providers. So we didn't study about it, listen to me. Over here, when I say that they bring nucleotides, how are these nucleotides looking? Until now, we have not discussed about that yet. Your nucleotides which are there, always remember, okay? So where shall I make? I'll make it here. The nucleotides, they look like this. The first carbon, second carbon, third carbon, fourth carbon, fifth carbon. First carbon, you have the nitrogenous base, glycosidic bond. Fifth carbon, you have the phosphate group. Actually, when a nucleotide comes, it is going to be having 3 phosphate and not 1 phosphate. It usually comes with 3 phosphates. Okay. Now, when another nucleotide comes and when it bonds with this particular phosphate, that's when this bond breaks. They have a breakup over here. This condition before the breakup is known as what? Deoxy. Okay, deoxy, whatever nucleoside it is, deoxyguanosine, or if at all it is G, it is deoxyguanosine triphosphate. You call it triphosphate. Okay, so when it comes, what is the substrate that we have? What does the DNA polymerase bring? They bring this. They bring this only. Then what happens when the nucleotides are stacked up together? There is going to be, because you want to make it free, you have to break this bond such that it will be becoming free. When you break this bond, what is going to happen? A high amount of energy is going to be released. A high amount of energy is going to be released for the next bond and everything to be made very, very easily. So they are the ones who are energy providers. They provide energy, okay? And they are the ones which acts like substrate also. With the help of them only we can make the new DNA strand. Okay, so this is what deoxy nucleoside, guanoside, okay, guanosine, right? Remember, deoxyribonucleoside triphosphates. They are going to be acting like substrates and also energy providers. Very, very important. Do not mug up. Deoxynucleoside triphosphate, deoxyribonucleoside triphosphates. Done. Now, this is a very important MCQ question. Okay, they have spoken about the efficiency of your DNA polymerase, and they have mentioned that in E. coli, okay, it is highly efficient. In E. coli, the efficiency is, E. coli has a genomic size of 4.6 × 10⁶ base pairs. It replicates in 18 minutes, tentatively 20 minutes. Okay, that's a general number. And how many base pairs are added per second by DNA polymerase? Just imagine. 2000 base pairs. 2000, 1 second you take to blink your eyes. 2000 base pairs are being added up during the process of replication. 2000, not 200, 2000 base pairs. This is a question. 200 sometimes can be put, and things like that will be there. Always remember 2000 base pairs per second is the answer for this particular thing. Do not forget it. Cool. Works? Yes, ma'am. Works perfect for me. Then that works perfect for me also. So this was all about it, and my dear children, I hope you have got it. And now over here I had a question about DNA replication. No? Let's solve that. This is a KCET 2017 question. Polymerization of DNA nucleotides during the synthesis of lagging strand occurs in which direction? In lagging strand, DNA synthesis occurs in which direction, my dear children? 3' to 5', 5' to 3', any direction, promoter or terminator, promoter to terminator direction. Polymerization of DNA nucleotides, that means a new DNA strand is being formed in which direction? That is the question. A new DNA strand is being formed in which direction? 3' to 5', 5' to 3', any direction, or promoter to terminator. The answer for this question is going to be 5' to 3'. Anything, whether it's leading, lagging, anything. DNA polymerase is going to work, and I'll tell you a secret. RNA polymerase, who makes the RNA, is also going to work in this direction only. 5' to 3'. There is no change in this. Got it? Clear? Okay? Okay, ma'am. Fine. So that's how your DNA, replication process is also done. That's a crazy thing that it does. And as I told, it's not a brain cell. It's just a molecule which is doing all of it. And in our body, it's happening. Isn't that crazy? Yes. Now, the next part, by the way, if at all you want to take a break, quick break, you can. Okay, I cannot, so I am just going ahead. And my dear children, I really hope that you're understanding things really well. And this particular class is helping you as well. Yes, yes. So let's go ahead with the next thing that is there, and that is, ma'am, how does DNA actually send its message out of the nucleus? Ma'am, you spoke about replication and all, agreed. Then you spoke about one more very important thing, that it makes the protein. It makes the protein, right? We spoke about a lot of things until now. Which was, first was the organisms, how many chromosomes and number of DNA, sorry, what's the genomic size and blah, 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 blah. We spoke about how DNA is packed. We spoke about, who gave the information that DNA is a genetic material. So who is better, RNA or DNA as a genetic material? We spoke about this also, semi-conservative DNA replication. Everything is around DNA replication. We know it's important. And you also said one more thing, DNA is important for making proteins. But that protein formation does not happen. That protein formation does not happen within the nucleus. In that particular case, someone has to go outside. Someone has to go outside, right? So listen, if you do not understand still, let me tell you. If this is your cell and this is your nucleus, where is my nucleus? Okay, just a second. This is going to be the nucleus. Your DNA is right over here. Your DNA is right over here. Okay, and you want to form proteins. You want to form proteins. Where are proteins being formed? For protein forming, you need machinery. What is a machine? Ribosomes. What is a machine? Ribosome. Where are they? Ribosome is there in the cytoplasm. Can the DNA go out? No. That's a big problem. DNA has to be in the safety vault. It cannot move out. So what do? If you want to send a message, basically we make an RNA out of the DNA. we pass this information into an RNA, and RNA basically can move out and help in the formation of a protein. That is how it was working. That is how it was working. And I told you one particular thing. From the DNA to RNA, if at all you want to make this, if you want to make a RNA from a DNA, this process is known as what? Transcription. It's known as transcription. Okay, now the RNA comes out over here. RNA, whatever information is there in the RNA, gives information to make the protein. This particular process is known as translation. You want to translate the message. No, one guy takes the message. That is your messenger RNA. This process of formation of RNA is known as transcription. Later on, this RNA, giving the information to make protein, that is you're translating. That is why it is known as translation. But anyway, the question is, how does DNA actually send its message out of the nucleus? As I already mentioned, it's with the help of RNA. And before I go ahead with that, if you have a little bit confusion also, I think this could help you to understand it. Library rule. The nucleus is a library where books, DNA, can never leave the building. Can you take the book forever? Okay, if there's only one book out there, that teacher will, librarian will not give you that particular book. So what do? You can either make a photocopy and let it go. And you can take it, okay, but you cannot take the book. So your real book is there in the library, and library is what? Your nucleus. You can take the photocopy, that is your mRNA, and that mRNA can be carried to the workshop for whatever thing is there. Okay, not for the workshop, basically that can be utilized by the ribosome, to go ahead with the whole process of making up the protein. So the next concept that we have, the 17th concept out of the 13th, is the transcription of DNA, and it's one of the most famous thing which is there. Look at this particular diagram. This is your DNA. It gets transcribed to RNA. RNA moves outside the nucleus, and here in the cytoplasm, the translation process is going to happen to form the protein. This is central dogma. Okay, central dogma. Do you know central dogma? As in, centrally, this is a rule that we have to follow throughout the world. And the rule is, DNA which is there, it will form the RNA. RNA which is there, it is going to form the protein. Okay, DNA helps in the formation of RNA, RNA helps in the formation of protein. That is how it goes. This is what central dogma. It is asked to you to write the central dogma. So how do you write? You write it like this: DNA to RNA. To RNA. What do you call this as? Transcription. Okay, RNA to protein. What do you call this as? Translation. So you have transcription and you have translation. Okay, and DNA to DNA, this is replication. This is the central dogma. Without it, nothing is happening. This is the central dogma that has to be followed. Exceptions are there, but not in this chapter, don't worry. Francis Crick was the person who proposed the central dogma of molecular biology. You know Watson and Crick? In that, Francis Crick who was there, James Watson and Francis Crick. So Francis Crick was the person who gave the central dogma, which is accordingly, like as you can see over here, right. Now, how does, or what does the transcription unit contain, or basically how does things happen? So listen to me carefully over here. This is, okay, what does the transcription unit contain? This was actually a question also. This is the strand that we are having. Okay, this is a 3' end. This is going to be the 5' end. You usually put it at the top only, generally. This is going to be the 5' end, and this is going to be the 3' end. Opposite. Anti-parallel, anti-parallel. Okay. Now, here, you're going to have something which is known as the promoter region. Promoter region. Why, ma'am? What does it promote? Why it's there? It is your DNA. By the way, it's your DNA, right? Yes, it's your DNA. And here we have a promoter region. What does the promoter region do? Baba, it's a region, a specific sequence of DNA out there, which allows, okay, which allows RNA polymerase. Until now we had DNA polymerase because we were making DNA, but now we have RNA polymerase over here to make RNA polymerase. It allows the RNA polymerase to make what? The mRNA. It helps in the formation of the mRNA. General, or basically we can just say RNA. Cool. It helps in the formation of RNA. So that particular region where it comes and attaches is known as promoter region. From this point only, the process is going to begin. Okay. Similarly, at the end over here, you have a terminator region also. You have a terminator region as well. What is the thing with terminator region? This is where, okay, suppose RNA polymerase is here. It is coming. This is RNA polymerase. And here you are making the RNA. This strand is being used. Okay, suppose. Okay, for that I have to talk about a lot of things. Okay, one by one, let's go then. This is the promoter. That is the terminator. That's where the transcription is going to end. I'm going to show you how transcription happens. I think that might clear a lot of things. Okay, in transcription, only one strand is going to be taken into consideration. And that strand which undergoes transcription is known as the template strand. Okay, what do you call it? You call this as the template strand. It is going to act like a template for us, okay, onto which we are going to do experiments. The other strand which is there, which is not doing anything much, is known as the coding strand. Now, children generally get confused. Coding is the one that should code the RNA and all. No, but it's a template strand that is going to act like a template that is going to help in the formation of an RNA. Cool, cool. Okay, listen to me very carefully. In the template strand that we have, here you have the coding strand also. In the template strand that we have over here, what is going to be seen? Let's check it out. First of all, your RNA polymerase is going to come and attach here in the promoter region to begin up the process. When the RNA polymerase comes, it says that I am not going to do the thing alone. I am really shy. I cannot come and basically join the promoter region like that. Okay, before I join, I need the help of someone. So basically, when RNA polymerase comes, it comes along with a factor which is known as sigma factor. It comes along with sigma factor. So both of them, they're going to come. The sigma factor is super important such that the RNA polymerase will come and bind over here. Without sigma factor, it is not going to bind over here in the promoter region. Now, the RNA polymerase, you have to use your imagination. It is going to move. Suppose it's not there now, it's moving. While it moves, the sigma factor goes away. So the initial shyness we have now, we take our friends and on to staff room to talk to teachers, and then we forget things. Right? Usually it used to happen, but now I don't think so that happens. But yeah, so that is how generally it works. Okay, in this particular case, initially it was there, then the friend left. Now the RNA polymerase is moving, and when the RNA polymerase moves, what is going to happen? What is going to happen? That is very important. So this is the A. Suppose I am putting certain things over here. AT, GC, AA, TT, CC, GAT. This is the DNA strand, suppose. 3' to 5'. RNA polymerase says, I am also going to be just like DNA polymerase, I will also work in 5' to 3' direction. So the new strand which is going to be formed over here. So the new strand which is going to be formed over here is going to be in which direction? 5' to 3' direction. And it is making up the new strand while it is moving. While it is moving, it is collecting one more. What do you say? Is it deoxyribonucleoside triphosphate, or it will be just ribonucleoside triphosphate? Just ribonucleoside triphosphate. There is no deoxy over here. You are making an RNA, right? So it will be ribonucleoside triphosphates that you're going to bring one by one. First of all, the first thing that you have to note over here is A is there. If A is there, what will be the base pair that you can see over here? Ma'am, T. Array, it's RNA, it cannot be T, it has to be U. It cannot be T, it has to be U. So over here it becomes U. T is here, ma'am. T base pairs with A, that's not an issue. T base pairs with A, where is it? Poya. Just a second. T base pairs with A, that is not a problem. G, C. C, G. A, U. A, U, T, A, T, A, C, G, C, G, G, C, A, U, T, A. That is how your RNA is going to be formed. Your RNA polymerase came and attached on the promoter, but before that, you need the help of sigma factor for coming and attaching. Post that, here from this point onwards, you're going to do the transcription process where you are going to carry along 1 ribonucleoside triphosphates are there. They're going to join, they're going to join, they're going to join, they're going to join, they're going to move. Now, once your RNA polymerase reaches up over here. Now it wants to end up the process. Now for ending up the process, it needs the help of another factor which is known as rho factor. So you can remember it's crying because it's the end of it. It's terminating the whole process of transcription. It's the end. DNA and RNA were together until now. Now they are ending up their relationship. So there is rho. So initially chi, so sigma factor, and over here then rho factor is there. Finally, what happens? This template, this RNA which is there, it moves. Okay, we have got the RNA. This DNA which is there, it combines together back. Here, where your actual everything is happening, this particular part which is there is known as the structural gene, which is actually getting transcribed. You call it as the structural gene. So how many parts do you have in a transcription unit? First part, promoter region. Second part, structural gene. Third part, what do you have? The terminator region. Apart from it, you have the template strand in 3' to 5' direction. Then you have the coding strand in 5' to 3' direction. Okay, ma'am. Cool. For starting up this process, you need what? RNA polymerase along with sigma factor. It comes and acts on the promoter and basically helps in the formation. At the ending, you need rho factor. At the end, you are needing rho factor as well. Got it? Generally, you say, when you talk in terms, where is the promoter region? You use the reference of coding strand, because coding strand anyway is not used anywhere. So you say it's in the 5' end. It's towards the 5' end of the coding strand. Where's the terminator region? It's towards the 3' end of the coding strand. You do not take the reference of these two things, 3 and 5 of the top strand. Okay? You will not take that. Cool. Whenever you make an RNA after transcription, have uracil instead of thymine. Got it? And make the RNA in 5' to 3' direction. Last thing, RNA polymerase was doing this. This RNA polymerase is also DNA dependent. It's a DNA dependent RNA polymerase. It is depending on the DNA to make the RNA. Got it. Now let's see it. So this is a picture that we just drew. Okay, promoter, where the RNA polymerase binds, sets the direction. Okay, structural gene, the stretch that actually gets transcribed. Terminator, they marks the ends of transcription. Okay, plus additional regulatory sequence may sit further upstream or downstream of the promoter. That means basically beyond it or after it, there might be some additional regulatory sequences as well. Transcription is done by DNA dependent RNA polymerase in 5' to 3' direction. Studied. These are the things that is given in your NCERT. Right. Now, if you're asked about transcription in the 3 acts, if at all you have to say initiation, elongation, termination, how do you define it? How do you explain it? Initiation, RNA polymerase binds the promoter with the help of sigma factor. It is going to bind the promoter with the help of what? Sigma factor. If at all sigma factor is not there, basically you should not be expecting binding also. So it binds with the help of sigma factor. Then what happens? Baba? The DNA helix opens and transcription begins. The DNA helix which is there, it is going to open up and the transcription is going to begin. Then elongation. RNA polymerase add nucleoside triphosphate. What nucleoside triphosphate? Actually ribonucleoside triphosphate. But okay, it's written as nucleoside triphosphate. That is also fine. Okay, it should not be deoxynucleoside. That's something important. Following complementary base pairing, only a short stretch of RNA stays bound to the enzyme. So everything else is like lying in the air. Okay, so it's like lying in the air while it moves around. Cool. Last, you have termination. At the end, at the terminator region, the rho factor, RHO, factor, causes the RNA to fall off. RNA polymerase detaches and transcription gets completed. Khatam. Tada. The process is done and dusted. That's all about transcription. And over here, you have few questions and few more things will be there attaching. We are going to talk about it, don't worry. So in prokaryotes, the transcription of DNA is initiated with the help of. In the prokaryotes, the transcription of DNA is initiated with the help of rho factor, elongation factor, sigma factor, termination factor. Is initiated. Sigma. S for shy, it is what was shy, it needed a help, and that help was given by sigma factor, right? So it is going to be sigma factor. Next thing, for the given sequence of DNA, identify the complementary sequence of base on the mRNA from the options given below. This is KCET 2020 question. You have been asked that you have to find the complementary base on the mRNA. It's an RNA that you have to form from this particular DNA strand. First of all, when you make the RNA over here, if it's in 3' to 5', the RNA is going to be in 5' to 3'. So this one goes off because this is in 3' to 5'. Okay. Now, here it is A. A is going to base pair with U, not T. It's going to base pair with U, not T. This is mRNA. Messenger RNA, it is going to be U, RNA, U. So only one starts with U. This would be the answer. This is G and all, this is something else. This is the only answer. Here T is there instead of U. This is going to be a DNA, complementary DNA strand, not an RNA. You get it? So you have to be careful when you get questions like this. Okay. Now, we have few things, okay? Prokaryotic and eukaryotic transcription process. Prokaryotes as in E. coli and everything, and eukaryotes such as us, as I told you. In prokaryotes, it is polycystronic. Multiple genes code for multiple protein. What do you mean by that? It means that, basically, if I talk about one particular structural gene, okay, one particular structural gene, here promoter, here terminator, here structural gene is there. This structural gene can have multiple parts. This can code for gene 1, this can code for gene 2, this can code for protein 1, this can code for protein 2, this can code for protein 3. This is known as polycystronic condition. Multiple gene code for multiple proteins over here. But when we talk about us, eukaryotes, if there is promoter and terminator, in between Baba, there is only one thing, one gene, and that one gene codes for only one protein. This is known as monocystronic. Polycystronic, multiple cystrons, multiple genes coding for multiple proteins. Over here, monocystronic, one gene coding for one protein. You can remember P for prokaryotes and P for this, polycystronic. P for prokaryotes and P for polycystronic. If you wish, you can remember like that. Got it? Got it. Now over here, prokaryotes. Prokaryotes do not have introns. Okay, prokaryotes do not have introns. What do you mean by that? Listen to me. Until now, we didn't study about introns and all. Okay, we didn't talk about introns at all. So listen, here when you talk about the DNA, and when I say that DNA is being made into an RNA, mRNA, and this mRNA later is going to make the protein. So what usually happens is, okay, what usually happens is, in the DNA, you will be having certain coding and non-coding regions. Certain regions that code for some proteins, certain regions that do not code for anything. The region that codes for proteins, you're going to call them as exons. But before I go ahead with that, listen, we assume DNA, then mRNA, then protein. This is how it works, but generally this is not how it works. Generally, we have DNA, we have something that is known as hnRNA. Okay, we have something that is known as hnRNA. Then we have something that is known as mRNA, and then we have something that is known as protein. mRNA, and then we have something that is known as protein that is being made. So what is the difference between this and this? Listen, here, as I said, there are some regions that codes for proteins, you call them as exon. And there are some regions that do not code for proteins, you call them as what? Introns. So you have exon, intron, that is how it goes. Okay, when you transcribe, okay, when you say transcription is happening, directly mRNA is not being formed. Instead, you get an hnRNA, heterogeneous nuclear RNA, hnRNA. So here, if at all I had exon, here also we have exon. Here if at all I had intron, here also I have intron. Here also if I have exon, here I have exon. Here if I have intron, here also I will have intron. This is how it works. All the things are getting transcribed. Now, mRNA is something that is going to code for proteins, that is going to be responsible for formation of proteins. Later, it is going to get translated over here. It is going to undergo translation over here. So, it has to be very specific. You do not want this and this. Who would want this? Does anyone want the introns? No one wants the introns. You just want the exons. So, what do you do? The hnRNA which is there, it is going to undergo a process which is known as splicing. Okay, it is going to undergo a process which is known as splicing, where the removal of the introns happen, where the removal of the introns are going to take place. So the introns, when they are removed,
what is going to happen? You will be just getting the exons, and that is exactly what is there in the mRNA. You just have the exons and nothing else. Just have the exons and nothing else. These exons are later going to form the protein. Got it? So we have removed the introns. We called it as what? Splicing. We called it as splicing. We have removed the introns. Now we just have the exons. And that exons are responsible for the formation of proteins at the end. This is something that we know. This is something that we have studied right now. Okay? Now in the case of prokaryotes, introns are not there, Baba. In the case of prokaryotes, they do not have introns. So do they require splicing and all? No. In the case of eukaryotes, we have introns, which is why they require splicing. Okay, so that is written over here. First of all, they are polycistronic. If we talk about one structural gene, they have multiple cistrons, means multiple gene coding for multiple proteins. Okay, here in our case, we are monocistronic. Prokaryotes do not have introns. In our case, we do have introns, okay, which is why we have to remove them with the help of splicing. Transcription and translation occur in the same area, the cytosol. What do you mean by that? Baba, listen to me. Until now, we studied over here in this particular diagram that this transcription happens in the nucleus, translation happens outside. Why in the nucleus? Because DNA is over here, right? In the case of prokaryotes, do they have a nucleus? Do they have a nucleus? No. So transcription will happen in cytoplasm. Translation will also happen in the cytoplasm. Both of them simultaneously that can take place. Okay, in the case of prokaryotes, simultaneously both of these procedures can take place. But in the case of eukaryotes, transcription and translation are in a different area. One in the nucleus and other in the cytoplasm. Again, there is no need to mug up. Imagine our cell, we have a nucleus within which cytoplasm, within which a transcription is going to happen, in the nucleus, and in the cytoplasm there is going to be translation. Done? Done. In this case, when I say both of them are occurring in the same area, I can use a term. And what is that term? I say this is a coupled process. This is going to be a coupled process. Works? Cool? Done? Fine. Once. Okay, now in pea plants, each character is governed by a gene, which is, this is a question bank question from second PUC. In pea plants, each character is governed by a gene, which is monocistron, polycistron, operon, intron. What do you mean by that? Pea plant, pea is a plant, and plants are basically eukaryotes or prokaryotes. Only bacteria are prokaryotes. Rest everything is eukaryote, whether it's a fungi, a plant, or animal. Okay, so here they are asking you, in a eukaryote, a gene is going to be monocistronic or polycistronic? Answer is what? Monocistronic. How can it be polycistronic? P is prokaryotes, right? Right. So that is how we do it. Now, there's a thing, that is the last concept about transcription, after which probably we can have a quick rest. Okay, we can take a quick rest over here for some time. At least me, my legs are paining so much. Okay, so eukaryotes, RNA polymerase and RNA processing. Listen, first of all, before I go with the different type of RNA polymerase, listen to me. RNA processing is there in our case. We know splicing, I told you removal of introns. Apart from that, you have just made the RNA. This RNA has all the crucial information. It is leaving the nucleus and going into the cytoplasm. Everyone inside the nucleus is saying that RNA, please wear the cap. Please wear the cap because you're going outside, just like how we have. Similarly, Baba, the thing is, outside when you go, there are so many enzymes which are there, which do not know. It's something foreign. It can be attacked. So there is a process that is known as capping. There is a process that is known as capping, where what you're going to do is, at the 5' end, of the RNA, you are going to add methyl guanosine triphosphate. You are going to add methyl guanosine triphosphate at the end, capping, such that that particular area has been safe and no one cuts it off. Similarly, at the other end, 3' end, you add so many adenine, okay, you add so many adenines. So it is known as 200 to 300 adenylate residues are added. This is known as adenylation. 200 to 300, you say poly-A tail, okay, you also call this as poly-A tail. Poly-A tail. So if at all you see this particular picture, I think that could give you clarity. When this guy is being formed here itself, okay, so this methyl guanosine, okay, methyl guanosine triphosphate is right over here. Methyl guanosine, not guanine, methyl guanosine triphosphate, that is a nucleoside. methyl guanosine triphosphate is being added up over here. Apart from that, in the tail region, you see poly A tail. So many adenylate residues are being added up such that when they move out, they are not being cut. So there are 3 types of RNA processing that is going to take place. Okay, 3 types of RNA processing that is going to take place. The first one is going to be splicing. The second one is going to be capping. The third one is going to be tailing. Okay, first one there is going to be splicing, second one is going to be capping, and the third one is going to be tailing. These are the 3 things that are going to take place. Got it? Got it. Okay, ma'am. Now listen. Prokaryotes, RNA polymerase and RNA polymerase. RNA polymerase and RNA processing. So this is the processing part that we have studied. Now we talk about the polymerases. In the case of us, okay, in the case of eukaryotes, we have 3 different type of RNA polymerases. In them, in the case of prokaryotes, they just have one and they're really happy with that. Okay, they just have one and they're really happy with that. We, we do everything extra, so we have some extra thing also. Okay, so we have some extra thing. Here, in this particular case, when I talk about RNA polymerase, we have 3 RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Okay, what does the RNA polymerase 1 do? In the prokaryotes, do we have it? No. Okay, RNA polymerase 1, ma'am, makes the rRNA, and I really want to use a different slide for this. So what do? Can I go back here? Yes. In prokaryotes, do we have it? No. So we can add it over here. In their differences, no, prokaryotes have just one RNA polymerase. Just one RNA polymerase. Now over here, they have multiple as in 3 RNA polymerase. And they are responsible for making 3 different type of RNAs also. So let's go ahead one by one. The first RNA polymerase that we have over here. Okay, the first RNA polymerase is RNA polymerase 1. I'm so sorry. RNA polymerase 1. The first priority is given to the one that starts with R only because it is R. So rRNA, you call this as ribosomal RNA. You know ribosomes which are there, they are responsible for helping in the formation of protein. The ribosomes are made up of RNAs and also proteins. rRNAs and also protein. So this is one rRNA. Okay, rRNA, there are different types, subunits. So we say 28S, this is one. 18S, that is another, and then there is 5.8S. Everything ends with 888. One is 28S, other one is what? 18S, and the other one is going to be 5.8S. These 3 different type of rRNA is going to be formed by RNA polymerase 1. Now we have RNA polymerase 2. RNA polymerase 2. The second priority is actually given for the transcription of hnRNA. Remember, first we make hnRNA. That is how generally works. hnRNA. So first we make hnRNA. That transcription is being done by RNA polymerase 2. Second priority. First, so rRNA only same letter. No, that's why. Okay, this is also known as precursor of mRNA. This is also known as precursor of mRNA because after this only you get mRNA. Okay. Finally, there is going to be RNA polymerase 3. Ma'am, why are you teaching all this? Because this comes in examination. Otherwise, why do you think I'll teach it? RNA polymerase 3, T. 3T. So basically tRNA. It is the one that forms tRNA. It is the one that forms tRNA. And you are going to know what is the tRNA going to do and all. Everything, we're going to study it together. Don't be worried. Okay? So we have the rRNA, we have the hnRNA, and now we have the tRNA. Apart from that, rRNA says that actually here all of them are ending with 8. I have one that ends with nothing. Can you please use that? Please, can you make that? So there is an rRNA. Okay, there is an rRNA which is going to be 5S rRNA. Okay, 5S rRNA. It is not ending with 8, so it is odd one out. Okay, and number if we see 28, 18, 5.8, less. Okay, here it is 5S RNA. Apart from this, there is small nuclear RNA also, snRNA. There is small nuclear RNA that is known as snRNA. So in total, how many polymerases do we have? We have 3 of them: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1 helps in the formation of rRNA, 28S, 18S, and 5.8S. RNA polymerase 2 forms hnRNA. This is a precursor of mRNA. This is what? A precursor of the mRNA. And finally, we have RNA polymerase 3. This forms tRNA. Okay, this is going to form tRNA, rRNA, which is 5S, and also SN, small nuclear RNA. These are the few things which are there, which you need to remember. Okay, which we need to remember. These are found in where? Eukaryotes. So what did we study about transcription? We studied about transcription, that is basically, ma'am, this is the unit, very important structure. You have to draw and draw and draw. Okay, then we studied about what? Ma'am, we studied about how the initiation sigma factor and the rho factor comes. Sigma is for initiation, rho is for termination. Okay, always remember it. So many questions have come from here. Now, when we make an mRNA, it is always going to be U, and basically mRNA stand 5' to 3', 5' to 3'. That is how it works. Okay, the other stand, template stand, is going to be 3' to 5'. Remember, prokaryotes and eukaryotes, here polycystronic, they are monocystronic. Here do not have introns, they have multiple introns. Here basically translation, transcription, everything together. There in separate region. Here only one polymerase, there 2 polymerases, 3 polymerases. What are the polymerases that we have over here? 1, 2, 3. rRNA makes the first one makes the rRNA, 28S, 18S, and 5.8S. Second one, hnRNA, precursor of mRNA. Last one, tRNA. RNA polymerase 3. T. Okay, it makes the 5S rRNA and also small nuclear RNAs. In the case of eukaryotes, you have 3 types of processing also in the RNA: splicing, where you cut down the introns. Second one, capping, where methyl guanosine triphosphates are added at the 5' end, and also tailing, where 200 to 300 adenylyl residues are added at the 3' end. Done. This is how things are going to be done. Cool. Now over here I have a question. KCET 2025 question. RNA polymerase 2 is responsible for the transcription of RNA polymerase 2. Second priority, tRNA, rRNA, hnRNA, snRNA. hnRNA, the precursor of mRNA. The precursor of mRNA. Got it? These type of questions could be coming in. Now in eukaryotes, the entire base sequence of a gene does not appear in mature RNA because in eukaryotes, the entire base sequence of a gene as a DNA does not get to see in mature RNA. Why? Because we are removing the unwanted things. Do we want the unwanted things? No, so that is what we are doing. Some gene sequences are removed by exonuclease. Transcription in eukaryotes consume more energy. Coding sequences are removed during processing. Why do you use coding sequence? Why do you remove? Introns are removed during processing. Introns, basically, the unwanted things are going to be removed during the process of processing. Okay. Finally, what do we have? We have a question. Read the following statement carefully and choose the correct statement. Read it carefully, okay? This is KCET 2016 question. In a transcription unit, the promoter is located at the 5' end of the coding strand. In a transcription, the promoter is located at the 5' end of the coding strand. Coding strand 5' to dash. Next one 3' to 5'. The other one 3' to 5'. Right. Okay. So this is located at the 5' end of the coding strand. Yes, ma'am. I told you coding strand does not have any function, so we give that function. That he always mention promoter in the terms of coding strand. So this is correct. Next one, the single strand DNA having the polarity 5' to 3' is a template strand. The single strand DNA that has the polarity from 5' to 3' is that the template strand? No, template strand will also always be from 3' to 5'. So this is going to be wrong. You have to find the correct one. So first one is only correct. RNA polymerase binds to operator during transcription. RNA polymerase binds to operator. Operator we do not know as of now. Okay, this is a thing that we need to know, but it does not. Basically, it binds to the promoter. No, it has to bind to the promoter, not the operator. So this is also going to be wrong. Okay, single base DNA difference occurring in humans are called single nucleotide polymorphisms. This is also not something that we know, but this is the correct one. Okay, single base DNA differences. So for example, you and me, I might have GATC, okay, but you might have GGTC. So this okay, this is known as single nucleotide polymorphism. That makes us different. That makes us different? Yes, that is actually something that is true. It is known as SNPs. So anyways, A and D statements are true, A and D. So your answer is going to be D. Your answer is going to be D. We are going to talk about all of it in the upcoming part. Okay, so now transcription is also done. Now what do we have? We have the next concept, that is genetic code. That is 8 out of 13. We have only a few more concepts which is left behind. Okay, who decided which codon means which amino acid? That means who decided that this part of gene, when it have this particular thing, it codes for this particular protein or amino acid or things like that. You said it forms proteins, right? Who decides it? So we are gonna talk about that right over here. Cool? Works? Yes, ma'am. Perfectly. It works perfect for me. And just give me a break. I'll be back right after the break. So, okay, now let's go ahead and talk about what happens after transcription. So, I think you might not understand this question right now, that who decided which codon means what amino acid. So, we are going to go ahead with the next topic, that is the 8th, as I said, out of the 13th one, and that is about the codons. And before I talk about what is codons and things like that, I need to give you a little amount of explanation. And it goes something like this. Okay, so what do we have over here? This is going to be our DNA. This is what? This is our DNA. Our DNA, we know, will be forming an mRNA sometime or the other, and then it is going to form the proteins. Now, how does it make the protein? How does it make the protein? So basically, when I say that we have nucleotides over here, and those nucleotides get a complementary nucleotides over here, suppose I have AUG, okay, UUU, and so on and so forth. There are so many nucleotides that I have over here. How do they help us in the formation of proteins? That is a major question. How do they help us in the formation of protein? So listen to me. Each nucleotide that we have over here, they are going to come up and make a group of 3. They are going to come up and make a group of 3. You call this as triplet codons. You call them as triplet codons. These codons which are there, Baba, each codon is very unique. They are going to code for different types of amino acids. So this is going to code for an amino acid, this one is going to code for another amino acid, and so on and so forth. Now, why is amino acid over here? What's the importance of it? Arey Baba, whenever you have amino acids over here, they are the ones that are going to combine together and are going to help us in the formation of protein. All the amino acids, when they combine together, what do you get? You get the proteins. So your mRNA, when I say that they have nucleotides, always understand these nucleotides are going to come in a group of 3, and all the 3 of them, each of them are going to code for something or the other. Either they code for amino acid, or they might not code for anything. They might say that stop the process. They might say stop the process. Some might say start the process. So each of the codons which are there have a unique function over here. Okay, got it? And who is going to read this? Who are going to read this codons? That we're going to talk about in the process of translation. Before that, these codons which are there, I'll call them as genetic code. I call them as genetic code, and we are going to talk about them first. So given below are the sequences of nucleotides in a particular mRNA and amino acids coded by it. Write the properties of genetic code that can be correlated from the given data. This is actually a question that has come in your second PUC. They had given a question like this where they had said AUG is coding for MET. What is MET? Methionine. UUU is coding for phenylalanine. Again, you have UUC coding for phenylalanine. You have GAG coding for glutamine. You have UUA coding for leucine. You have GUG coding for valine, and UAA coding for nothing. Basically, it's saying stop. Okay, this is a scenario that is given to you, and you are being asked, what are the features of genetic code that you can understand from this particular example? Can I have a quick guess? Let me have a quick guess, at least the easiest one. I can clearly say this codon which is there does not code for anything, rather it gives the signal that you have to stop the process of translation. Well, yes, this is known as stop codon. So in the genetic code, you have few stop codons which terminates the process of translation, okay, which stop the process of translation. This is going to be UA, UGA, and UAG. Okay, UA, UGA and UAG. These three of them are going to be what? Stop codons. Okay, got it. I have an understanding of that. Next feature that I can understand is basically, ma'am, as you said, all of them are made up of 3 nucleotides. 3 nucleotides are coming together to form a codon. And each codon is coding for an amino acid. That is something that I can clearly see. Okay. Now the next thing that I can see over here is that UUU is coded by coding phenylalanine. UUC is also coding phenylalanine. Both of them are coding phenylalanine. There is a feature which says that sometimes a codon will be coding for more than an amino acid can be coded by, an amino acid can be coded by more than one codon. What do you call this feature as? You call this feature as degenerate feature. Okay, what do you call this feature as? You call this as degenerate feature, where multiple codons can code for the same amino acid. Can one codon code for any other amino acid? No. One codon codes for only one amino acid only. Multiple codons can code for one amino acid. That is different. Okay? Okay. Cool. So can you see that GAG will code for another anything else? No. Will you see UUU codes for something else? No. It will always code for phenylalanine only. But this can also code for phenylalanine. That is why I said one amino acid can be coded by multiple codons. This feature is known as what? Degenerate feature. This is also something that I can study from here. Okay. Then what? Then what is something that I can see? I can see that all of them, they are read in a continuous fashion. There is no full stop, comma and anything like that. Do you have any full stop and comma and things like that? No. So they are going to be read in a contiguous fashion. Contiguous fashion, as in basically without any full stop, comma and things like that. Okay. Yes, so what are the features that I got to understand right now is basically all of them, they come in a number of 3, and that's known as codon. A codon is going to code for only one amino acid. A codon, only one amino acid. Yes, then I saw that basically they are read in a continuous manner. Then I saw that an amino acid can be coded by, an amino acid can be coded by multiple codons. Yes, that is known as degenerate feature. Then I know that stop codons are there, which do not code for anything. That helps in termination process. UA, UGA and UAG. UA, UGA and UAG. Right. This is clear? Yes, ma'am. This is super clear to me. But is that all? No. You have few other features as well that we have to study. First is triplet. We studied the codon is triplet. 61. Okay, this we are going to come to that. Okay, 61 codons are there. Blah blah blah. We'll come to that. We'll talk about that. Degenerate. Some amino acids are coded by more than one codon. Correct. Commaless. Codons are read on mRNA in a contiguous fashion. No punctuations are there. Full stop, comma. Nothing is there. Okay. Nearly universal. This we didn't study. Nearly universal means, for example, UUU in my body, UUU in your body, every body it is going to code for phenylalanine only. UUU in the terms of bacteria also it's going to code for phenylalanine only. That is what you mean by nearly it's universal. Now nearly universal is mentioned that some exceptions could be there. Some places, some exceptions could be there. So it is clearly mentioned. From bacteria to humans, UUU is coding for phenylalanine. Exceptions, mitochondrial codons and some protozoans. So there are a group of organisms that are known as protozoans. Okay, Plasmodium and all that causes malaria comes under this. Anyway, so protozoans as well as mitochondrial codons could be an exception. That in that case you might not see the universality out there, but yes, remember protozoans and the mitochondrial codons. Here, rest everywhere it is going to be nearly universal. Next thing, we have studied stop codons, UA, UGA and UAG. These are stop codons, they are terminator codons as well. Okay, now listen to me. There is something that is known as start codon. Start codon. And that start codon is only one, AUG. And AUG which is there, it has a dual role. That means it has 2 roles. If at all you have seen this particular picture also, you might be able to see AUG is coding for an amino acid which is known as methionine. It is known as methionine, and this methionine is what? A start codon. it is going to start the process of translation as well. Okay, so methionine is there, it is coding for methionine, it is an amino acid. AUG is there, it is coding for methionine, an amino acid. Apart from it, it gives the signal that here you have to start. Mao was there, he said that, Baba, we have 20 amino acids. How many amino acids are there? 20 amino acids. Okay, how many bases are there that we have? We have 1, 2, 3, 4. In the case of mRNA, we have 4 of them, right? In the case of RNA, we have 4 base pairs. In the case of DNA, also 4 base pairs. Okay. Now, we want to make sure that they code for amino acids. They code for amino acids. So, 4 of them, if at all, they come in the pair of 2. How many codons will you get? 16 codons. Arey Baba, if at all you have 16 codons, will they be able to code for 20 amino acids? No. One codon might have to code for 2 amino acids. That cannot be possible. That cannot be possible. So he said that, okay, then this is not possible. Let's calculate this. What happens when we make the 4 of them group into 3, 3, 3. Then what happens? If we do that, 4 into 4, 16, 16 into 4, 64 codons will be there. So here extra codons are there. But is that a problem? No, it's okay. One amino acid can be coded by multiple codons. One amino acid can be coded by multiple codons, that is fine. But one codon coding for multiple amino acid, that is not fine. How does the body know whether I have to code for this amino acid or this? You get it? So we have got 64 codons because of this particular reason. Now in those 64 codons, you should know 61 of them are going to code for amino acid, 3 of them are not going to. So 3 of them which are not going to, they are known as stop codons. And rest of them which are going to code, they are going to be known as what? They are going to be known as normal codons. That's all. Got it? So the properties of genetic code, they are going to be triplet. 61 codons are there coding for amino acids. If you're asked how many codons code for amino acids, 61. Only 61. Don't write 64. Because 3 of them are going to be stop codons. Okay. Then there's a feature of degeneracy where some amino acids are coded by multiple codons. Okay, multiple codons. Now, commaless, the codons have been read like that. Then we have nearly universal. Almost all the places the codons are going to code for the same amino acid itself. Exceptions are there, mitochondrial codons and also protozoans. Okay, AUG is going to be what? Having a dual nature. It's a start codon as well as it codes for an amino acid that is known as methionine. Then we have a stop codon, 3 of them actually, UA, UGA and UAC. Done, done. This is it. Okay, now listen, there are a few more names given in the NCERT, the scientist name, about which we just have to have a clarity. There is Severo Ochoa, there is Hargobind Khorana, and there is Marshall Nirenberg. Okay, first of all, George Gamow was the person who came up with this 4:3. Okay, that means 3 triplets are going to come. George Gamow was a person, he was a physicist. Okay, Severo Ochoa, what did he do? Severo Ochoa actually came up with an enzyme which is known as polynucleotide phosphorylase. Okay, polynucleotide phosphorylase. It's an enzyme that synthesized RNA without reading the DNA. Okay, without having a DNA, you can actually make an RNA. You can actually make an RNA with the help of this particular enzyme. With the help of this particular enzyme, this enzyme, polynucleotide phosphorylase, was basically discovered by Severo Ochoa. He just discovered the enzyme with which you can make an RNA without the help of DNA. So that means no transcription is needed. Okay. Hargobind Khorana came into the picture. He knew about this particular thing. He actually used that and made up an RNA sequence. He made up an RNA sequence. Now when I make up an RNA sequence, I know what all nucleotides did I use. I used AUG, then UUU, and what all I used, I know it. I know it. I synthesized it. This was done by what? Hargobind Khorana. Now, why are we doing this? Why do you think we are doing this? Baba, if at all I know the nucleotides, and then I make translation to happen. I let the translation to happen. Basically, when translation happens, they are going to code for amino acid. When they code for amino acid, will I not able to understand what is this coding for? Because I already know what is this made up of. AUG is the codon. Now when I say the amino acid, I know that this codon codes for this amino acid. You get it? I already have a reference. If I didn't know, then I have to basically sit and find a way to find the nucleotides are there. But I only made it. I know. I just want to see what does it make. If I know what does it make, I can assign that role to it. You get it? So Hargobind Khorana chemically synthesized the RNA sequence. Now lastly, Marshall Nirenberg came. He developed a cell-free system outside living cells to experimentally confirm which codon codes for which amino acid. So basically he made a cell-free system where translation could be taken place without, like, you do not need to go within the cell for translation, everything. You could do it by yourself, okay, such that you get to understand that how translation happens, what type of amino acids are going to be formed. And basically if I know the amino acids, I know that who are the ones coding for it also, so that I can give a designation for all. AUG codes for this. That is how I came to conclusion that AUG is a one that has dual function, or UAA is the one that is a stop codon. You get it? So Severo Ochoa is the one who gave the enzyme that is known as polynucleotide phosphorylase. Okay, next one is Hargobind Khorana, who synthesized the RNA sequences by his own. And finally you have Marshall Nirenberg, who created a cell-free system such that basically we could understand what codons code for what amino acid. Done. That is a basic thing that we need to have in your head. That's all. In your NCERT, it's too complicated. I do not know why they have made it so complicated. Now, when you say 64 codons are there, it is going to look like this. It is going to look like this. Okay? You are going to have a formula, UCAG, UCAG, okay? UCAG, UCAG, you can arrest the gangster. You can arrest the gangster. Put it over here also. You can arrest the gangster. So UCAG there, UCAG over here. UCAG there, UCAG over here. Now what do? Listen, here also you're going to put UCAG together. UCAG, UCAG, UCAG. This, UU, and starting one U. UU, starting one C. UU, then A, UUG. That is how we are going to put all of it. We are going to put all of it. CU and then this. So all the UUU ones are going to come over here in middle. CCC ones are going to come over here. AAA ones are going to come over here. G ones in the middle are going to come over here. Okay, in the middle you're going to get U here. In the middle you're going to get C in the second row. In the middle you're going to get A in the third row. In the middle you're going to get G in the fourth row. Got it? Got it. You can arrest gangster, you can remember like that. Now listen to me. They can ask you questions, okay, because they have done that previously. So they can do that again, but they can ask you, what does this particular thing code for? General thing that you should know is basically AUG for, as I said, methionine, and it's a start codon. UAA, UGA, UAG, stop codon. Apart from that, there are only two codons which are there. Okay, sorry, two amino acids which are there that just has one codon for them. Two amino acids are there that just has one codon for them. One is going to be methionine, having what? Having AUG, and the other one which is there is going to be tryptophan. Tryptophan having UGG. Having UGG. So write it somewhere. If at all you feel that you might forget it, write it somewhere. AUG is there. Okay, AUG is there that codes for methionine, this is the one, and UGG is there that codes for tryptophan. Got it? Very, very, very important. Now listen, over here, what do you have? Fill leaf in my wallet. If at all you want to remember it, you can use like this. Fill as in phenylalanine, okay? Leaf as a leucine, in my wallet. Wallet goes off. So this is the only way you can remember. Otherwise you can just use your time. Like, you need to invest some time for this particular thing if at all some question comes. In your second PUC does not. In KCET it could. Okay, so this is just a trick I'm saying. Okay, I will not be teaching you that right now because it might take a lot of time. So fill leaf in my wallet. So the first row I have written, fill leaf in my wallet. That is how you have to go. Okay. Serious professor through alarm. Serious, serine. Professor, proline. Through, threonine. Alanine. Alarm as in alanine. Serious professor through alarm. This is the easiest one, having C in between. Okay, serious professor through alarm. Then what do we have? Okay, tyrosine, stop his gun. Tyrosine, stop his gun. So this is tyrosine, this is stop codons, histamine, and finally glutamine. Okay, histamine and glutamine. So please remember it as well. Remember this as well. Okay, then Asian like aspartic glue. Asian like aspartic glue. 2, 2, 2, 2. All of them are going to be 2, 2, 2, 2, 2. Asian like aspartic glue. So aspargine, lysine, aspartate, glutamic acid or glutamate. glutamic acid. Okay, aspartic acid, glutamic acid, asparagine, and over here lysine. So these few things are there. If at all you can use this particular slide and try to learn, it will be really good. If at all you get a question related to this. I'll show you a question related to this. As I said, I will not invest too much time into it, otherwise we might not be able to complete the chapter ever. Okay, so let's go ahead and we'll see if at all it's needed. We'll come back to it. Okay, the codons UUU and UUC code for phenylalanine only. This feature of genetic code is called UUU, UUC. Both of them are coding for phenylalanine. This feature is known as what? 2 codons for 1 amino acid. Degenerate feature. It is not comma-less feature. Okay, it is not going to be non-overlapping feature. It is not going to be non-ambiguous. This is going to be what? It is going to be degenerate. It is going to be degenerate. This is going to be your answer for the question. Okay, this is going to be, I think after teaching for so long, even my, screen has went off. Okay, so this is going to be degenerate feature. Next question. Okay, this is a thing that might require the knowledge up there. Find, this is KCET 2018 question, by the way. The previous one was KCET 2016. Find the nucleotide sequence of mRNA which codes for the sequence of amino acids methionine, leucine, valine, arginine, and alanine. So over here, alanine means basically that comes at the end. Serious professor threw alarm in the second row. Remember, alanine comes at the end. So it has to, in the middle term, it will be having C. In the middle term, it is going to have C. Over here, I see 2 options with CC. This one. GCC, TCC, both of them are going to be C. Now methionine, you know, it is going to be AUG is there. Let me cut the other options. Okay, now leucine, fill leaf in my wallet. The first one, leucine. Okay, in that the middle one was what? U. In that the middle one should be U. So here if at all I check U, okay, this one is U, so I see this. This is going to be the answer. This is going to be the answer, yes. So my dear children, here, that's what I said. Use your time, okay, spend a little bit of time over here to just look at the middle one and just try to understand phenylalanine, leucine. Just have that in your head once. For 10 minutes if you spend your time in this particular thing, I have given you the formulas, okay, the tricks that is there. If you can, it will be the best thing that you can do for yourself. Cool. Now, rest of the questions are not like that, which is why I said I will not be investing too much time into it, because I don't think that many questions could be coming from here, right? Now, which of the following amino acids is coded by single codon? I told you there are only 2, methionine and tryptophan. Methionine is there? No. Tryptophan is there, so we will go with tryptophan. Cool? Only 1 codon. Next thing, done. That is done. So that's all about genetic code as well. In genetic code, we studied about its feature. You might be asked to write about the features of it. Otherwise, you might be getting a question like that where they will give you a scenario and they will ask you what are the features that you can visibly see. Don't check for the things that you can visibly see. Just think of what all processes you know and just write it off. That saves a lot of your time. Okay, now ma'am, what happens if one single letter changes? What do you think what happens? Mutation happens. This is going to be concept 9 out of 13. We are gonna almost finish this up. Mutation happens. What do you mean by mutation? Mutation is basically change in the sequence of DNA that is going to have an effect on the genetic nature and also your physical appearance, phenotype. The character is going to be changed. This we have studied where? In the previous chapter, that is principles of inheritance and variation. Now over here they have mentioned more about frame shift mutation. The same thing as I told you before. Okay, this is going to code for 1 amino acid, this is going to code for amino acid, this is going to code for 1 amino acid, this is going to code for another amino acid. Suppose I am inserting a nucleotide over here. Supposing I am inserting a nucleotide over here. So over here what they have done is they have inserted a nucleotide at this particular location, B nucleotide. What will happen? Ram has, it stays the same. Ram has red cap. Oh, Ram was really happy. Now Ram has, until that particular point it remains the same, but B comes in between, so it becomes Bree. The whole thing got changed, you get it? The frame got changed. Now will this produce the same amino acid? No Baba, it is going to produce a different amino acid. The same thing can happen if at all I remove the R. If at all I remove the R, what becomes? It becomes EDC. EDC, again a different, because different codon is there. A different codon means different amino acid. Sometimes it can lead to a stop codon also. It can make up a stop codon. For example, okay, for example, over here you had UUU, okay, and here you had AAG. Here you had AAG. What happens is that this is coding for phenylalanine, this is coding for something else out there, right? Yes. Now I have inserted two nucleotides over here. Suppose G and G I have inserted. What happens then? In that particular case, it will be GGU, and this becomes UUA. Ooh, sorry, not U. Just a second. Yeah. Suppose it was like this. I inserted this. What happens then? What happens then? Okay, it becomes GGU. GGU comes together. UAA comes together. The process stops right there. The process stops right there before even forming the full thing. Anything can happen like this. For example, it can be because of insertion, it can be because of deletion as well. This is known as frame shift. You're shifting the frames, and that is why mutation is happening. Sorry, that is why problem is occurring. This is known as frame shift mutation, where insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. This is known as frame shift mutation, or you also call this as insertion or deletion mutation. Okay, always remember. And mostly it's because when one or two is being inserted or deleted. If 3 has been put, basically over here, I am putting 3 of them. TWO, then it becomes what? Ram has 2 red cap. Ram has 2 red cap. Now this 2 thing can also code for something else, can also code for stop codon. In that case, Ram has until then it will get stopped. Or probably it can code for another amino acid. You get it? But at least they will code for something. But here in this particular case, it's even more worse. It's even more worse. So frame shift mutation, in any case, it is going to be worse. But if you have a comparison, then it will be insertion of either 1 or probably 2, or probably deletion of 1 or 2. That causes more trouble. That is going to cause more trouble. Got it? Yes, ma'am. That is also clear. Okay. If that is clear, then let's talk about the next thing which is there. So mutation is clear. We studied about the genetic code also. Baba, the next thing that we have, I think a little less energy is there right now, right? So we have to increase our energy. Come on. So we had done everything now. We know everything. DNA, RNA, we have formed transcription, we have done the transcription, we have done everything. Now we just want to make the proteins. How does the mRNA actually become a protein? This process is going to be known as what? Translation. This is going to be concept 10 out of 13. Very, very close to the end. Now listen to me. Before I go ahead with anything, you have to be carefully listening to me. This is the cytoplasm that we have. This whole thing is what? The cytoplasm that I have over here. In the cytoplasm, you had got what? The mRNA. You have got what? The mRNA. Got it? Yes. Yes, ma'am, got it. The mRNA is in which direction? 5' direction to 3' direction, because that is how I made it. The first codon is going to be what? AUG, ma'am. The last codon which is going to be there is going to be what? UAA or UGA or UAG. UGA, UAG, and basically all of it, right? Either of it. Got it? Got it. Now listen to me carefully. When this mRNA comes into the cytoplasm, it will be like, oh my God, nice place. It looks so great. And then it meets its first friend out there. First friend. Who is the first friend here? The first friend that you have over here is going to be a tRNA, which has a 5' end over here, a 3' end over here. This is a new world. This RNA is this RNA is what? A different RNA. What do you call this RNA as? This is going to be known as a tRNA, transfer RNA. What do you call this? Transfer RNA. Remember, RNA polymerase 3 makes tRNA? Yes. So what is it transferring and what is happening over here? So listen to me carefully. In the tRNA, which looks like this over here, this loop which is there is known as anticodon loop. Anticodon loop. Anticodona. If this is codon, this will be anticodon. That means that here if it is AUG, then here it is going to be complementary of it. U A, C. Wow! So it comes and reads it, and that's how it comes and binds? Exactly. And what's the point of binding? Baba, one more thing is going to be happening over here. The 3' end is going to be attached to what? An amino acid. The 3' end over here is going to be attached to an amino acid. So basically, I'll call this end as amino acid acceptor end. Amino acid acceptor end. So there's amino acid over here, and here you have the anticodon loop, which comes and binds over here, the anticodon loop. Okay, where is the amino acid acceptor? 3' end, not at the 5'. Here the 5' is free. Here the 5' is free. Every time the 5' is generally free. Okay, here it is going to be 3' end where the amino acid is coming and attaching. Now, guess what? Which amino acid will be there? If this is AUG, AUG codes for methionine. So, Baba, it will be methionine that is going to be there. So, ma'am, that is how it is going to code and make the protein exactly. But now listen to me. Okay, before all of this happens, your tRNA, okay, your tRNA is right over here. Suppose with the codon UAC, anticodon UAC. This is the 5' end and this is the 3' end. Until now, it didn't have any amino acid. There's a process that is going to take place, which is known as charging of tRNA. Charging of tRNA, or you also call this process as amino acylation of tRNA. Amino acylation means you're adding the amino acid, that's all. Charging of tRNA means you're adding the amino acid, that's all. Okay, and this process requires a lot of energy requiring process. This is known as charging of tRNA. So all of them are ready, super ready to meet the mRNA. So charging of tRNA happens first. You have to keep that in your head. Okay, first process that is going to happen is this. Second thing is that it comes and binds on the mRNA. This particular tRNA, charged amino acyl tRNA, is going to come and bind. Okay, okay. Now, ma'am, I know one thing that you told that is coming out because the machine is there to form the protein, and that machine that we have is ribosome, an organelle. In the ribosome, always remember, the ribosome which is there, it has two subunits, one larger subunit and one smaller subunit. Larger subunit and smaller subunit. In between that, you would have the mRNA. So as of now, I made something like this. I have made something like this. Now I'll make a better picture over here. Not a better picture, basically, just to have your understanding. Over here, you have the 5' end. Over here, you have the 3' end. Suppose you have AUG over here and you have UAA over here. You have got the tRNA that is coming and binding with an amino acid that you have, and here the 5' end. Here you have UAC. Right, right. Now what happens after this comes and bind? The smaller subunit is the first thing that is going to come and bind over here. Smaller subunit comes and binds to the RNA. This is going to be what? The third process. Third process, smaller subunit binding. Okay, okay. Now just give me a second. A little bit of confusion over here. Okay, just a second. Little change, a little change over here. Okay, so over here what happens, suppose this is your mRNA. This is your mRNA, and this mRNA is in which direction? 5' to 3' direction, right? Right. Over here, the first codon that you're going to have is going to be AUG, and the last codon can be anything, UAA, UGA, UAG, everything, UGA, UAG, and everything. Okay, there are stop codons. Now listen, it's for the first time mRNA is going to come in the world of cytoplasm. It is very excited. And you know what happens there?
it meets its friend there, long gone friend. And this friend, Baba, looks something like this. There is a 5' end and there is a 3' end in this particular friend, and this is known as what? tRNA. If at all you try to remember, okay, RNA polymerase 3 transcribes tRNA. This is that tRNA that is there. And in this tRNA, Baba, you are going to have a loop which is known as anticodon loop. It's known as anticodon loop. What do you mean by anticodon? If this is the codon, this is the anticodon. Opposite side of it, A is going to have U is going to have A, and G is going to have C. This is what I mean. So, ma'am, you are saying, depending on the codon, if at all it matches, it comes and binds over here. Exactly. And not just that. Here at the 3' end, you are going to have one amino acid bound to it. At the 3' end, you would be having one amino acid bound to it, which is why you call this as amino acid acceptor end. You call this as what? Amino acid acceptor end. And always remember one very, very, very important thing. Okay, and that is basically if you have AUG here, which amino acid will be there? AUG codes for methionine. So this is going to be methionine. So what have we studied? We have studied that it meets its long grown friend, that is tRNA. In tRNA we have 5' end and 3' end. Okay, and apart from it there is anticodon loop which binds with the codon on the mRNA, and the next thing is that you have amino acid acceptor end at the 3' end. Got it? Got it. Now listen, before all of this happens, there are so many other things that is going to happen. The first thing that is going to happen is your tRNA was never like this. Your tRNA was having 5' end. Okay, it was having anticodon loop, that and all agreed. Okay, it was having 3' end, but it didn't have amino acid. It prepared for it. It prepared for it. It went and took an amino acid to bind with it. Well, this process is known as charging of tRNA. It's known as charging of tRNA, or sometimes you also call this as amino acylation of tRNA. So either charging of tRNA or amyloacylation of tRNA happens, and please do remember this requires a lot of energy. It's a highly energy taking process. Okay, so before any of the translation process happen, this is super ready. tRNA is super ready with its amino acid, and finally it is going to meet its mRNA also, but you have to wait for some time. So charging happens. This is the first process. Second process, if you remember, I told you, why does it come out of the nucleus? Because in the nucleus there is a machinery which is going to help in the production of protein. What is that machine? It's actually ribosome. It's actually a ribosome. And in ribosome, Baba, you have a larger subunit and a smaller subunit, if you remember, right? So over here, if this is the mRNA with the 5' end and the 3' end, your smaller subunit is going to come and bind over here. Your smaller subunit will come and bind over here. This is going to be the next step. Smaller subunit comes and attaches to the, what do you say, RNA. This is going to be mRNA. That is going to be what? The next step. After which, what is going to happen? After which, your RNA is going to come and attach. Your RNA is going to come and attach. This is going to be the third thing. And along with the RNA, what do you have? You have the larger subunit also coming and attaching. The larger subunit also comes and attach. And now finally, you have made a complex which is known as initiation complex. We can initiate the process. Okay, it is known as initiation complex that we have made up over here, which will go ahead and do the entire process. How will it do that? If you remember, I told you, ribosome which is there, it is made up of rRNA, and apart from that, it is made up of a lot of proteins. This RNA is what, if you remember, a catalyst. It's a ribozyme. It has enzymatic action. That enzymatic action is going to work around here. Suppose I have an amino acid being formed over here. Another tRNA comes with another amino acid. What is going to happen? It is going to read like this. You remember, you just understand. If this is your RNA, your ribosome is going to move from 5' to 3' direction. And while it moves, suppose here you have an
amino acid. What happens? All of this goes away. The amino acid combines with the other amino acid which is there. Similarly, it moves. Again, here also you have amino acid. Here also you have tRNA. This combines with this. This tRNA goes away. So what you are getting? You are getting a chain of amino acid. And who is helping in formation of in this chain? The enzymes which are over here. The enzymes and the proteins which are over here in the ribosomes are going to do that particular process. And finally, it is going to do until we are going to reach the terminator codon. Until the point we reach the terminator codon, this process is going to go on. Go on. That is something that came into my head. Okay, so if at all you have to just have a quick thing. Okay, if you are asked to explain the steps, first is charging of tRNA, where the amino acid comes and binds, then initiation, elongation, termination. But I do not know about all of it, so I have to read it. So let's read it over here. The first thing, activation or charging of amino acid. Amino acylation of tRNA, where attaching of amino acids to its corresponding tRNA occurs. This is the first thing that had took place in the first phase. Second, initiation. From here the things are going to go ahead. mRNA binds to the small ribosome and submitted. I told you. Start codon AUG is recognized by initiated tRNA carrying methionine. The first tRNA is known as initiated tRNA. So it is recognized by that. Okay. The larger subunit assembles. Now you have what? Initiation complex super ready. Okay. Now what happens? tRNA brings amino acid and codons match anticodons. Peptide bonds form. What do you mean by peptide bonds? A bond between one amino acid and the other amino acid is known as what? Peptide bond. It is going to be known as peptide bond. So over here peptide bonds are formed. Peptide bond form catalyzed by peptidyl transferase. Peptidyl transferase, the ribosome itself is the catalyst. Okay. Peptidyl transferase is going to help with this process. It's an enzyme. Please do remember it. The ribosome itself is a catalyst as well. By that I mean it is made up of RNA and things like that. Okay. So for example, in the case of bacteria, there is 23S. 23S rRNA. This is an enzyme. This acts like an enzyme. This is an RNA molecule, but this acts like an enzyme. In the case of bacteria, you call this as a ribozyme. Okay. So over here, the peptide bonds are going to be made with the help of what? With the help of units of ribosome itself. What is there in the ribosome? rRNA is there, which is going to act like a catalyst. Especially in the case of bacteria, you have been named one, 23S. Okay, 23S rRNA. 23S rRNA is going to do the function. Ribosome moves along mRNA from 5' to 3'. Everything. RNA polymerase, DNA polymerase, everything moves from 5' to 3'. This also moves like that. Your ribosome is going to move. Finally, at the stop codon, release factors bind and free the new polypeptide. At the stop codon, when you reach the stop codon, there is no tRNA to come and bind. Always remember, there is no tRNA to come and bind on the release codons. Okay, it's just there. The release code, on the terminator codons, there is nothing. There's a release factors that will come and bind, and the whole process is going to be stopped. Got it? So we have activation, then we have initiation, then we have elongation, and finally we have termination, where your stop codons are going to be combined with what? Release factors, and the whole process is going to be done and dusted. Cool. Works. This is how it goes, and there are a few things that is given that is very important for you to know. And where is it? Here. Listen, this is a tRNA that we have. tRNA is also known as soluble RNA or sRNA. Okay, it is also known as soluble RNA or sRNA. Now it is known as transfer RNA because it is transferring the amino acid, but initially it was known as soluble RNA or sRNA. Okay, amino acid acceptor end, the 3' end where the specific amino acid attaches, that is known as amino acid acceptor end. Then you have an anticodon loop. It carries the anticodon that base pairs with the mRNA codon. So we have two ends are there. Soluble RNA, it is known as soluble RNA. One more very important thing, the secondary structure of tRNA looks like clover leaf. It looks like a clover leaf, but the actual structure is a compact molecule shaped like an inverted L. So if at all you see, a L looks like this, but this looks like inverted L. It looks like an inverted L. Okay, so secondary structure of tRNA looks like a clover leaf. Okay, but the actual structure is actually more compact. It looks like an inverted L. So what is that thing that it looks like inverted L? tRNA. What is the other name for it? Soluble RNA. Okay, two ends are there. Amino acid acceptor end, 3' end. Okay, and then anticodon loop. Done. That's all. Clear? Okay, one more thing, last thing that we have about this, okay. So Baba, In the case of an RNA, mRNA specifically, if you have 5' end over here and 3' end over here, there will be certain region here and here. Okay, since it is before this, you will call this as upstream. This is known as upstream. And since it's below this, you will call this as downstream. So upstream of 5' end, or basically AUG, you have something, and downstream of 3' end, or basically UAA, you have something. Okay, very, very important. Very, very, very important. Cool. So you have AUG and you have UAA, upstream, downstream. What do you have? That is important. Untranslated regions. These are sequences. They are known as untranslated regions because they are not undergoing any sort of translation, but they are still important. Why are they still important? Because, my dear children, they are the ones that are going to make sure that the mRNA is stable, that the initiator codon comes and binds up there only, that the whole process goes on really, really smooth. If they are not there, it is not going to be happening up smoothly. Okay, you get it? So untranslated regions are there. They are not going to get translated. And I'm telling you, they are upstream and they are downstream of 3', upstream of 5' and downstream of 3'. Efficient UTRs at the 5' and the 3' end are required for the translation process to be efficient. They protect and stabilize the mRNA while the translation is going to happen. Okay, they allow smooth initiation and termination of the translation process. So they make it stable. They help in the smooth starting and the ending of the whole process of translation. Is it clear? Very, very important. Cool? Cool, ma'am. Okay. Now, if an mRNA 3' undergoes translation, how many different type of amino acids are present? If something like this undergo translation, how many amino acids are going to be present? Come on, try Muddy. So translation based question. Ma'am, AUG, methionine, UU, phenylalanine, same, and over here, khatam. That's all. So I'll say one is methionine and one is going to be phenylalanine. Two different types of amino acids are going to be formed because this is a stop codon, no? So 2 is the answer. 2 is the answer, 2 type. Oh yes, 2 is the answer. No, 2 is not the answer. Listen to me. Listen, look at the question really carefully. If an mRNA like this undergoes translation, children, mRNA undergoes translation from 5' to 3' direction. From 5' to 3' direction, not from 3 to 5. So you will not read this as AUG or UAA. You are going to see this as AAU 1, UUU 2, UUU same, UUU same, and GUA another thing. GUA another thing. So 1, 2, 3. How many types of amino acids? Basically, you get 3 different types of amino acids. You get 3 different types of amino acids. You get it? You get it? So the answer for this question is going to be 3 types. It cannot be something else. It has to be 3 types. Just give me a second, okay? GUA. Yeah. Got it? It is going to be 3 different type of amino acids that are going to be formed over here. Children do mistakes here. That's why I'm saying look at this thing. This is very, very important. 3 and 5. Okay, the direction is that is actually previous year model question paper question. A mature processed mRNA transcribed from a gene is 636 nucleotides long. A mature RNA transcribed from a gene is what? It is having 636 nucleotides long. Assuming that if it codes continuously without UTR, untranslated regions, okay, and stop codons for a polypeptide, if there is no stop codon and it is continuously going on, how many amino acid does it form? If 636 nucleotides are there, okay, 636 of them, they are going to form a group of 333. So I'll say 212. 212 codons will be there. Each of them are going to code for what? 1 amino acid. So 212 amino acid. Provided they have clearly mentioned there are no stop codons in between, there is no UTR that I have to consider, nothing. All of them are coding. In that case, all of them are, as I said, come in the group of 3. In that case, 212 codons. 212 codons code for 212 amino acid. So your answer for this question is going to be A. It is going to be what? A. Got it? Is it clear? little difficult to probably have all of it together. I really admire you children. You know what? You have been attending this lecture for so long right now. And as a teacher, since I have taught for so much time, I know the concept, so it is becoming easy for me to talk about it. But I really admire that you're still stuck over here, going through the entire thing, putting in your efforts. For you, it's very new. And I know it's difficult also. You're doing a wonderful job, and I pray that you get whatever you deserve. Okay? If you need, take short breaks, have a quick revision, and then come back. That is how we have to go ahead. Otherwise, you might not get it. If you want, you can just pause, look through your notes, or you will get the PDF, by the way, in the WhatsApp channel. Join the WhatsApp channel right now. Okay? So, yes. Cool then. Let's go ahead. Now we have one question. Find the sequence of binding of following amino-acyl tRNA complex during translation. Amino-acyl tRNA complex. This is amino-acyl tRNA complex. This is tRNA basically having an amino acid. Okay. During translation to mRNA transcribed by a DNA segment having the base sequence. Okay. mRNA transcribed via DNA segment. What is the question? I'm coming again. Find the sequence of binding of the following amino-acyl tRNA. So you have to tell me in which manner does this particular tRNAs are going to bind. A, B, C, D is given. You have to tell the order. Okay, order by how? You need to know the DNA. You need to know the mRNA. Do we have the mRNA? Let's check that. During translation to mRNA transcribed by a DNA segment having the base sequence this. So this is the DNA segments that we have. 3' T A C A T G G G T C T T C C G 5'. This is a DNA segment. First of all, check isn't 3' to 5'? Yes. Now I have to make the RNA. When you make a new RNA, that has to be in 5' to 3'. That has to be in what? 5' to 3', right? If you remember, right? So first I had checked this. This is correct. Now I can make the RNA. T base pairs with A base pairs with U, C base pairs with G, A base pairs with what? A base pairs with U, T base pairs with A, and it goes on. And it goes on. This is going to be your mRNA. Now you have gotten your mRNA. Can you try to check out what is the tRNA that is going to be there? Here you have the codon. You just have to look for anticodon. AUG anticodon is going to be what? UAC. This is the first one. B is going to be the first one. B is the first one. I have got my answer. But listen to me. You do not have to do all of it. Why? Because you have got the DNA. This is the DNA. You made a complementary strand of it. That is the mRNA. mRNA's complementary is going to be what? The anticodon. Arey Baba, aren't you making it back again? Just that instead of T, you would be having U. Instead of T, you would be having U, right? That is a basic logic. And here it is going to be this. Yeah. That is how it is going to work, no? Instead of T, you are going to have A. Instead of T, you are going to have U. A remains A, C remains C. Everything remains the same. Just the T replaces by U. You are making a complementary, then again you are making a complementary. That means you're ending up here only. You just have to look at one thing. All the T's over here will be made U, and you have got the sequence. You have already got the sequence of what? The anticodon. So UAC comes first, then comes AUG, then comes AUG, and that is how it goes. AUG, why? Because this is T, again U. You get it? So that is how you can do it. The answer for this question is going to be B. This is a KCET question that had come before. Okay. Now, which of the following sequences of mRNA are required for translation process but are not translated? Are required for the process of translation but are not going to be translated. Stop codons, anticodons, and codons UTR. Which of the following sequence of mRNA are required for translation process but are not translated? And over here you have stop codons, anticodons, sense codons, UTR. UTR, untranslated regions, right? Because they, as I said, they are required for the process but are not going to be translated. That is why you call it UTR, untranslated regions. Cool. Sorry. KCET 2024 question. In tRNA, the region that binds with mRNAs. In tRNA, the region that binds with mRNA. Anticodon loop. Anticodon loop of tRNA, amino acid acceptor end, amino acyl synthetase loop, or ribosomal binding loop. Anticodon loop of tRNA. This is going to be the answer. In tRNA, the region that binds with the mRNA is going to be the anticodon loop of tRNA. As simple as that. Got it? Done. Okay. The mRNA has untranslated regions. Where? At 3' end beyond terminator codon. Correct. At 3' end beyond the terminator codon, it's there. Let's see the next one. At 5' end before AUG. Before AUG, before the start, that is also correct. At both 3' end and 5' end beyond terminator, at 3' end beyond terminator and 5' end before the AUG, correct order also everything, that is also correct. And this is actually sum of both of them, so I'll say this is the one that is more correct. AUG and terminator codon flanks the UTR. AUG and terminator codon flanks the UTR, that is not correct. That is not going to be correct. So flanks the UTR means AUG would be over here. This is the UTR, okay? And this is going to be terminator codon. No, that's not the correct one. The only answer in that case is going to be the C one. Got it? Got it, ma'am. Okay. Now, we have officially completed the translation process as well. And now we are going to go ahead with the next one. Almost we are there. We are ending up the process. Ma'am, do genes ever get switched off? Concept 11 out of 13. Give a big tap and you're back. And my dear children, whoever is watching this video, if you have not hit on the subscribe button yet, make sure you do that, only if you're liking this. Okay, so over here this thing goes, gene regulation and the lack of parone. That means, can you switch off the genes? Example, I do not want to make the proteins. Can you switch off? Okay. Answer is yes. Answer is yes. But that becomes easier in prokaryotes. Okay, that becomes the thing in prokaryotes. Anyways, so in what all ways can I stop the gene expression in eukaryotes? Is there any way that I can stop? Yes, multiple ways. For example, the first way is, over here, the DNA has to form the mRNA. Transcription has to happen. I can stop over here. That is known as transcription level. I will not allow the primary transcript to form, that is hnRNA to form. That is one way. Second thing, this mRNA which is there, before it goes out, it has to undergo some processing, splicing, capping, tailing and all of it. So I can regulate that. I'll make sure that there is regulation of that. This mRNA, that is hnRNA which is being formed, it is not properly converted to mRNA. There is no splicing that has taken place or capping or tailing, some process problem is there. In that way I can stop it, processing level. So this is basically a thing which you do not have to mug up. Why? Because this is something that you have studied. The flow of it you know already. Transcription happens first, then processing happens, Baba, then the transport happens to cytoplasm, and after that in the cytoplasm translation happens. So basically at 4 different levels I can stop it. In the transcription, where I can stop from primary transcript from forming. In the processing level, where I can stop splicing, capping or tailing. And the third one, transport of mRNA, where can I stop basically the movement of the mRNA outside. Okay, I can stop that. And finally, in translational level also, where I can control the protein synthesis. I can control what the protein synthesis. I will not allow translation to happen. At these 4 levels, gene expression can be regulated in the case of eukaryotes. Now, do we have the same type of liberty in the case of prokaryotes? The answer is no. Why? In them, my dear children, processing and all is not there. Processing is not there. Transport of mRNA, it's all happening in the same position itself. So for them, there has to be something different. And that is what we're going to talk about right now. But just know, this is very important in the terms of eukaryotes. How can we control? We have 4 different ways to control it. And I hope you will remember it. Transcriptional, processing, transport and translational. Okay. This is actually a 2-marker question that had come in the model question paper of 2024-25. Okay, cool. Now, ma'am, you were talking about this particular thing. Ma'am, do genes ever get switched off? And you spoke about, you said that it is more happening in the case of prokaryotes. In the eukaryotes, we have something else to control all of it. Yes. So when I talk about prokaryotes and I need a space, where is that particular space? Do I have a space over here to probably make and draw and probably tell? Okay, I'll make it over here. Okay, listen to me very, very carefully. You have something like this. Okay, this is going to be a gene. This is going to be the next set of thing that is going to be there. Here, for this gene, you have a promoter region. Okay, you have a promoter region. And This particular gene which is there, I call this as regulatory gene. What do I call this? Regulatory gene. Why does it have promoter? Because onto this promoter, what will come and bind? Basically, if you remember, that thing comes and bind. What thing? RNA polymerase. You have to make the RNA, no? You have to make the RNA. We're talking about DNA out here. We have to make the RNA and we have to make the protein and everything. Alright, that is the thing that is going to happen. So listen to me carefully. At this particular point, this regulatory gene which is there is going to code for a protein. It's going to regulate in a negative manner. That is why it is going to code for a protein. What do you call this protein as? Repressor protein. Repressor protein. Okay. This is a regulatory gene having its own promoter region. Now, we have another thing out there. This is another promoter thing that we have. Apart from it, there's an operator region, a new thing which is there. And apart from it, it has 3 genes. Structural gene divided into 3 parts, Z, Y, and A. This is the promoter region that we have for these 3 things. Z, which is there, is going to code for a protein as an enzyme. It is known as beta-galactosidase. Beta-galactosidase. Y, which is there, it is going to code for an enzyme which is known as permease. Okay, and A, which is there, it is going to code for an enzyme that is known as trans-acetylase. Okay, beta-galactosidase, permease, and trans-acetylase by Z, Y, and A. This is promoter region. This is the operator region that we have for this. What is operator region? We do not know about it, so we're going to learn about it. Okay, listen. What does the beta-galactosidase usually do? And where is it happening? What are you talking about? So we are talking about E. coli. E. coli is what? A prokaryote. Inside the E. coli, you know, E. coli can survive on, if at all we are providing some glucose medium and all, it can easily survive. Or else probably you give something else that is lactose. It can survive on lactose also. Because lactose can provide it energy. How? Basically, lactose which is there, if at all it is getting lactose, with the help of this particular enzyme, it is going to break the lactose into glucose plus galactose. So we have already got the glucose. Now this glucose can be used for energy. Right? Right. So basically, even if lactose is there, we can still survive. Why? Because we have this enzyme beta-galactosidase. Okay. Okay. Now where is lactose? If this is E. coli that I am going to grow over here, I am going to put the lactose in the medium. It needs to take up this particular lactose. For that, it needs some opening here and there, some pores, right? For that, we have permease. It allows to increase the permeability. To increase what? The permeability. Permeability, the ability, okay, there will be some small openings through which your lactose can easily get in. Now there is trans-acetylase. What does it do? It just stays like this, huh? No, it is the one which is going to deal with every other thing that is going to happen. Any other toxic byproduct which is going to be formed, basically it is going to deal with that. So I'll say trans-acetylase which is there, it is going to deal with byproducts. It can be some toxic byproducts and stuff like that. Now, when do you think I'll require all of them? When do you think I'll require all of them? Only when the lactose is there in the medium, right? Only when I'm putting the E. coli in a medium that is containing lactose. If lactose is not there, do I require all of them? No, it will be waste of my energy. If I am E. coli, it will be waste of my energy. I do not want all of them. So what do I need to do? I need to turn this off. Off. Switch off. How do I switch it off? There's only one way. The one way that we have over here is this regulatory gene which is there, it has to be transcribed and then translated. You should be getting the repressor protein. This repressor protein which is there should and go and bind over here. Ma'am, what's the point if it goes and binds over there? That's a good question. If it goes and binds over there, now we have what? Your RNA polymerase. Your RNA polymerase is over here to do the transcription. But when it sees operator over here, it's like, oh my god, how will I move ahead? Arey, side. I have to. It says that no, No, I cannot move ahead. Basically, I have to switch it off. I cannot allow you to transcribe everything. You please go back. So basically, transcription is being stopped when lactose is not there. Okay. But on the other hand, what if lactose is there? What if lactose is there? Don't we need this particular guys? All these guys? Yes. So what happens is that, suppose this is the E. coli where everything is happening. Lactose is there. Lactose is going to get inside. How lactose is going to get inside, ma'am? Permease is not being formed, use is switched off. It's not like when it's switched off, it's 110% switched off. Still some amount of transcription happens, but very less. So permease will allow basically lactose to get in. Now we have the lactose, and I'll represent lactose in a color that is purple. Is it visible? Okay, it's purple. This lactose comes and says, "Hey, repressor, Macha, what are you doing there? Come here. That long time we have not met. We are best friends." Okay, so what happens is that whenever lactose comes, it is going to actually bind with the repressor protein. Now, if it binds with the repressor protein, can the repressor protein go and bind up there? No. When it is not binding up there, do you think RNA polymerase can do the transcription? Yes. So is it switched off now? No. It turns switch on. It turns into what? Switch on condition. You get it? This is very, very, very important. This is what lac operon. This is known as lac operon that you see in E. coli and other organisms as well. So over here, what is exactly the case? There is promoter region, there's a regulatory gene, you call this as I gene. This can regulate positively, negatively, but over here we're talking about the negative one. So it is going to form the repressor protein. It is going to form the repressor protein. Okay, this repressor protein which is there is going to go and attack over here on the operator region. When it comes and attaches over here in the operator region, what is going to happen? Basically, it will not be able to do the whole process of transcription, right? And these things are not going to be formed. This is what is generally going to happen, right? Yes, got it? This is switched off condition. Now we want that because lactose is there. So what do? We are going to make sure when lactose comes, repressor protein comes and binds over here. There is nothing up there. They will do the transcription and tada, everything is smooth. Beta carotesidase converts the lactose to glucose and galactose. E. coli gets the energy. Permease allows more lactose to get inside, and transacetylase deals with everything in between. Remember ZYA, Zyada. In Zyada you have Zya, you can remember like that. This is going to be what? Your lac operon. And you can read it with the help of this. Just give me a quick second. So over here, what do we have? If at all we check. Okay, arrangement of the genes of the lac operon. This was actually a question also. The arrangement goes like this: promoter, then I gene, that is basically the regulatory gene out there, then the promoter, operator, structural gene ZYA. Cool. And here, I gene is a regulatory gene, produces the repressor protein. P is the promoter site where RNA polymerase binds to the starch transition. O is the site where repressor is going to bind. Structural gene lac Z codes beta carotesidase, breaks lactose into glucose and galactosase. Lac Y, permease, helps lactose enter the cell, and lac A, transacetylase, that helps in detoxification. This is the whole thing that you need to know. There is nothing else which is there. Okay, now there is one more question. By the way, what type of questions do you get? Basically, state the arrangement of different genes of lac operon. This type of questions you can get. So you have to talk about the arrangement. Pyposia, you can remember like that. Describe the role of lactose in the regulation of lac operon. How do you explain that? Basically, in the absence of lactose, operon is off. The repressor protein binds to the operator. This blocks the RNA polymerase. Structural genes are not transcribed. On the other hand, in the presence of lactose, lactose acts as an inducer. It acts as what? Inducer. This is a very important term. It acts as an inducer. Also, always remember, it's not just lactose. It can be allolactose. Okay, it can be allolactose as well. Either lactose or allolactose. So lactose or allolactose acts as an inducer. Okay, it binds the repressor, changing its shape. It can no longer bind to the operator region. It cannot no longer bind to the operator region. RNA polymerase transcribes lac Z, Y, and A. Lactose metabolism enzymes are being produced. So this is going to be happening. If it is not there, none of it is going to be produced. If it's there, it is going to be produced. So role of lactose in regulation. This is how. This is the role. Okay, and arrangement of the genes in the lac operon. So this question is divided into two. That's what is mentioned over here. Arrangement the genes. This is how the arrangement is. And this is basically what it stands for. Okay, and Zya, always remember the flow of it, ZYA. Cool. Cool. In lac operon, concept of gene expression allolactose S. In lac operon concept of gene expression, allolactose acts as what? That is the question. Allolactose. Repressor, inducer, co-repressor, coenzyme. Inducer. It helps. So either lactose or allolactose is going to come up and take the pole position and do this. Okay, either lactose or allolactose. So this is going to act like an inducer. Next thing. Done. Only one question. Okay, only one question. So more of the second PUC questions are there. It usually gives 5-marker question. Very important concept. Okay, in terms of 5-marker, it is going to be very important. So gene regulation we have studied, Baba. In the case of eukaryotes, at 4 different levels it can happen. In the case of prokaryotes, you have been given an example, and that is of lac operon, and we need to know about it. I hope you're clear with it. Okay, lac operon is known as inducer, or basically it is also, yeah, that's all. And or basically it can be allolactose also. Fine. Right. Now we are going to come up with the second last portion of this particular chapter, that is Human Genome Project. So we now know everything about it, how DNA is being made, or how mRNA is being made, how proteins are being made, can the expression be controlled of the gene in any levels, all of it. Now the thing is, ma'am, did we actually read all the 3 billion letters of our DNA? So we have so many nucleotides out there. Did we actually go and read it? How are we able to go ahead with all of it? Is it actually true that we did it? So let's talk about it. Is it actually true or not? So the next topic that we have, concept number 12 after 13, is the Human Genome Project. And what were the aims of Human Genome Project? A very important question. It can be a 3-marker question that can be coming in. Okay, the first aim is to identify approximately 20 to 25,000 genes in the human DNA. Genes, segments, okay, genes. How many? 20 to 25,000 genes. To identify all this 20 to 25,000 genes. Second, to determine the sequence of 3 billion chemical base pairs that make up the human DNA. To determine the sequence of 3 billion. So did we read it? Yes. Okay, we did. So determine the sequence of what? 3 billion. So we are talking about genes. Genes are made up of nucleotides, and we need to understand the sequence of the 3 billion chemical base pairs that make up human DNA. That was the aim. Second aim. Third aim, store this information in the database. Okay, we have to store it somewhere. Fourth thing, improve tools for data analysis. After storing things, we have to improve our tools for analysis of this particular data. Obviously, storage is not enough. You need to use it whenever you require. And for that, it should be easily accessible. Next thing, transfer related technologies to other sectors and industries. Wherever they are doing some research, you need to be able to transfer it. Last thing, address the ethical, legal, and the social issues, which is known as LC. Ethical, legal, and the social issues which is there, all of them have to be addressed. So how many things are there? What are the aims? The first one, identify approximately 20,000 to 25,000 genes. Okay, after identifying them, that's about the genes. We also need to identify that what are the nucleotides that are there in all of this whole genes. That is basically 3 billion nucleotides out there, base pairs out there, right? And apart from this, store this information in the database. Okay, find some tools for improve the tools for better analysis, and finally transfer related technology, transfer related data to different sectors wherever it is being studied, and also address the ethical, legal, and the social issues that could be arising. Right. This is something that is super important. Now, other genomes sequenced. So is it just humans genome which was sequenced in the Human Genome Project? So listen to me carefully. There were some other other organisms, and this was a question. This was a pre-board question, if at all I remember it correctly. So bacteria, yeast, and Cyanobacteria elegans, that is C. elegans, you can remember like this. Drosophila, that is fruit fly, and plants such as rice and Arabidopsis were something that were being sequenced. What all things? Bacteria, yeast, C. elegans, very important. Okay, C. elegans, a free living non-pathogenic nematode as Kelminthus, if at all you know. It looks like worm. Okay, so what all we have? Bacteria, yeast, C. elegans, which is a nematode. And then apart from it, Drosophila, that is fruit fly, Drosophila melanogaster. And apart from it, plants such as rice and Arabidopsis. Okay, these were the things that were already sequenced. This is a very small line of your NCERT, but very important. we cannot tend to just leave it off. So yes, we have to cover it up. Cool? Cool. Now, what are the salient features? So we know the aim. The aim is this. The salient features of our human genome is different. Aim is different. Human genome salient features are different. Please do know it. What are the salient features? The salient features are that we have 3164.7 million base pairs in the human genome. How many? 3164.7 million base pairs in the human genome. Everything almost is around 333 over here. Okay, this many millions of base pairs are there. Base pairs are there. Now, 30,000 total genes are there. So we assumed it to be 80,000 to 1,40,000, but it was 30,000 genes which were there. 30,000 unique genes. 99% of the nucleotides, 99.9% of nucleotide bases are exactly the same in all of us. 99.9% is exactly same in all of us. That means we are just 0.1% different, and that is a huge difference. And that is going to be huge difference. Millions of them are going to be, millions of base pairs are going to be different, but still, okay, but still, less than 2% of genome actually codes for protein. Only less than 2% of genome codes for protein. Rest of it actually is repetitive sequences which are chumma, randomly repeating. Okay, so these are the few points. We are going to go ahead. There are so many salient features. First thing that you have to remember, 3,164.7 million base pairs were found. 30,000 genes were there. Okay, less than 2% of the genome actually codes for protein. Rest of it is going to be repetitive sequences and things like that. And we are going to be 99.9% similar in terms of nucleotides. There's 0.1% difference. Okay. Now, when you talk about an average gene size, we're talking about 30,000 genes, right? So when you talk about one gene, how many nucleotides generally does it have? Average gene size is going to be 3,000. 3,000 base pairs are going to be there in one particular gene. In one particular gene. This is the average gene size. But the largest known human genome is dystrophin. Gene is dystrophin, which is having 2.4 million bases. 2.4 million. This is million, this is million. But here it is 2.4, here it is 3,164.7. Right? So usually one gene has almost generally 3,000 bases, but here in this particular case, the human gene, which is dystrophin, it is going to have 2.4 million bases. Okay, functions are unknown for over 50,000 of the discovered genes. We do not know. Less than 2% is actually coding for proteins, and in that also we do not know functions of 50, more than 50, over 50% of the genes. Cool? Yes. Now, over here, I told you if less than 2% of gene is actually coding for protein, what is the other thing doing? Is basically repetitive sequences. Repetitive sequence make a very large portion of the genome, repeated hundreds to thousands of times. No direct coding function, but they shed light on chromosomal structure, dynamics, and evolution. So that gives an idea about the evolution pattern. If at all an organism before us also had these repetitive sequences, probably that gives an idea about we have evolved probably from them. You get it? So repetitive sequences are there. You need to know it. Now, this is important, this is important, this is important, this is important, and now this is the most important thing that is usually asked in the form of MCQs. Which one was the first? Okay, which one has the most genes? The chromosome, which has the most genes, and the chromosome that has the least number of genes. Chromosome means a compact DNA itself. So which one has the most and which one has the least? Chromosome number 1 was the first to be read, and that has the most number of genes. It is 2968. 2968. Over here there is no 7. If at all you look, there is no 7. Okay, so you can remember like that. 2968. That is chromosome number 1. Chromosome number Y, which is there, it is actually found in the case of men, and there's a that is the shortest chromosome ever. It just has 231 nucleotides. It just has what? 231 base pairs out there. So we have chromosome extremes. Chromosome number 1 has the most genes, 2968, and chromosome number Y has the fewest, that is 231. Very, very, very important. Clear? Clear? 231 and 2968. Now, SNPs. What are SNPs? Single nucleotide polymorphism. That means you and me, as I said, are different only at one particular base pairs. 1.4 million locations with single base DNA differences are there. 1.4 million locations are there between each of us where we can find only a single nucleotide difference. Promise to revolutionize for finding disease-associated sequences and tracing human history. So that could be difficult for you to remember. Just know that there are SNPs, single nucleotide polymorphism, where a single nucleotide is only different between both of us, and that is almost at 1.4 million locations. That many differences are there, but only 111. Okay, so if I have to come together with the salient features, 3164.7 million base pairs are there, 30,000 genes are there, less than 2% of the genome actually codes for protein, and rest of it is repetitive sequences which gives a light about the evolutionary relationship and stuff like that. In this 30,000 genes, if at all we talk about the gene, how many base pairs are there in each gene? The average gene size is going to be 3,000 base pairs, but there is one gene that is known as dystrophin, which has 2.4 million bases out there. That is a unique one out there. Cool. Now, when you talk about the chromosomes, which one has the highest and the least gene? Chromosome number 1 has the highest, 2968. Chromosome number Y has the least, 231. And you have SNPs, single nucleotide polymorphism, where we are different only in one particular position, nucleotide position. And almost 1.4 million locations are there in each of us, where we are going to be different. These are the findings of HGP, that is Human Genome Project. These are the aim that we have of HGP. These were the aims of the HGP, and these were the other organisms that were basically being sequenced. Cool? Cool. And always understand, just know this, okay? They are not going to ask you a lot of things around it. Frederick Sanger's method was used for DNA sequencing. Okay, DNA sequencing annotation method. Frederick Sanger's method was used for DNA sequencing. Frederick Sanger's for DNA sequencing. S for sequencing, S for Sanger. Okay, know about that. There's no other question that probably would be coming from here. Now, the average length of a gene in human is 3000 base, KCET 2017 question. 3000 base, 2.4 million bases, 1500 bases, 500 bases. Average gene, 3000 bases. No? The 2.4 is there in dystrophin. Why are we talking about dystrophin right now? So there's no point. Okay. Next one, according to Human Genome Project, the total number of genes in human genome is estimated at 30,000. The number of genes present on the Y chromosome are. The number of genes present on the Y chromosome. Y chromosome has the least, it has to be 231, right? And the first chromosome is going to be having 2968. Yes, this is KCET 2019 question. Okay. And children, are we ready? Oh my God, I feel to cry. Are we ready to go ahead with the last concept for the day? And that is going to be DNA fingerprinting. So, ma'am, how do police catch criminals with just DNA, ma'am? How do police catch it? So listen, the last thing that we have is known as DNA fingerprinting, concept 13 on 13. Everyone, Baba, who have reached over here, thank you so much, and I love you a lot. Okay, let's go ahead. So, how and why the DNA fingerprinting works? Okay. Human genome is what? Having 3 billion base pairs. When I say 3,164.7 million, I mean 3 billion base pairs are there. 99.9% we are identical, and we all know that only 0.1% difference is there. 0.1% of 3 billion is what? 3 million base pairs. We are going to be different with 3 billion base pairs. Yeah, that's a huge difference. That vary between individuals, making each person very, very unique. This 3 million base pairs difference between you and me makes me me and makes you you. You get it? That's the whole thing that you should know. Okay? And that difference is something that we are going to use in the DNA fingerprinting process. The clever shortcut. Instead of sequencing all 3 billion base pairs, scientists compare specific variable regions. Okay, specific variable regions. Instead of comparing the whole thing out there between you and me, I'm going to compare unique regions that I have and you have in common. These sequences, these regions, repetitive DNA, contain short sequences repeated many times, differently in each person. For example, I say over here, what do we have? We have AUGC. I have AUGC almost 15 times. You have AUGC almost 5 times. Almost 5 times. Okay, this is a difference. Basically, on the basis of this also, I can find who is going to be the criminal. These are known as repetitive sequences, and I said their number varies. We are going to see how and everything, because I know that could be a confusion in your head. Analyzing the differences identifies the individuals very, very quickly. So listen to me. These repetitive sequences, they actually come under a category which you call as satellite DNA. So you have what? If I talk about DNA, they can be divided into two things: bulk DNA and satellite DNA. Bulk DNA is the actual one that codes for our proteins and stuff like that. Satellite DNA, it is the one that is responsible for what? That is responsible for all the repetitive sequences. Okay, bulk DNA codes for protein. Satellite DNA, which is there, it is what? It is small, unique repeats that we have. Repetitive sequences. Okay, now there is mini satellite and microsatellite. Mini basically means according to the size. The smaller one, okay, some repetitive sequence might be small, some would be very, very large. You get it? This is AUGC, this is just 4. Okay, but you can be having a larger sequence that is getting repeated. Similarly, you have that. Okay, so anyways, I just want to tell you that. So DNA is equals to bulk DNA plus satellite DNA. Satellite DNA varies from person to person. It does not code for proteins. This is very important. This is not something that is going to code for any protein, which is why any mutation which is going to be there, it is going to accumulate over generation, and it is not going to hamper any of us. Cool, because it is not good coding for any protein. So what I have said right now is that you and me are different at 0.1% in weight of 3 billion base pairs. So that means 3 million base pair differences is there. Now, we both have repetitive sequences, but in my body it might be repeated 10 times, but in your body it can be repeated 5 times, and with the help of that also, I can find who is basically the criminal or basically the whole process. So how does this thing particularly work? Are we ready to go ahead with it? Let's see. First is isolation of the DNA from the sample. So basically, suppose I need some space. There is a crime. There is a crime that has taken place over here. This is the crime spot. This is the crime spot that is there. From the crime spot, I had got some hair follicles. Okay, I am the police. I am not the police. Okay, you and me are basically victims. Not victims, sorry. What do you say? Suspects. Okay, we are the suspects. We have a police. We have the police. So I am suspect number 1. My name is Jasmine. You are suspect number 2. Your name is Puchu. Okay. Listen to me carefully. Over here, police takes this particular thing, understand their DNA. What are they going to do is, they will make a complementary thing to it. They're going to make a complementary strand to it. Suppose you had something over here. Now here, you're going to have a complementary thing that is ATGC. This is the complementary thing that you have made. Okay, you have made it. This particular thing is known as probe. And I'm going to attach it with some radioactive thing. It becomes a probe. I got it. I got this particular hair follicle. With that, okay, with the help of that, I could find the DNA of the person who probably might have done the crime. I coded it. I have got the complementary strand of it. I made it. Okay, this is going to be known as a probe, and I'm going to attach it. This particular probe is known as a probe when I attach it with some radioactive thing or something like that. Okay, so now it is known as a probe. I have it. Now, both of us were called. Jas and Puchu, both of us were being called, and we were like, did you do the crime? I said, no, I didn't. Probably Puchu might have done it. I, to anyway, will not do any crime. Okay, fine. In that particular case, we need a DNA sample. Okay, I was like, don't take it. But anyway, police, they took it. Okay, they took it. Now, what do they do after taking the DNA sample? After taking the DNA sample, first of all, they're going to isolate the DNA. Our isolation is going to happen from all the different things. Then, the next process that is going to take place over here is we are going to digest the DNA with the help of restriction enzymes. Okay, so we are going to, suppose my DNA is taken. Puchu's DNA is also taken. Okay, Puchu's DNA is also taken. Then we are going to do digestion. Digestion. That means basically cutting off the DNA strands which is there. Post that, we are going to make it run through a electrophoresis chamber. Okay, we are going to make it run through an electrophoresis. Oh, why did I make 2? We just need 1. Okay, we are going to run through an electrophoresis chamber. Now what happens in electrophoresis chamber is that they are going to separate on the basis of their, what do you say, size or length out there. Okay, so the bigger ones will stay over here, will move slowly. The smaller ones will move faster. Okay, so when I separated it, it is going to separate something like this. It is getting separated something like this. For me this, for you this. Okay, first I isolated, then with the help of restriction enzyme, that means with the help of enzymes, I cut it off, and then basically in the electrophoresis chamber, I run it. Okay, on the basis of their size, they are going to move. The smaller ones will be moving faster. Okay, now you have a different pattern, I have a different pattern. Okay, okay, fine. Now what did I do is, I am going to basically do one very, very important thing. Before that, we'll just see this, okay? Isolation, digestion, gel electrophoresis is what you call it, and then you do something that is known as southern blotting. Southern blotting. So here I have to show you the picture. That is why I brought you here. Listen, electrophoresis is done. I take a nylon sheath, okay, and I transfer everything onto this particular sheet, because I cannot find, I will not be able to look at any of it that is going to be happening up over here. So what do? I am going to take a nylon sheet and I'm going to transfer everything which is there. Now everything is there in the nylon sheet. Everything is going to be there in the nylon sheet. After that, after we have it on the nylon sheet, by the way, what do you call this as? What do you call this as? This is known as southern blotting. This is known as southern blotting. Why? Because we want to hybridize it. We are going to hybridize it. What do you mean by hybridizing it? We are going to put this particular props up there. We want them to go and bind to the complementary strands. We want them to go and bind to the complementary strand. That is why we transfer it. In the electrophoresis chamber, we cannot do it. So that is why, what did we do? We did southern blotting. Southern blotting. Okay, okay. Now we are taking this probe, okay, and we are making it run through it. So what happens? It came and bound over here. It came and bound over here. It came and bound over here. For me, it came and bound over here. Okay, got it. Okay, for the other person, it bounded over there. For me, it bounded over here. Now, Baba, this particular DNA that I got from here, ayo, where is it? This particular DNA that I got from here was also cut, was also run through electrophoresis, and basically it looked something like this. This we already have. Now we did this, okay? We are not able to see it yet. The probe is there, probe has went and attached. Now, basically, if you put it under an autoradiography machine, it is going to emit light, and basically we're able to see it. When we see it, what do we see? This and this, same. This and this, same. This and this, same. But here, if at all you look, this and this, not same. What do you understand? That this person did the crime. If at all you had not used the probe, will you be able to see it? No. You will not be able to see. That is why you are using a probe over here. This process after southern blotting, basically where you are letting the probes go and attach, this is known as hybridization. This is known as hybridization. Okay, after hybridization, you are going to use either of the process. Autoradiography you can do, and anything that you can do. Basically, under fluorescent light also you can use, basically to view how it appears. After that, you just have to match. And that's how the person would be caught. And that's how the person is going to be caught. So over here, what do we have? Isolation of the DNA. DNA extracted from the sample. It can be blood, it can be hair, it can be saliva. Blood, hair, saliva. But in blood, not RBC. Why not RBC? In the RBC, Baba, there is no nucleus. If at all you know, there is no nucleus. They have a lot of space for basically storing the hemoglobin. There is no nucleus that is going to be there. So not RBC, but in the blood, other cells are there which can be used, and blood, hair, follicle, saliva, and everything can be used. Then digestion by restriction enzyme, DNA is cut to the fragments. Then gel electrophoresis, where the fragments are separated on the basis of the size on the agarose gel. And finally southern blotting, where the fragments are going to be transferred to a nylon or basically nitrocellulose sheet. Then hybridization, where radioactive or fluorescent VNTR probes are bind with the variable regions are there. And finally visualization, where X-ray film or UV light reveals a unique band pattern compared for identity or relationship. Similarly, the same thing can be used for finding whether someone is the mother of some other person. Okay, some baby is there. So basically over here, I have a mother, I have another mother, and this is the child. So a mother and the child will not have the exact similarity, but they will be having so much things in common. They will be having so much things in common. So with the help of the same technique, we can find it. Okay, now in your NCERT there is one more thing that is given, VNTR, variable number of tandem repeats. So basically, as I said, repetitive DNA and things are there, right? So variable number of tandem repeats, as in basically it can be this 15 is there, here it is 5, here it is 5. So this is what you mean by VNTR. It's basically repetitive sequences. The term that you use is for telling that they are in variable number in different organisms. That's all. Okay, so if they can be asking you that define the VNTR method. Okay, it's nothing with the DNA fingerprinting method only, and it was given by Alec Jeffrey. So F for fingerprinting you can remember like that. Double F you have, and you also have IR, and you have everything together. So it's like fingerprinting. Okay, please remember DNA fingerprinting by Alec Jeffrey. F Sanger for DNA sequencing. That was used in HGP process. Cool. These things can be asked. So variable number of tandem repeats, the satellite DNA regions that probes latch into. That means basically, as I said, the unique thing that is there. So why did it go and attach up there? I told you. Why did it go and attach up there also? Because in them also they have that repeat, but in them it's not the same way that we have in us. Okay, the number could be different. That is why it's not going attach in every. It is going and attaching at every single location out there. Got it? That's all. So now you have some questions to do, and then we're going to stop it. My energy is puss, guys. Okay, so which of the following steps in DNA fingerprinting technique are labeled VNTR probes used? In which of the process do you use VNTR probes in DNA fingerprinting? During isolation of DNA, during digestion of DNA by REN. REN means restriction endonucleases, the one that cuts. Endonuclease means inside the nucleic acid they're going to make the cut. Okay, during electrophoresis, no. During hybridization, yes. Okay, during hybridization we are going to use it. During hybridization we are going to use it. Okay, hybridization means after you get it on the southern blotting on the nylon sheet, basically you're going to let the probes go and attach out there. Okay, in a crime investigation, the investigating officer collects different biological samples from the crime spot for DNA fingerprinting analysis. Which of the following sample is not helpful in this analysis? Skin shreds, erythrocytes, semen, or hair follicle? Semen helps is, hair follicle is, skin shred is definitely going to be. What about erythrocytes? Erythrocytes basically means what? It's your RBC. It's your RBC. RBC does not have nucleus, I told you. It has the whole space reserved for hemoglobin. So this is also not going to be the answer. Got it? Got it. Now, which aspect forms the basics of DNA fingerprinting? The satellite DNA showing high degree of repetition in DNA segments, the ratio of purines or pyrimidines, the amount of DNA found in samples of blood, saliva, and skin, the sequence of DNA present in the ridges and the grooves of the fingerprints. No, DNA fingerprinting relies on this thing, the satellite DNA showing high degree of repetition. We have that, and basically with the help of that we can try to find who is the criminal. Okay, it's not amount of DNA. If the amount of DNA is not useful for us, then there's no point, right? Basically it has to have something that is useful for us, and that is the degree of repetition that is there. Okay, that's all. Bond forming the sugar phosphate backbone. This is the last rapid fire. Bond forming the sugar phosphate backbone. 3,5 phosphodiester bond. Griffith's bacterium. Griffith's bacterium, remember, it was Streptococcus pneumoniae. Enzyme that joins Okazaki fragments, DNA ligase. The initiator codon, AUG. The lactopron inducer, lactose or allolactose. DNA plus histone octamer plus H1. Nucleosome. Measles and stall organism, E. coli. In hnRNA is transcribed by an RNA polymerase 2. Untranslated ends of mRNA. UTR. Okay, they are going to be known as UTR. Genes on the Y chromosome. You let me know in the comment section how many genes are there on the Y chromosome. And my dear children, we have completed each and everything that is there in the chapter. Thank you so much. Thank you so much, and please take care of yourself. I hope that this session had helped you in the best possible way. Wishing you the best, and if it did, I'm telling you again, do not forget to hit the subscribe button, and do check out the AIO page, all-in-one page given in the description section, where you will get all the things, and also join our WhatsApp channel. Bye and lots of love.